Hi everyone, am I audible? Hi everyone, am I audible? Am I audible? Hi students, if I'm audible or not, do let me know in the chat section. Okay, so let's start the class. I hope I'm perfectly audible to everybody. Let's start the class. So today we are going to start morphology of the flowering plants, and not just we are going to start it, we are going to finish it today. So are you guys ready? Yes? Are you people ready? So let's start the morphology. So very first thing that we need to know is what exactly is the meaning of morphology. So logy means study, morph means form. So here, what are you talking? You are talking about the, yes please, you can see that, you are talking about the study, right? Study of the external structure. So this is what we have to discuss. What is morphology? It is the study of the external structure, external characters, right? Like the size, form, shape, structure, color, etc. So when you talk about the external things of the plant, like when we talk about the externally, about the root, about the stem, about the leaf, we consider it in the morphology, right? So internal study is anatomy, external study is morphology. That's what you have to remember. So when it comes to the angiosperms, what are angiosperms? Angiosperms are the flowering plant. We know that even in the plant kingdom, we have discussed it. So you know that when you talk about the plants or the kingdom Plantae, we talk about algae, bryophytes, pteridophytes, gymnosperms, and angiosperms are there, right? And angiosperms are there. That is what we have. So when you talk about these angiosperms, they are the flowering plants, right? They are what? They are the flowering plants. So here in this case, you people will see that the flower will be there, and in that flower, in the female part, that ovule will ultimately form the seed. We are well aware of it, and that is why in the angiosperms the seeds are covered as well, right? That is why in the angiosperm seeds are also what? Seeds are covered, right? So angiosperms are nothing but the flowering plants, right? And they show the diversity in their size, in their habitat, in their lifespan, right? So overall, when you talk about these flowering plants, when you talk about the plant body, so it will be divided into root system and the shoot system. Is that clear? It will be divided into root system as well as the shoot system. We'll discuss this part later as well, right? About the embryo thing we will discuss later as well. But let me tell you, whenever you talk about a seed, okay, you know that that seed will germinate when the conditions will be favorable. It will form the plant. So in the seed, what exactly do you see? You see the seed coat, you see the embryo. Yes or no? You see the seed coat, and what do you see? You see the embryo. This is what we know, right? Seed coat is there, embryo is there, right? So when you talk about the embryo, in that embryo, the plumule, the part plumule, and there is a part known as radicle. So it is the plumule that forms the future shoot. Are you getting my point? It is the plumule that form the future shoot, and it is the radicle that form the future root. So from R, you can remember the word root. So plumule will form the shoot and the radicle will form the root. So here you can see when you talk about the root system, the underground part develops from the radicle of the embryo. This is what a root is. It develops from the radicle. And when it comes to the shoot, right, it is developed from the plumule. So very first thing that we have to start here is the root. We need to know about the root. What exactly are what is the meaning of the root? This is what we have to discuss. So we know that roots, they are underground. Yes or no? Yes or no? Yes or no? Tell me, students. So here what do you have? You have the root. So root is the non-green part. It is the underground vegetative part. It is non-green, it is underground vegetative. It doesn't possess nodes, internodes, leaves or buds. That's a very, very, very important feature. We know that underground portion is the root, and the one which is above the ground, that is going to make the shoot system, having leaves, having branches, right, having these branches, having leaves, flowers, etc. It's not like that modifications are not there, or stems, they are not underground. Stems are also underground, right? Roots are also they also show they are also, you know, little above the ground as well. We'll talk about that, we'll discuss everything there. But in general, whenever you see the root and the shoot, in the case of shoot, you are going to find the nodes and the internodes, right? What are you going to see? You are going to see the nodes and the internodes in the case of shoot. You all have noticed this part, like this is the node, okay? So the part here, this is another node. So the part here in between is internode, right? The part here in between is internode. So we have the nodes, we have the internodes here. That's what you have to keep in your mind in the case of stem. So when externally you have to differentiate a root and a stem, right? Let's suppose you got a question, right? Externally, when you have to separate what? Externally, when you have to separate the root and the shoot. I'm not saying check the internal structure. I'm simply saying just look at them. Externally, how can you differentiate that? Root will not have nodes and internodes, but the shoot will have. So this is a very, very important point. So roots, we know that they grow towards the soil. They are positively geotropic, right? We know that they are negatively phototropic, right? So these are the points, these are few points that you have to keep in your mind, okay? So root system, it arises from the radicle of the embryo. So these points clear? Yes, please. So roots are positively geotropic, negatively phototropic, and even they are positively hydrotropic as well. Am I clear? They are positively hydrotropic as well. So these are the features of the root. And here you can see this particular diagram. This is showing the regions of the root. We will discuss that soon. So here you know it already that it is the radicle, right? It is what? It is the radicle that will give rise to root. Am I clear? It is the radicle that will give rise to what? That will give rise to root. Now, when you talk about the root, let's see. We all have seen this thing, right? Where from radicle, this primary root will form. Then secondary, tertiary, root hairs are there. Like this well-developed system, I hope you all are aware of it. Yes, please. You all are aware of it. So this type of root system is the taproot system. So in the case of taproot system, what you people will see, in the case of taproot system, you guys are going to see that there is a primary root, and there are secondary roots as well, and there are tertiary roots as well. Root here are also present. So when this root system is that well developed and it is arising from radicle of the embryo, then you are calling it as the taproot system. What do you call it then? You call it as the taproot system. And we have another category of root, that is adventitious roots. So adventitious roots, right? Adventitious roots, they arise from any part other than radicle, right? They arise from any part other than radicle. So if a root is developing from any part other than radicle, that is, it can be a stem, it can be a leaf, right? Roots can arise from the stem, roots can arise from the leaf. So as I said, any part other than radicle, right? Any part other than radicle, then what will you call it? You will call it as the adventitious root. So they arise from any part other than radicle. So there is one more type of root system, that is the fibrous root system, okay? So if you follow the new NCERT, you are not going to find this portion there. This is the part of your syllabus. The modification of root, shoot, even the leaf modification, they all are the part of your syllabus, but they are not given in the new NCERT. They are given in the old NCERT. So yes, you have to revise them from the old NCERT. Is that clear? And you can just follow this particular PDF as well. So now there is one more type of root system, like fibrous root system, that you see in the case of what? In the case of grasses. So here what will happen? You will see that there will be, you know, bunch of roots. Right, there you'll see the bunch of roots here. They are not very deeply penetrated into the soil, and here you'll see primary root will be there, but that is going to be short-lived, right? That is going to be what? That is going to be short-lived. Is that clear? Is that clear? So there is a bunch of root, Primary root is going to be the short-lived one, right? In the case of grasses, you see, and this fibrous root is also the adventitious root. Why? Because it also rises from the base of the stem. So this is the root system that you have to keep in your mind. So just have a look. So here you guys can see these types of root systems. So taproot system is there, fibrous root system is there, and then comes the adventitious root systems, right? So taproot system, in majority of dicots you will see this system. There is a direct elongation of the radicle that will lead to the formation of primary root, which grow inside the soil. Is that clear? Which grow inside the soil. Is that clear? So it bears lateral roots, which are secondary, tertiary, right, which will narrow down at the tip. Obviously, I believe you all have seen the roots. So primary root and its branches, they are making this taproot system. So in the dicots you will see, so here they have given the example of what? They have given the example of mustard plant. Is that clear? Is that clear? Then fibrous root. So mainly in the grasses, I have given you the example of grass. So grass is a monocot, right? So in monocotyledonous plants, the primary root is short-lived and is replaced by a large number of roots. Primary root short-lived, replaced by a large number of roots. So they are underground roots that arise in groups either at the base of erect stem or the nodes of the horizontal stem. Am I clear? Then the main root are of equal length, give off small branches. So they are thin and thread-like. So they are the fibrous roots. So they do not penetrate deep into the soil. Okay, they do not penetrate deep in the soil. Am I clear? Am I clear? Then comes the adventitious roots. So they arise from any part other than radicle, right? So they can develop from the hypocotyle. One by one, you'll understand these words. From the stem base, from the stem nodes, from internodes, from leaves as well. I'll give you one example, like in the case of rose plant, what can you do? How can you regrow it? You can just take the cutting of that rose plant and you can just grow a complete plant from that, right? Right, you know that. So from that stem cutting, the roots will arise in that too. So adventitious root here also, grasses, they also show that. Monstera is the example, banyan tree is the example. One by one, you'll discuss that. So in some plants, when branch and leaf cuttings, they are placed in soil or they are partially submerged in water, they can develop such adventitious root. These are the examples. So rose, sugarcane, etc. They are the examples. So this line again, and look at this particular diagram. So this is the fibrous root, the bunch of roots, not deeply penetrating in the soil. So primary root is short-lived here, right? And this is the taproot, proper primary root, secondary root, tertiary roots are there, rootlets or root hairs are present there. So here you can see that fibrous roots are adventitious in nature. They originate from the base node of the stem. They do not arise from the radicle directly. Is that clear? They do not arise from the radicle directly. Is that clear? Is that clear? So see, right, in this particular plant, right, from this part, roots are arising. In that cuttings, you will see the roots. So they are the example of adventitious roots. So what roots are going to do? We know it very well. Roots, they help that plant to get attached to the substratum, right? So we can say that anchorage, right, that is this fix the plant and support the aerial shoot system in absorption and transport of water, right? In absorption and in the transport of water, it helps. In the prevention of soil erosion, it helps. In the storage, it helps. Extra mechanical support, nitrogen fixation, reproduction, photosynthesis, and even in the synthesis of plant growth regulators, it helps. So yeah, root helps in the synthesis of plant growth regulator. Important it is. So remember, roots, they synthesize cytokinin. Is that clear? Is that clear? So here you have taproot, fibrous root, and adventitious roots are there. Understood? Now, what we have to discuss, we will talk about the regions of root. What's the next topic that we have to complete? It is the regions of root. So when you look at the root part, so roots, they are going to penetrate in the soil, right? They are going to penetrate in the soil. So in the case of root, this part is actually the root tip. That is the apex. And this part here is the base. Are you getting my point? This part which is going to penetrate in the soil, this is showing the root tip, and this part here is showing what? This part here is showing the base. Okay, so here you are going to see some regions. First of all, we all know it very well that in the case of plants, they grow throughout the life. They grow continuously. Why? Because in the plants, we know that meristematic tissue is there. Even in the anatomy part, we have discussed it. Yes, meristematic tissue is there in the plants. We know it very well. And we know that it is the group of actively dividing small-sized thin walled compact cells, isn't it? That's how it is. Small size, thin walled, compactly arranged, with dense cytoplasm, with large nucleus, with abundant plasmodesmatal connections, that is continuous cytoplasmic connection. We know that meristematic tissue, it is present in the case of plant. Where is it present? It is present in the case of plant. We are well aware of it, isn't it? So in the case of meristematic tissue, especially when you talk about the primary meristem, what's that primary meristem? That primary meristem helps in primary growth of the plant. That is, I would say, increase in length. Okay, increase in length. It is going to help like that, isn't it? Isn't it? Okay. Sorry. So now here in that case, we know that the meristem is present at the tips, right? So if it is present at the tips, we will call it as apical meristem. And you know that you have shoot apical meristem and the root apical meristem. Why am I explaining it here? When you talk about the root, right? When you people talk about the root thing, right? You know that these are the regions of root. That's how we make the diagram, right? So here you will see that there is in the root also, there is a region where you will see meristematic activity, or I would say it is a zone of meristem. So that's what you guys are going to see. And here what do you have? You have root cap. What do you have here? You have the root cap. So that's what you have to keep in your mind, that in the root also we will see the root apical meristem, but it will not be directly at the tip. It is subterminal. What's that? It is subterminal. So there is a root cap which is a thimble-like structure. There is a root cap which is a thimble-like structure, covers the tip of root. It's a thimble-like structure that do what? That is going to cover the tip of the root. So it is having, right, subterminally. Here you will see the region of meristematic activity or meristematic zone is there. So it means here you will find the cells which will keep on dividing. Okay, so proximal to it, that is nearby to it, you will find the zone of or the region of elongation. So basically these meristematic cells, what they do, students, these meristematic cells, as I said, they will just divide. They are actively dividing cells. Now in the region of elongation, these cells, like some cells, they'll keep on dividing, but some cells, they'll show the increase in the length, right? Some cells, they'll show the increase in the length. In their case, you will also see the vacuolation will be there, that is vacuole formation will be there, right? You'll see the cell will increase its length, right? So initially in the meristematic zone, we know that the cells, they were just dividing, but out of that, some cells, they'll start growing as well. They'll start differentiating as well, right? Differentiating means they'll start acquiring a permanent fate as well. So what will happen here, guys, you people can see this very clearly, that here there are some cells which will increase their length, right? They'll show what? They'll show the vacuolation, right? They'll show what? They'll show the vacuolation. So this is the region which help in increase in the length, okay, in the length of the plant. That is how it helps. Now after that, what will happen? So the cells, they were elongating, and finally what will happen? These cells, they will become mature. So we know that there will be the, so next to it is the maturation zone. Next to it is what? It is the maturation zone, and here in the maturation zone, you are going to see the root hair. Am I clear? What are you people going to see? You people are going to see the root hair. Is that clear? Is that clear? So you have, this is something you have to keep in your mind, right? The increased vacuolation is there. The cell, it will become mature. Am I clear? The cell, it will become what? It will become mature. It will basically acquire a permanent function. So that is why here in this maturation zone, you will also see root hair are developed. They develop from the epidermis. Why? Because obviously there is a particular function of the root here. The cells, they are divided, right? They have a proper function as per the position. Like those who are at periphery, they'll form the epidermis like this. So from that epidermal cells, these hair-like appendages will grow, right, beta? So they are unicellular and they will help in absorption, right? Right, they increase the surface area as well. So these are the regions of root. So what you have to remember ultimately, if somebody will ask you that what will be the sequence of these regions from the tip to the base, then what will you see? Region of meristematic activity, region of elongation, region of maturation. And if somebody will ask you the same sequence, right, but how? From base to apex, then what will you see? Then first will be your maturation zone, a region of maturation or maturation zone, then comes the elongation zone, and then there will be the meristematic zone. So ultimately you have to read the question carefully, right? So what exactly are they asking? That's what you have to focus. Am I clear? That's what you have to focus. So here you have root cap, meristematic activity region, elongation region, maturation region having the root here. So that is again a previous year question. Okay, that in which region you will see the root here, you know that it is the maturation region. So let's read that points here. So here you people can see, the root is covered at the apex or at the tip by a thimble like this, thumb-like structure, which we call as root cap, which protects the root meristem. So here the cells, they are also having the starch grain, right? That will help in this geotropic responses here. So cells of the root cap, they also secrete, beta cells of the root cap, they also secrete the mucilage here. They also secrete the mucilage here, which reduces the friction as it makes its way through the soil. Is that clear? The cells of the root cap also secrete mucilage that will reduce the friction. And one more point that you can write, that in aquatic plants, that is hydrophytes, root, instead of root cap, root pockets are present. That help in buoyancy. Am I clear? That help in buoyancy. Am I clear? So that's what you have to remember. So region of the meristematic activity, that is the growing point. So this is few millimeters above the root cap. Okay, this is just few millimeters above the root cap, right? So it is 1 mm long region. Cells here are, the properties are very important. Even in plant growth and development, you talk about it. They are very small, they are thin walled with dense protoplasm, right? They divide repeatedly, they produce new cells for the root cap as well as the basal region of the root. Am I clear here? Yes, please. Am I clear here? Done. So let's focus on the next part. So then comes the region of elongation. So it is 4 to 8 mm in length. So here the cells, right, because this is the region of elongation. So here cells, they have lost the ability to divide. So in the meristematic zone only, right beta, in the meristematic zone only, you people will see that the cells are actively dividing. But here you guys are see that the cells of this region are newly formed and they lost the power of division. Here they undergo rapid elongation. Right, so they increase their length. They will elongate here, and the enlargement and are responsible for the growth of the root in length. So overall, increase in length of the root. For increase in length of the root, this region is responsible. Am I clear? Please have a look. Am I clear? Then comes the region of maturation, right, which is also the root hair zone. So different types of tissues, right, different types of tissues, that is xylem, phloem, cortex, etc. They differentiate and mature, right? I think this thing I have explained multiple times, that always remember DM. That is, first there is the differentiation, then there is the maturation. So due to this differentiation, a cell is acquiring its permanent fate, right? So here what is happening, different types of tissue, be it xylem, phloem, cortex, etc. they differentiate and mature in this region. So hence this zone, which is proximal to the region of elongation, it is the region of maturation or differentiation. So some of the epidermal cells of this region, they form very fine, right? Here in maturation zone, they'll form very fine and delicate tubular outgrowths, which we call as root here. So they absorb water and mineral from the soil. Am I clear? They absorb water and mineral from the soil. Am I clear? Am I clear? So here this mature cells, they obviously they form the bulk of the root, right? Here the cells, they will not undergo any further changes, and the lateral roots arise from the interior of this region. Beta, you will soon get the third part of the anatomy on the channel in the recorded format. So there you will learn. Just we have the pericycle there, endodermis, pericycle. So from that pericycle, these lateral branches, they arises in the case of roots. So in the case of root, the branches, they are endogenous, like they develop from the internal structure. But in the stem, that branches, they arise externally. So we call them as exogenous. Am I clear? We will call it as exogenous. Am I clear? Am I clear? Done, beta? So now we have to talk about the modifications. So those who are just preparing for the boards, so do check your syllabus that whether you have the modifications or not, because it is deleted from the new NCERT, but it is still there in the old NCERT. So modifications are the part of your NEET syllabus, right? Right? In schools, teachers, they miss this, right? When they teach the 11th and 12th portion, just the 11th portion, board portion, they delete it. They don't teach it, but it is actually important, right? And it is a part of your NEET syllabus, the modification. So please keep it in your mind. So taproot modifications, may you have fleshy taproots, nodulated, and the roots for respiration. Very, very, very important. So I'll give you an advice. You have to make this flowchart. Just give the title modifications of taproot, because if we have to discuss modifications of taproot, we will also discuss the modifications of adventitious roots as well. So what you have to do, make the flowchart. So morphology is something where you have to cram a lot of things. You have to remember many examples. So I will suggest you to do this particular thing, guys, right? I'll suggest you to do this particular thing, and that is ki make the flowcharts, write short notes here with the class itself, write the running notes, so that you can later use them for the revision. Okay, so have a look. Fleshy taproot. So here you have conical, fusiform, nepiform, and tuberous. Okay, so see this. So fleshy taproot, when their word is fleshy, so basically they are swollen. And why is it so? Because they'll store food, right? So fleshy taproots, right? These taproots, they will become swollen and fleshy, right? They will become swollen and fleshy with the stored food, right? Why are they? They are with the stored food. So secondary roots, they will remain thin here. Stem is reduced and discoid in the beginning and bears radical roots. Am I clear? Am I clear? Done. So next point that we have here is conical root system, right? What do we have here? We have the conical root system. So from this conical, from C, cone-like structure, of course, remember the example carrot. So what's the scientific name? Decus carota. Okay, so try to remember the scientific names as well. Am I clear? So this root resembles a cone-like structure. It is thick towards the base. Just imagine a cone. Just imagine a carrot. So it is thick towards the base and gradually it is tapering towards the apex side, right? Thick towards the base. This part is basically the base, and it will be tapering towards the apex. So it has thread-like secondary roots, obviously, right? Example is carrot. So C for conical, C for carrot. That's how I remember it. Is that clear? So next word is the fusiform. What's the meaning of fusiform? We know that spindle-shaped roots they are. Fusiform means spindle shaped. So spindle shape is something like that, right? That this part will be thick, ends are tapering. Can you see that? So thickest roughly in the middle, narrow towards both base and apex. So here the example is reddish. What is the example here? The example here is reddish, that is Raphanus sativus. Then comes the nepiform, turnip, and the beetroot is the example. Okay, so nepiform. So remember the examples. Then the shape will be clear. So you all have seen the turnip, right? Or either the beetroot, very healthy. So we know that here it is thick at the base, almost spherical, and it will become thin towards the apex. Right, so secondary roots also develop in the pointed apical region. So turnip and the beetroot, it is the example. So turnip, it is scientific name is Brassica repa, right? Repa ya Nepus? Then beetroot, Beta vulgaris. Done, beta? So scientific names, I'll suggest you to remember the scientific names. Is that clear? Is that clear? Sure? Done, beta? So Beta vulgaris. Done? Okay, so next is the tuberous roots. So they do not have a regular shape. They are swollen and fleshy at any portion of the roots. So example is Mirabilis jellipa, that is the 4 o'clock plant. Even in the genetics we study about that. Okay, so these are the modifications here. So look at this. In the NCERT also they have given you the example for the storage, right? B is for the respiration. So respiration mein here you guys can see, right? These are the respiratory roots. Right, here you talk about the pneumatophores. So pneumatophore, that is a PYQ, and I've seen many students making the mistake here. So it's a taproot modification, not the adventitious root modification. It is the taproot modification. What's that? It is the taproot modification. We will talk about it. So see, the sweet potato is also given. Yes, sweet potato is the root modification. But here they didn't specify taproot or the adventitious root. That I am specifying you. It is the adventitious root modification. Okay. Okay, chaliye. So this is the conical carrot, the fusiform reddish, and then this is the beetroot and tuberous roots in the case of Mirabilis jellipa. Okay, so next is the nodulated, nodules, mane some condensed structure, right? Some condensed structure, nodules, isn't it? I hope you know that. So nodulated or tuberculated roots, so they are the secondary, tertiary, and sometimes even the primary root. They develop numerous, means many, irregular swellings called root nodules, right? Even in class, I think in 6th, 7th or 8th, we used to discuss now root nodules of leguminous plants. And root nodules of leguminous plants. You remember? They are having a nitrogen fixing bacteria, rhizobium. So yeah, so these nodules have nitrogen fixing bacteria, jaise leguminous plants, your soybean, groundnut, pea or gram. So nodulated roots they are. Okay, so they have these root nodules in which there is the bacteria will live that will fix the nitrogen, jaise rhizobium. Right, so it is a nitrogen fixing bacteria, but it will only fix the nitrogen when it is here in this symbiotic association. So rhizobium, it is not going to fix the nitrogen in its free living state. Understood? Right, so the next is pneumatophore, very important. So remember that pneumatophores, they are the modification of the taproot but not of, so it's the modification of taproot. Okay, not of adventitious root. So this is important to remember. Understood? So these are the respiratory roots. They are found in plants that are basically in swampy areas, that plant that grows, right, where the salinity is high. So basically in mangroves or in saline, right, swamps, they will grow. So here roots, what will they do? They grow basically not positively, but these pneumatophores are negatively geotropic. Do you know that? They are negatively geotropic, right? They will not grow towards the soil. They'll grow against it, like something like this, if I have to show you, right? Just I visited the Kerala. So in Kerala, I've seen mangroves there, right? There I noticed these pneumatophores. So they have this pore-like structure, which is known as lenticels or pneumatodes. Right, lenticel or nematodes, through which the gaseous exchange will occur. Right, so these are so they are what? They are negatively geotropic, not positively geotropic. Please keep it in your mind. Am I clear? Am I clear? Sure. So they come out of the water for the exchange of gases. So they have small pores called lenticels, so rhizophora, or even Soneraceae is also the example. Okay. Next is the modification of adventitious roots. Okay, so this part, the mechanical support part, is very, very, very important here. And epiphytic, reproductive, parasitic, these three are also very important here. Okay, so when it comes to the modifications of adventitious root, we have the fleshy roots, right? We have the fleshy roots, we have the storage of food here. So tuberous roots, fasciculated, fleshy, palmate, and nodulose is there. Okay, and here in the mechanical support, you have the prop roots, you have the stilt roots, you have the climbing roots, you have the buttress roots. Vital functions may assimilatory, that is photosynthetic. Yes, you heard me right. Some roots are also photosynthetic. So assimilatory, parasitic, epiphytic, floating, or reproductive roots are present there. Is that clear? So if it is fleshy, one thing is for sure that there will be the storage of the food, right? So if it is fleshy, what is sure? That there will be the storage of food. Okay? Done? So just have a look. So here sweet potato, that is Ipomia batatus, and remember, so it is very important. So sweet potato is the adventitious root modification, right? Again, very important. Like your pneumatophore is very important. This is also important. So no doubt, sweet potato is a root modification, but it is the adventitious root modification. So what are adventitious roots? The roots that arise from any part other than radicle. So here I'll mention one important question. Right, here I'll mention one important question. You all can write it down. This is sweet potato is what? Root modification. Potato that we eat is a stem modification. So they both are the examples of analogous organs. Do you know that? So what are analogous organs? What are analogous organs? So in the case of analogous organs, you people will see, right, that they have different structure but same function. Done? So one is root, one is stem. Structure is different, but the function is same. What is that? Their function? Storage of food. Okay? So they are the example of analogous organ. Keep it in your mind. This is important. Okay? Done? So next is the fasciculated fleshy root that we see in dahlia or in the asparagus. So these are the swollen roots or the root tubers. Right? Swollen roots or even the root tubers, they are in clusters. Clusters means they are in groups. Right? They are in clusters means they are in what? They are in groups. So in dahlia, you can see this here. So in dahlia, fasciculated roots, right? Fasciculated roots, they lie at the base of the stem. Am I clear? They lie at the base of the stem. Am I clear? So in asparagus, right? In asparagus, the fasciculated fleshy roots occur at intervals on the normal roots. Done, beta? Done. So dahlia and asparagus, they are the examples. Then palmate root, not so important. So obviously it's palm-like. So it is in the case of orchis, right? And then nodulose roots are there. This one you will see in the case of mango ginger, right? What am I saying? Mango ginger or in the turmeric. And mainly you remember the mango ginger because turmeric you will also study in rhizome. You might get confused. So remember the mango ginger. So here there are near the tip of the root, there will be the swelling. So see the sweet potato, right? Again, very healthy it is, right? Ipomia batatus. So fasciculated root dahlia, fasciculated root asparagus. So you can see they are in the groups, they are in the clusters. So these are the branches of that palmate fleshy root, that is the orchis. This part is extra, you can leave it. And this is turmeric as well. So yes, it's not mandatory that one plant will only show one modification. It can show more than that, but here, right, it is showing the nodulose roots. Okay, now this is very important modifications for the mechanical support. So the number one thing is prop root or the pillar roots, right? The best example of it is the banyan tree, right? Ficus bengalensis or bengalensis. This ficus species basically. So these are the prop or the pillar roots. So these roots, they arise from the branches of the plants. They grow downward, right? They are just like if it is providing the mechanical support, so you can understand it, it is basically doing what? It is basically supporting, right? It is basically doing what? It is basically supporting. So from the branches, they are growing downwards. So obviously it is not arising from the radicle, right? It is not arising from the radicle. So definitely it is the example of the adventitious root. So they grow towards the soil and hang in the air. So as they reach the soil, they will become thick and pillar-like, and they'll give the extra mechanical support, right? So example is the ficus, that is the banyan tree, right? Ficus species it is. Okay, then in the rhizophora, even in that mangrove plant also, you'll see prop roots are there, right? So you all, so see this. So in our village, we have a very big, very, I would say very large banyan tree there, right? So that tree, it shows that prop roots. So see, they look like pillars, okay? They look like what? They look like pillars. Is that clear? Okay. Okay. Sure. Then comes the stilt roots. So let's look at this. These are the stilt roots. These oblique roots arises from the base of the stem, right? In sugar cane, you can see that. In maize, you can see that. Okay, so see this stilt roots, also known as brace roots. So they are short, we have seen that, but they are also thick supporting roots. So they are arising obliquely from the basal node of the stem. So they are arising from the stem, obliquely they are arising, right? Obliquely they are arising. So after penetrating the soil, they will form the fibrous root. Important it is. So firstly, they are arising from the basal node of the stem. So they'll penetrate the soil, they'll form the fibrous root, which hold soil firmly to provide the support. Example is sugarcane and the maize. Is that clear? Is that clear? Okay. Then clinging or the climbing roots, right? Clinging or the climbing roots. So just have a look. These are the clinging or the climbing root. In the money plant also you see that. This is sometimes, you know, some these plants which are having these weak stems. So from their part, from these stems, you find some roots which will help them for the attachment to a surface. So these are the clinging roots. And here, some trees you might have noticed, like their roots, they just come, they are just above the ground like this part. I hope it's clear in the pictures. Right, I hope it is clear in the picture. So this is what? This is a buttress roots in the people, that is Ficus religiosa. Also we see this. So clinging or the climbing root, they are non-absorptive. They are not going to absorb anything, right? They are found in the climbers. So they may arise from the nodes, internodes or both, and help in plant climbing. So every plant, money plant, they are the examples. And then comes the buttress or the plank roots. Right, buttress or the plank roots. So they occur in large trees. They arise from the basal part of the main stem. So they are horizontal and travel along the ground to provide the extra support. So people, in the case of people tree, we see that. So people, scientific name is Ficus religiosa. Understood? Done? Mr. Madesh Made, how much money you are getting for doing all that stuff? Huh? Right? Matlab, I don't know why students are having obsession with the series name. You want a chapter, the chapter is there. A teacher is teaching you. What else do you want? Why are you so obsessed with the series name? Array, that series is incomplete, that series is completed. You check our channel, you will get all the content. Then what's the big deal? I don't know why you, like, I don't think that you people are having any life. You are just here, you're just free and just spamming here unnecessarily. Useless people. So next is modification for the vital functions, right? So here comes the assimilatory roots, which are also known as photosynthetic roots. Right? So these are green roots which are capable of photosynthesis. Right? I still remember these examples, Trapartinospora. There is one more that I'm not able to recall because I always used to revise Trapartinospora. Right? We have studied that in our graduation as well. So assimilatory roots, but a Tinospora and the Trapa is there. Tinospora and the Trapa is there. Then parasitic or hostorial root. See, this is even in the ecology. Cuscuta. Cuscuta is a total stem parasite. Cuscuta is what? Cuscuta is a total stem parasite. Cuscuta is also known as dodder plant or amarbale. So, like in some trees, you might have seen this this vein, yellow colored, right? Yellow colored. It looked like a wine, right? It looks like a wine. But actually, what is it? This is basically Cuscuta. This is basically what? This is basically Cuscuta. So here you will see that it is a total stem parasite. It will take all its nutrition from this host plant. So it is having the hostoria, which will grow in that host plant, and it will take out nourishment from that. So here you guys can see, right, the parasitic or the hostorial roots. So these roots, these roots occur in the parasitic plants for absorbing nourishment from the host plant. Is that clear? So these roots, they occur in the parasitic plants for absorbing, right, nourishment from the host plant. So they are called the sucking roots or the suckers. Example is Cuscuta, total stem parasite it is. So they make connections with both the xylem, right? They make connection with both xylem as well as the phloem, right? And absorb water and the food. Understood? Absorb water and the food. Understood? Done. So next is epiphytic or aerial or hygroscopic roots. See, this is again something very interesting, I'll tell you. Just say, I'll give you an example. Let's suppose this is a branch of a mango tree. So on it, we see sometimes an orchid named as Venda, it grows, right? That's the scientific name, right? Venda, it grows. Now what's happening? It is having the root, we are calling them as epiphytic root, right? Epi means above, phytic mane above plant. So basically this plant, it will grow on a branch of a tree, number one, right? And it is having the epiphytic roots. Actually, what's the point? These roots are having dead spongy tissue. This is very important, please highlight it. Important for 2026. So it is having dead spongy tissue called velamen. It is having dead spongy tissue called velamen, and this velamen is hygroscopic in nature. What's that? It is hygroscopic in nature. So what is the meaning of being hygroscopic? So it absorbs moisture. Am I clear? It absorbs what? It absorbs moisture, right? So this plant, the roots, they are not taking anything from the plant. They are not taking anything from this mango plant. Are you getting my point? But it is having these roots, they can absorb the moisture from the surrounding because they are hygroscopic. Why? Because of this velamen. So it is a dead spongy tissue present in the roots. Very important topic. And such interactions where one is getting benefited, right, and other is neither harm nor benefited, this is commensalism, right? So it is an example of commensalism as well. In the ecology also you discuss that. So it is the example of commensalism. Is that clear? Sure. Done, beta? So epiphytic or aerial roots, so roots are thick. They are irregular, they hang down in the air, they do not have the root caps and root hair. This is important to understand. They do not have the root cap and the root hairs, right? They do not have root cap and the root hair. So instead they possess a covering of dead spongy tissue which is known as velamen, right? Which is known as velamen. Is that clear? Is that clear? So with the help of this velamen, the epiphytic roots are able to absorb the water from the moist atmosphere. So orchids such as Venda, this is the example, and I told you it is showing the commensalism. Understood? Next, floating roots, helping in floating, so definitely be there in the aquatic plants, right? So these roots, they store air, they project out of the water, they make the plant buoyant and help the plant floating on the surface of water. So it helps in the gaseous exchange. Am I clear? It helps in the gaseous exchange. And then comes the reproductive root. So reproductive roots, what will they do, beta? With the help of these roots, we can grow a new plant. Is that clear? Is that clear? We can grow a new plant because of these roots. So here you can see these adventitious roots. They are generally fleshy, right? They develop adventitious buds, and that buds can grow into a new plants under the favorable conditions. So sweet potato, that is also showing what? This, right? These adventitious buds, and even dahlia. So this is, if you will solve last 20 years ka PYQ, you'll get it. You'll find this question. Okay, you will find this question. Understood? So in some aquatic plants, they develop only for the balancing. These are hydrophytes, obviously, pista lamina, and in some for the fixation, that is hydrilla. So this is all about the root. Am I clear? So root done. So I don't know why you people just keep complaining, ma'am morphology, root is already done. Hai na? In less than 1 hour, we are done with it. Yes or no? So this is all about the root, and how can you revise it? Just open the flow charts and pick up the example. Like in the taproot, modifications of taproot meant for storage of food, conical, carrot, like this. Keep on revising the examples. And if you think, ma'am, you are also telling us extra points, just first of all, they are important. Second, download the old NCERT, revise it from there. Understood? So the next part that we have to discuss is STEM. So before starting the STEM, because you have to cram the things, just rewind the lecture a bit, right? And keep on recalling this. So STEM we will start after a break of 5 minutes. Okay? 5-minute ka break. Then we will start the STEM. So quick, recall it. Quick everyone. Right? And try to make the short note. So the next part is of what? Next part is of stem. So when you talk about the stem, you know that it is the ascending part of the axis that is bearing branches, leaves, flowers and the fruit. So in the stem, if here they are present, they are usually multicellular. And let me remind you that what do we call stem here? We call them as trichomes. What do we call them? Trichomes. So they are usually multicellular but can be, right? But they can be what? They can be unicellular as well. Understood? They can be unicellular as well. Is that clear? Right? Okay, Komal, what happened is ki ma'am, okay, there must be some confusion, beta. Your sweet potato is Ipomia batatus, but there the beetroot is Beta vulgaris. Right? You can just correct it. Right? You can just check it, or you can just correct it. So basically, sweet potato is your Ipomia batatus, but your beetroot is Beta vulgaris. You can just check it. So now just look at it. The stem is generally green when young and later often becomes woody and the dark brown. Is that clear? Right? So we were talking about the trichome. So I told you they are usually multicellular. Right? They can be single-celled. They can be soft or stiff, branched or unbranched, glandular or non-glandular. Is that clear? So this is basically the stem here. How do they help? Like root here help in the absorption. So it prevent loss of water in the form of, it prevent transpiration. It prevent the loss of water in the form of water vapors. Am I clear? So the stem is generally green when young, later will become woody and dark brown. We know that positively phototropic it is going to be, right students? Negatively geotropic and hydrotropic it is. So I hope this part is clear. Any doubts? Any doubts? Okay, so characteristics of stem. So stem, we have already discussed that stem basically it arises from what? It arises from the plumule of the embryo. So in that germinating seed, in that seed embryo, radicle will form the root and plumule will form the shoot. Is that clear? Is that clear? So stem is, as I said, negatively hydrotropic, positively phototropic, negatively geotropic. So it always show the distinct nodes and internodes. A very important point, on the basis of that you can just differentiate the root as well as the stem. Am I clear? On the basis of which you can differentiate the root as well as the stem. Is that clear? Is that clear? Yes? Done? So the region of the stem where leaves are born is called a node, while internodes are the portions between two nodes. Right? So regions of the stem where leaves are born, it is called node, while internodes are the portion between two nodes. So stem bears bud. That buds can be terminal, that can be axillary. So some fleshy buds, they take part in the vegetative reproduction and they are called the bulbils. Bulbils, sorry, not bulbilis, bulbils. Okay? So let's talk about it. So form of stem mein, the aerial stems are also there, subaerial are there, underground are there. But before that, let me tell you one thing. Now when you talk about the stem, if that stem is unbranched, right? Like let's suppose if that stem, this is the stem is unbranched, right? Let's say here you have that all the leaves or something, yeah, branches, but overall the stem is what? This is unbranched. So this type of stem is known as caudex. This type of stem is known as what? It is known as caudex. Is that clear? Is that clear? So in plant kingdom also, but not in the case of angiosperms. Here so we are talking about the angiosperms. So in plant kingdom we have gymnosperms, and that gymnosperms, and there we discuss the example of cycas. So cycas is having unbranched stem and we call that as caudex. But remember, that is the gymnosperm. So even in angiosperm, like in general, whenever you say the stem is unbranched, you will call it as caudex. Now the another example, that is of bamboo, right? I hope you know what bamboo is. So in the case of bamboo, you know that internodes are hollow. Internodes are what? internodes are hollow, but nodes are solid. Internodes are hollow, but nodes are solid. Is that clear? Internodes are hollow, but nodes are what? They are solid. So such type of stem is known as culm. Right? Such type of stem is known as culm. Okay. So now in the stem also, aerial stems are there, right? They are usually upright, my dear students, right? They are erect as well as weak. They are erect. They can be weak as well, right? So they can be branched or unbranched. So weak aerial stem are either, what do you call them? Twinners or climbers. Twinners or climbers. Understood? So subaerial, right? I made some mistake. Oh, my body is demanding a cup of tea, hai na? Wait, I'll get it. So this is what we have. Next is subaerial. So here stems are weak. They take support of the ground to spread because this is subaerial. Here they will have trailers and creepers. So though in that much detail they are not going to ask you the question, but just for your knowledge I'm telling you, twiners and climbers, they are the aerial modifications. Your trailers and creepers, they are subaerial. So creepers may the stem, right? Let's suppose there is a stem, right? Like it is attached to the ground or a wall or something. So at the nodes you will see that the roots are coming out. Is that clear? Roots are coming out. We will discuss their examples also. Then comes the underground stem modification. So if it is underground, that is obvious. If the stem is underground, like your potato, so it is going to be non-green. It will store the food. It lie below the surface of soil. So in few monocots with underground stem, aerial shoots develop only for bearing flowers. Yes, in monocots, this is what you see. And what these underground stems will do, they will act as organ of perennation. So what is that organ of perennation? So when we say organ of perennation, basically it will help to survive in the adverse condition, right? Right, it will help to survive in the adverse condition and when conditions are favorable, because they have the stored food. So when conditions are favorable, a new plant will grow from it. Okay, a new plant will grow from it. So that's what you have to keep in your mind. Am I clear? Right? So stem overall, we know that, right? It will just spread out. It will bear the branches, leaves, fruits, flowers. So conduction, it conduct the water and mineral, right? From roots to leaves and fruits. So it transfer the food manufactured in leaf to other plants. Now the addition of new cells. So stems, they also add new cells and tissue that will help in the continuous growth and the functioning of the plants. And moreover, stems, they also perform the function of storage, right? They help in the climbing. They are modified into the tendrils. They help in climbing, and they also get modified into thorns. They help in protection then, and even they help in the vegetative propagation. So these are the different functions of the stem. Now modifications may underground stem modification. Here I'll specify one thing. Like if you will read it from the old NCERT, you will find that runner, they have mentioned it as underground stem modification, right? So just keep it in your mind. Otherwise it is subaerial, but in old NCERT they have mentioned it as the underground stem modification. So underground mein rhizome, tuber, corm and bulb. Do read that, right? And here stolen, offset, sucker, even runner is subaerial, but in old NCERT they mentioned the underground. Aerial mein tendril, thorns, phylloclade and cladodes are there. So these two are very important. If you solve the PYQ, you'll get the question on the thorns as well, right? And for rhizome, corm, bulb and corm, rhizome, tuber, bulb and corm, let me tell you beta, in the NCERT they didn't use the term. They have given the example like zamikand is given, right? Your ginger is given, but they didn't use these terms. So we need to remember that. Okay, right? So I told you this thing that in the form of stem, we talk about the caudex and the columnar. Right, what we discuss here, caudex and columnar. So unbranched, so what are they? They are unbranched, they are erect, they are cylindrical stem and marked with scars of fallen leaves, right? So they are unbranched, erect, cylindrical stem and marked with the scars of fallen leaves. So culm, jointed stem with solid nodes, but the internodes are hollow. Example is bamboo, right? Example is bamboo. Okay, so the next part here is underground stem modification, right, which is used for the food storage as well as perennation. So perennation means it will help, right, to survive the unfavorable condition, and when the conditions are favorable, because these underground stem, they have the stored food, so new stem will arise from it. Is that clear? New stem will arise from it. So rhizome is the word here. So it grows parallel or horizontally underground. This is important, right? How this rhizome, so rhizome mein example, very important, ginger, banana. So banana is, and it's not mandatory that one plant will have only one modification. It can have more than that. Even turmeric also comes there, right? Don't get confused, but ginger and banana, they so for sure you have to remember, right? Even the turmeric part. Okay, so they has nodes, internodes, scale leaves and buds. Rhizome. So you all have seen the ginger, right? You all have seen the ginger. Done? Right? So this is what you have to remember. So next is tuber. So swollen, right? Tuber is what? Tuber. The best example that we have to remember is potato, right? Potato, best example that you have to remember. And we just discussed that potato and sweet potato, they are the example of analogous organs. So tuber, so swollen terminal part of an underground stem due to the storage of food. So it's the swollen underground part due to the storage of food. So it is having the buds, right? So scars which are axillary buds. So it is having the buds, right, which are eyes basically. We have seen that potato eyes, right? So these are basically the scars which are axillary buds. Okay. So potato is the example for the tuber. So make sure you write it. So if I have to make the short notes, running short notes, what will I do? I'll write the rhizome. Then I'll say grow parallel or horizontally. Obviously, we know that it comes under the underground stem modification. Then I will write this thing, example. Oh, if only that much you can remember, na, trust me, you'll get good marks here. Okay, so next I will write tuber. That will be swollen. Terminal part. Example, potato. That's how you can write. Now it is comb. C for comb. C for colocasia. Colocasia or Amorphophallus. Zamikand. This is again given in the NCERT. Zamikand is also known as elephant foot. Right? Both are edible. So comb is, from comb you can remember VC. VC we use for video calling, na, video conferencing. So vertically comb. So comb is vertically swollen. It is usually unbranched. It is usually what? It is usually unbranched. Is that clear? Is that clear? So they are short underground stem. They have the adventitious roots. So they bear nodes, internodes, scale leaves. So colocasia and Amorphophallus, that is Zamikand, is the example. So we'll show you the example. Ye dekho. This is your comb. This, you know, brownish thing. Right? Potato. This is the bulb. We'll discuss about it. Okay. So next modification is what? Next modification is bulb. Next modification is what? Next modification is bulb. Are you getting my point? What is the next modification? That is bulb. So that bulb is reduced disc-shaped stem. What's that? It is the reduced disc-shaped stem, but is surrounded by many concentric scale leaves. Many roots arise from its base. I believe you all have seen the onion. So you know that many roots arise from its base. It is having the covering. That for that covering, we use the word tunic. So they are simple tunicated bulb. They are having a sheath around them. So in that onion, the part that we eat, that is basically what? They are the scale leaves. They are the modified scale leaves. And even the garlic is also the example, but garlic comes under compound tunicated bulb. Are you getting my point? Garlic comes under compound tunicated bulb. So concentric rings of bulblets surrounded by a white sheath or tunic. So it is the garlic. Is that clear? Is that clear? So in the onion, these are the storage leaves. Leaves are modified. They have stored the food. Understood? Leaves are modified. They have stored the food. Understood? Yes? Sure? Done. So see, this is the line given in NCERT that potato, ginger, turmeric, colocasia, zamicum, they all are modified to stored food. So potato, we know that example of tuber, rhizome, corm. Okay. Done. So now the aerial modification, right, above the ground, obviously. So here what do you see? You see the stem tendril. What do you see there? You firstly see the stem tendril. What are you going to see here? You are going to see the stem tendril here. Are you getting my point? So a cylinder coiled structure which help the plant in climbing, and it arises from the axillary bud. Even in the anatomy part, I told you, when your shoot apical meristeme grows, a part of it left behind, right? It is at the base of the leaf. We know that. What do we call it? It is the axillary bud. Remember? Remember? So a cylinder coiled structure that help that plant in climbing. So it arises from that axillary bud. So you know that it will replace the branches, because axillary bud, from axillary bud, branch or the flower can grow. So it will replace the branches. So it support and climb, right? So it helps in supporting and climbing in the weak stemmed plants. So example is grapevines, right? In the cucumber, right? So in cucumber, pumpkin and watermelon, you see that. Right? So in cucumber, pumpkin, watermelon, you see this. Then comes the stem thorn. That is again a very recent PYQ. So stem thorn, right? Stem thorn. Where do you see this? You see this in citrus. You see it in bougainvillea and even in pomegranate. So guys, you have seen this bougainvillea as well. Ruk jao. That's bougainvillea. है ना? You know this, right? A very common plant. So that is bougainvillea. Okay, that is bougainvillea. So next is colocasia. So see, these are the leaves of colocasia. These are the leaves of colocasia. This is colocasia. These are the leaves here. So it is the underground stem modification. So this one we are talking, okay? This one. So that's colocasia, okay? So do check these words. They are very common examples. They are very common examples. Okay? Done? So a sharp, woody, pointed structure that is protective in nature, it arises from the axillary bud. So this one also arises from the axillary bud. So its function is to provide protection against the herbivores, right? So it may bear leaves or the flowers, but example is bougainvillea, citrus or pomegranate. Okay, stem thorn. So beta, thorn, spines, prickles, these are the terms that we use for anything which is, you know, pointed. But when you talk about the thorns, in the case of thorns, you will see they are very thick. Okay, just check it out in the case of bougainvillea. Just check it out in the case of citrus. You'll find it's quite thick. It is having that bark, right? It is having the vascular strand as well. So it is a modified stem structure that possesses the vascular cylinder and barks. So spines are modified leaves. So if the word is thorn, you are talking about the modification of stem. If the word is spine, you are talking about the modification of the leaf, right? That will have the vascular strand, but here you will not see well-developed bark. And prickles, they are just the superficial outgrowths of the stem. Just sometimes some pointed structure, we can just break them very easily because they don't have that vascular cylinder also, and they are just the superficial outgrowths. Okay, so next is very important, that is phylloclade. As I said, very, very important. So look at the example here, that is opuntia, right? That is opuntia, your cactus. So here this green part is actually stem. Here actually the stem will become flat. Here actually the stem will become flat. It will become green, and here you will see that it will have the spines. Understood? It will have the spines. Okay, so here the stem will become flat, stem will become green, stem will become photosynthetic, and the leaves, they are going to make the spines. Leaves will get modified and will form spines. So phylloclade, what's that? It's a green, it's a flat or a cylindrical stem that gets transformed into a leaf-like structure. Am I clear? Am I clear? So what is this phylloclade? So this phylloclade is a green, it's a flat or a cylindrical stem. It's a green, it's a flat or a cylindrical stem that gets transformed into the leaf-like structure. Is that clear? Is that clear? So they are of unlimited growth, that is they are of indefinite growth. Okay, the growth is unlimited, the growth is indefinite. Very important to remember. So this stem will take over the function of what? It will take over the function of the photosynthesis. We know it very well. So opuntia and even euphorbia is there. Please just search it once. Right? Kindly search it once. You will find, right, euphorbia. You have, I hope you have seen it. Right, so these are not the petals here, these are the brackets, right? This is the inflorescence here. Ye dekho, this one. This one. Euphorbia. This one. Do you know this? Yes? So the next is cladode. Again, the stem modification. These are green branches of limited growth. Right, they are the green colored branches. What are they? They are the green colored branches of the limited growth, which is usually 1 internode long. So it is what? These are the green branches. They are of limited growth. They are usually 1 internode long, and they have also taken up the function of photosynthesis from the leaves. So they are basically, true leaves are reduced to scales or spines here. So look at this. So this part which you can see the leaf-like structure, it is actually the stem. So this is your cladode. This is your cladode. So there are 3 words: phylloclade, cladode or phylloid. Phylloclade or cladode, they are the modification of stem. Phylloid you will see under the leaf modification. So this is important. Ruscus and asparagus, they are the examples. Am I clear? Ruscus and asparagus, they are the examples. Okay, so their comparison in these two, it's very important. So you can see phylloclades, what they represent, they has unlimited or indefinite growth, right? Cladode has limited or definite growth. So it is having, phylloclades are having several nodes and internodes. Cladodes are usually 1 internode long. So phylloclade true leaves are commonly caducous, like they are not persistent. They will not persist. They'll shed off early. So here true leaves are either reduced to scale or are modified to spine. So opuntia, euphorbia, very important example. Here the examples are ruscus or asparagus. So they are, what is the main difference? Their growth thing is main difference. Okay, this growth thing is their main difference. Am I clear? Sure? So the next part is the stem modification. So I told you already that this is a subaerial stem modification, but I said that runners in the NCERT, they have mentioned or explained it under what? Right, runners, they have explained under the underground modification. So in NCERT, this is the line. The underground stem of some plants like grass and strawberry, they spread to new niches. And when older parts die, they form the new plants. This is given in NCERT. So that is why I'm saying that in NCERT, they have mentioned it like this. But actually, it's a subaerial stem modification. So how can you remember the example? You can simply remember that a boy is running on the grass with a strawberry in his pocket. Boy is running on the grass with a strawberry in his hand or in his pocket, whatever you want to say. So obviously from running, I will remember runner. And I have 2 examples here, grass and strawberry. Okay, so wild strawberry is also given as the example of the stolen in some books. But here, when it comes to the example, we are focusing mainly on what? We are mainly focusing on the stem modification, okay? Oh, sorry, on the NCERT. So runners, they are the special, narrow, green, horizontal. I hope you all have seen the grasses. So they are special, they are narrow, they are green horizontal branches that will develop from the axillary bud. that will develop from the axillary bud. So they'll creep on the ground and the roots at intervals. Am I clear? Am I clear? So you know that green branches, they'll just creep on the ground and at intervals we'll see the roots. So they have tufts of adventitious, right? Tufts of adventitious roots that will attach to the soil. So as you know, grass and the strawberry is the example. So next is the stolon, like mint jasmine, having very good fragrance. So somebody is trying to steal it. So here what you have to remember, like in plants, like, so this is complete NCERT. And here in the case of stolon, one more thing is very, very, very important, right? See, firstly example, mint and jasmine. What is happening? A cylinder branch, right? It arises from the base of the stem, and then it will do what? It will arch downwards. It will arch downwards. Are you getting my point? So in plants like mint and jasmine, a cylinder lateral branch arises from the base of the main axis, and after growing aerially for some time, it arches downwards. Is that clear? Is that clear? So it arches downwards to touch the ground, right? Next, offset. So this one you will see in the aquatic plants. So here a lateral branch with a short internodes, and each node, this is the lateral branch with a short internode, and each node is bearing the rosette of the leaves, right? And it is having the tuft of the roots. So you know that they are hydrophytes, so they have the root pockets, not the root caps, but you will see the root pockets here. Is that clear? So you have the example of pistia and acornia. So this acornia, we also call it as water hyacinth. In sexual reproduction in flowering plant also we discuss it. It is also known as terror of Bengal, right? What do you call it? You call it as terror of Bengal. That's what you have to remember. Why? Because this acornia, right? It is basically an invader. It was brought to India for its beautifully shaped leaves and beautifully colored flowers. So what happened when this acornia, it grows, right? It grows very excessively. It is having offset. So it propagates at a very faster rate. Okay, it propagates at a very fast rate, and due to which it will cover that, you know, that aquatic body completely. So what is going to happen there, right? Because it will grow excessively, it will use all the dissolved oxygen. So the aquatic life in that particular aquatic habitat, that will not get the oxygen and will die. So that's why it is also known as terror of Bengal or water hyacinth. Okay, right? Trust me, you all have seen it. Just check this. Check the picture of it. Then sucker. So here you will remember PCB sucks. So pineapple, chrysanthemum, that is the scientific name of course, and banana, right? Suckers. So in banana, pineapple and chrysanthemum, the lateral branches, right? See this. The lateral branches, see in banana also I told you the rhizome is also present. So lateral branches, they originate from the basal and the underground portion of the main stem. They grow horizontally beneath the soil and then come obliquely. Can you see that? Then come obliquely upward, giving rise to leafy shoots. Read it carefully. So it will grow underground, horizontally beneath the soil, and then comes out obliquely. Understood? Done? Done, beta? So the next is leaf. Okay, the next part that we have to discuss is what? It is leaf. Ready? Done? So it's not a difficult chapter, it's a very easy chapter. but all you have to do is you have to remember many things. So I think I have discussed this thing multiple times, that please divide these chapters. There's chapters where you have to mug up the facts, the chapters where you have to cram a lot of examples, like in the diversity part, in the morphology part, and revise them every day. You will just take 10 or 15 minutes for its quick revision. Done, beta? So now let's focus this part. So the part here is what? It is leaf. What's that? It is the leaf. So leaf is a green, it's a lateral, it's generally a flat structure. It's a green, it's a lateral, it's generally a flat structure, right? And we know that it born on the stem. So it develops at the nodes and it bears a bud in its axil. So whenever you will talk about a leaf, you will see that in a leaf here, we'll find the axillary bud. What are we going to find, students? We are going to find the axillary bud. Are you getting my point? What are you going to find? You are going to find the axillary bud, and you know that it is nothing but the shoot apical meristem only. So this will form branch या फिर flower, है ना? It will grow into a branch or a flower. Okay, so mainly into a branch. So leaves, they originated from shoot apical meristem, right, beta? So that shoot apical meristem will form leaf primodia, and they are arranged in acropetal order. So acropetal order means young one will be at the tip, old one at the base. Okay, so they are very important vegetative organ for the photosynthesis. Am I clear? So let's see the next part. So look at this part here. This is the leaf. Okay, so if a leaf is having this stalk-like structure, this is known as petiole. So students, sometimes they confuse petiole with the pedicel also. So pedicel is the stalk in the case of a flower that you see in the case of a flower. Here you are talking about a leaf, so you are having what? You are having the petiole. So petiole here, sometimes at the base of this petiole you also see this lateral outgrowth which you call as stipule. This structure here is what? This is the leaf lamina, or also known as leaf blade, है ना? Having this central part. This is the midrib, and here you will see veins, and then that veins also further form veinlets, right? And here, as I said, the axillary bud will also be there. Understood? Understood? So in the case of a leaf, we will see, in the case of a leaf, we will see that there is a leaf base, there is a stalk, and then this. So leaf base, what's the term that we use for leaf base? It is also known as hypopodium. It is also known as hypopodium. Then this leaf stalk, that is petiole, that is mesopodium. What's that? It is mesopodium. Then comes this leaf lamina, या leaf blade. It is epipodium. It is epipodium. So these are the three parts there. And as I said that here at the base you will see axillary bud is present. Okay, and see in the leaf, if it's a simple leaf, it's a midrib, okay, having the vascular bundle. These are the veins, right? They are providing rigidity through this leaf lamina, and this midrib veins or veinlets, what do they have? They have the vascular bundles in them. Okay, they have the vascular bundles in them. Done, beta? So here you will see that if a leaf is without petiole, right? If it is directly attached to the stem here, so if it is without petiole, then what do we call it? We will call it as sessile. Is that clear? We will call it as sessile. Understood? We will call it as sessile. Understood? So you people can see this here, right? So let's see this part, that is petiole. So it is a cylindrical stalk of the leaf that helps to hold the blade. So in the horizontal position it is present, and it will capture the sunlight. Okay, so there is a long, thin, flexible petiole here that will allow it to flutter in the wind, right? To flutter in the wind. So leaf without a petiole is termed as sessile. Am I clear? Leaf without a petiole is termed as sessile. Am I clear? Sure? Done? Okay, so if there is a stalk-like structure at its base, you'll call it as stipule. Okay, so look at this part, the leaf base. Again, a PYQ. Leaf base, it can be swollen. You'll see it in the Leguminosae family, and Leguminosae family is also the part of your syllabus. So if it is swollen, right, if leaf base is swollen, for swollen leaf base, you will use the term pulvinous, right? And if, let us in the grasses, in the monocots, you'll see that leaf base, it forms a sheath around the stem, right? So these two examples are very important. So here you can see that it is the lowermost part of the leaf by which the leaf is joined to the node of the stem. Right? It is the lowermost part of the leaf by which the leaf is joined to the node of the stem. So this leaf base, it protects the young axillary bud. So if, I'm not saying that it will always contain that, but yeah, often it contains two small lateral outgrowths which we call as stipules. Okay, so these stipules, they protect the young leaf and its axillary bud. So stipules, they are found mainly in dicots, they are absent in monocots. So for monocots, what will be the term? It will be X-stipulated. It will be X-stipulated. Okay, so in some leguminous plants, the leaf base may become swollen. You'll call it as pulvinous. You'll call it as pulvinous. That is a very important question. And in monocots, the leaf base expand into a sheath which will cover the stem holier partially. Understood? Understood? Sure? Done? So next is the lamina or leaf blade, right? So it is the green expanded part of the leaf, right? This part, green extended part. Look at the diagram, the midrib, the veins, veinlets, carrying the vascular bundle, having the xylem and phloem, and this is providing it the strength as well. So it is the green expanded part, right? It has two surfaces. In botany, generally, we do not use dorsal and ventral. In the old literature, definitely they have used dorsal and ventral. But in the case of, in botany, we do not go for these terms. So here in the case of leaf, yes, you will see two type of surfaces will be there. Adaxial surface is there and abaxial surface is also there, right? Adaxial surface is there and abaxial surface is also there. So when it comes to the adaxial surface, D, you remember it like DU, right? That is adaxial upper surface, then obviously abaxial will be lower. So adaxial is also referred as the ventral part and this one as the dorsal part. So in the case of leaf, beta, in anatomy we discuss it that if we have dicot leaves, so dicot leaves may, you know, dicot leaf is dorsiventral, right? Dicot leaf is what? It is dorsiventral. So in dicot leaf, you see that there is upper epidermis, lower epidermis. Lower will have more stomata, upper will have less. So that's how we divide. In monocots, both the sides are same, right? That is isobilateral leaf. So leaves are having two surfaces, adaxial, which I told you, ventral or the upper part, abaxial, dorsal or the lower part. So this is supported by veins and veinlets. So these veins and veinlets will provide rigidity to the leaf blade. Is that clear? Is that clear? Is that clear? So middle prominent vein is your midrib. So veins will provide rigidity, help in the transport of water, minerals and the food materials. Am I clear? So beta, this is the function of leaves here, right? This is the function of leaves, जैसे gaseous exchange, transpiration, storage, climbing, protection, and the reproduction is also there. Understood? And the reproduction is also there. So gaseous exchange may transpiration, that is loss of water in the form of vapors, right? You know that through stomata, right? Even in the storage also they help. Aloe vera is the example. Climbing may, they all help. So now there are two types of leaves. One is simple, another is compound. In simple leaf, what is the story? In simple leaves, you will see, let's suppose if this is the leaf surface, hai na? If this is the leaf surface, if this is the leaf surface, you will see its leaf lamina, hai na? This is the leaf lamina. So this is the border here, these are the cuts. So this is going to be complete, and we know that axillary bud is present at the base. Now if in that margin, if there are cuts, that is incisions, so they should not touch the midrib. Even if there are cuts, right, we also call them as in seasons, so they should not touch the midrib. That is the simple leaf. Now in the case of compound leaf, these in seasons, they'll touch the midrib and you will see some leaflets. Right, what are you going to see? You will see leaflet. Now here, this is not the midrib. The midrib here will become rachis. Okay, so in compound leaf, how can you differentiate these? So these are not the leaves. You will call them as leaflet because this complete structure is a leaf here. Okay, so here in seasons, they have touched the midrib, they are divided, they are forming the leaflet. So how do you differentiate leaflet and leaf? How do you differentiate leaflet and leaf? At the base of leaflet, you will not see the axillary bud. So this type of compound leaf is your pinnately compound leaf. And here, let's suppose if this palm-like structure, this is going to be palmately compound leaf. So here you will see no rachis. Okay, this is palmately compound leaf. Here you will not see, rachis is not there. So rachis is just like midrib. Midrib in compound leaf is known as rachis. So see this part here. So a leaf is said to be simple when its lamina is entire or when incised, the incisions do not touch the midrib. Is that clear? Is that clear? So a leaf is said to be simple when its lamina is entire or when incised, means when there are cuts, they should not touch the midrib, right? Now, in compound leaf, these incisions, they touch the midrib, they will break this leaf into number of leaflets, right? They will break it into number of leaflets. So the leaf here is the compound, right? So if it is pinnately compound leaf, this is the arrangement. Rachis is the term that you have to remember here. Okay, so it represents the midrib here. So neem, Azadi recta indica, medicinal plant, that is the example. Examples are important, okay? Now palmately compound leaf, all the leaves are attached here to a common point at the tip of the petiole. So bombex, that is silk cotton, is the example. Do mark it. Quick. So it is important. Done, beta? Noted? Chaliye. So the next part is there. So that is venation. The arrangement of these veins and veinlets, right? The arrangement of these veinlets in the veins and veinlets in the lamina is the venation. So how will they help? They help in conduction of water and minerals to the xylem, translocation of organic nutrients to the phloem as well, right? Because vascular bundles are present, so it is just like the skeletal system here. Okay, so it is just like what? It is just like the skeletal system here. So there are two types of venations, and I believe you have seen such type of leaves as well. Dekho, one is your reticulate venation. What sort of venation do you have? You have reticulate venation. The other one is parallel venation. So you have reticulate as well as parallel. So in reticulate, in general, what's the meaning of reticulum? Reticulum means network, right? Reticulum means network. Yes, please. Reticulum means network. So just see, here you will see the midrib. Okay, so you'll see the midrib, then veins, then veinlets will be there. Done. The veins and the veinlets will be there. So kind of network is there. So this is reticulate. So you will see it in the case of dicots, but yeah, exceptions are there. So you will see here that in the midrib, right? Obviously midrib is comparatively thick. So vascular bundle here will be larger, right? In the veins, it will be comparatively smaller, and there will be very small in these veinlets. So in the dicot leaf, because of this venation, you'll see vascular bundles are of different size, right? Vascular bundles are of different size. Then comes the parallel venation. So parallel venation mein what will you see? You'll see that all that, right? This midrib is little, I would say, they all are generally same, right? So parallelly these veins are arranged. So midrib is, I would say, slightly broader than the rest of it. Otherwise, you'll see the vesicular bundles, they all are almost of the same size. So in the reticulate venation, what you people will see, you'll see that, sorry, the veinlets form a network. The venation is termed as reticulate, commonly found in dicot. So people, mango, rose, etc. So parallel mein, when the veins run parallel to each other within the lamina, you can see that this is midrib. These are the other veins. It is usually found in monocots. So the grasses, banana, maize, etc. they are present. Okay. Now, these are the exceptions not given in NCERT, but you should know that, right? These are what? These are the exceptions. So Calophyllum, Corymbium, they being a dicot, they still show parallel venation. They are dicot, but still which venation are they showing? Parallel. And monocots, they also show reticulate venation. Smilex and colocasia is the thing. Is that clear? Smilex and colocasia is the thing. Is that clear? Sure? Sure? So next is the phyllotaxy, right? So phylon means leaf, taxis mane arrangement. So beta, nature is a great mathematician. Nature has arranged every leaf on a particular plant or a tree in such a way that they all get the sunlight. So see, the phyllotaxy is the pattern of arrangement of leaves on the stem or it's branched. On the stem or it's branched. So the purpose of phyllotaxy is to arrange leaves in such a way that all of them will get the proper exposure of sunlight. Am I clear? Sure? Done. So this is what phyllotaxy is. Okay, so this is what phyllotaxy is. Done? Done. So three types of phyllotaxy you are going to discuss. But before that, I'll discuss a trick with all of you. So it will be CM sir reading GOC. CM sir reading GOC. Word read Elstonia nereum. Done. CM sir reading GOC. Word read Elstonia nereum. Now, how do we remember this? It's simple. CM sir. First of all, there are three types of phyllotaxy. Firstly, this is C. From the C, you have to remember China rose. What do you have to remember from the C? You have to remember China rose. M stands for mustard. From the sir, you can remember sunflower as well as you can remember spiral. Spiral, which is also known as alternate. This is one type of phyllotaxy. So we are talking about the phyllotaxy, which is basically the arrangement of, right, leaves on a plant or a, right. What is phyllotaxy? So phyllotaxy is basically the arrangement of leaves. So here you will see we have a trick that is CM sir reading GOC, word read Elstonia nereum. So what does it mean? We have three types of phyllotaxy: alternate, opposite and world. So for the alternate, which is also known as spiral, right, from this trick you will cover China rose, mustard and sunflower, right. Then come to GOC. So G here will be guava, right. G here will be guava. O here will be opposite phyllotaxy. I'll tell you what exactly it is. And C here again is calotropis. Okay, these two examples clear. Then from word, you have to remember world phyllotaxy. Okay, so Elstonia nereum are the two examples here. So that is what we are going to learn from this particular trick. So three type of phyllotaxy is there. So in the case of alternate, which is also known as spiral, what exactly is happening? You'll see this is the stem, right? This stem is having what? This stem is having the nodes, right? This stem is having the nodes. So one leaf will grow here, other leaf will grow here, one leaf will grow here. This is going to be the arrangement. So if you will see this pattern, don't you think it looks like a spiral? It looks like a spiral. So spiral phyllotaxy, China rose, mustard, sunflower, even the grasses are also the example here, right? From the trick, you can remember the example. Even the grasses are also. Now, next is the opposite phyllotaxy. Next is what? Next is the opposite phyllotaxy. So now in the opposite phyllotaxy, what will you see in the opposite phyllotaxy? You will see two things. In the opposite phyllotaxy, you will see opposite decussate, you will see opposite superposed. Yes, opposite decussate, opposite superposed. Now when I'm saying superposed, like let's say this is the stem, here you have the nodes. One leaf here, one leaf here. Like this. It is superposed. And when it comes to the decussate, like both, this node will have leaf on both the sides. So when I will say decussate, so you'll see, let's suppose if leaf is there, leaf is there, something like this. So it is decussate. Like this is the node, both the side will have leaf, one here, another here. Next time here, like let's say one side this, 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 this. So this is decussate and superposed. So superposed will be there in the case of guava, and decussate you will see in the case of Calotropis. Understood? And when I'm talking about the world phyllotaxy, like here in at one node, you will see wall of leaves. A group of leaves, they will grow in such wall like structure. Is that clear? Done? Sure? Okay? Sure? Are you sure? So look at the arrangement here. Look at the arrangement here. See, types of phyllotaxy, obviously guava, right? This is alternate. One leaf at this node, another at this. Alternate. And here you will see the whorl of leaves, right? At one node. So these are the three examples. So next is the modification of leaves that we have to discuss, right? Next is the modification of leaves that we have to discuss. Then it is a very simple topic. After modification, we'll start the flower. Flower wala complete topic will take around 2 hours or 2.5 hours, because I will explain each and everything in detail. So I'll say 2 hours maximum. So in another 3 hours, we can finish it. Okay. So I'm going slow. Why? Because the students, they have this thing ki ma'am, this chapter is not there on the channel. Okay, rest all the chapters are there on the channel, but this particular chapter is not there on the channel. So that's why for class 11 students especially, I'm going that slow so that they can understand everything. Okay, so let's have a break. So this is the break time. So let's meet at 3:30. Okay, eat whatever you want to eat, then we'll see you at 3:30. So everyone, let's start the next topic. So I hope everything is clear up to this part. So now the next topic that we have to start is the modification of leaves, right, beta? What we need to start? We need to start modification of leaves. That is what we are supposed to discuss now. Now, when you talk about the modification of leaves, right? So for support, leaf tendrils, leaf hooks are there. Leaf tendrils and leaf hooks are there for what? They are there for the support, right? Number one. For storage, succulent leaves and scale leaves are there. For protection, spines are there. And phyllodes, leaf bladder and leaf pitcher. That's what we are going to discuss. Is that clear? That is what we are going to discuss. Is that clear? So here you can see, I'll tell you which part you need to remember. So this trick is over. See, leaf tendril. Leaf can also form the tendril. Even leaflets can also form the tendril. Leaf can also form the tendril. Even the leaflets, they can also form the tendril. So leaf tendrils, right? They are the thread-like tendrils. We know that they are thread-like sensitive structure that will coil around the support, that will give support to that plant in climbing. Are you getting my point? So sometimes what happen? This leaf, the complete leaf is forming tendril, right? Sometimes what happen? At the base of leaves, I told you often we'll see this stipules, है ना? So when this complete leaf, when it will get modified into the tendril, the stipules here, they will become foliaceous, right? Basically, that stipule will become leaf-like to perform the photosynthesis, right? Right, student? So the example is wild pea, right? So garden pea, wild pea, sweet pea, these three are different. When it is wild pea, Lethyrus afaca, right? So here the complete leaf will be modified into the tendril. Now, sometimes what happen? It is the leaflet. Now you know that where do we see the leaflet? In the compound leaf. So when it gets modified into tendril for climbing, so sweet pea, Lethyrus odoratus, edible pea, the garden pea, it is Pisum sativum. So here the upper leaflet will get modified into the tendril. So see this. So obviously it's not that easy for you to differentiate. But see, these are the tendrils. So in the case of garden pea that we eat, the leaflet is turning into the tendril. That's what you have to remember. And sometimes these leaflets, just in bignonia, right, they form some, hooks-like structure. Here, leaflet hooks are also there. These are claw-like, structures which will help in the attachment. So how to remember this? See, in the NCERT, they have given only this particular part. But when you see this paragraph of NCERT, no doubt they talked about the tendrils, even the spine. Even in the case of Australian acacia, they have talked about this thing, that petioles will expand and they will become, green. So actually they are talking about the phyllodes here. What are they talking? They are talking about the phyllodes here. Understood? Chaliye, so let's talk about it. So modification for storage. So here you guys can see the number one thing is succulent leaves. So here they will become fleshy and swollen. So in the case of aloe, right, aloe vera, you all have seen that aloe vera. So in the case of aloe and even in the case of bryophyllum, you will see the leaves, they will be fleshy. So when leaves are fleshy, they are swollen, right? They can store water, mucilage or the food. So they are found in the plants of saline and the xerophytic habitat. Saline habitat where the salt concentration is high, xerophytic means dry condition, xeric condition when there is the water scarcity. So aloe vera, that's what you have to discuss. Even the bryophyllum, I'll show you the picture as well, right? These are the two examples that you need to remember. So this diagram is not, this picture is not added here, so I'll suggest you to check it. So like in our area we see the bryophyllum. I don't know about your place, you just search it once, right? Because these are very common examples. Okay, honestly these are very common examples. Okay, chaliye. So the next is leaf spines. Next is what? Next is leaf spines. So basically, just here you talk about this thing, right? This is the phylloclade, right? You know that the stem will become flat, the stem will become green. So here you can see these leaves, they are modified into spine because they have to reduce the water loss as well. And this is an adaptation in the xeric condition. Okay, okay. And here also you can see the spine in the case of acacia. Okay, so this is what? This is the spine. So leaf parts are modified into spines. So their function is to protect the plants from grazing animals and to reduce the transpiration. So in Opuntia, which we discussed under the phylloclade as well. So this green part is what? This greenish part is phylloclade. It is the stem modification. But here, right here, these leaves are what? They are basically the spines. Is that clear? So then other example is asparagus. So acacia may be beta, stipules are modified into spines. So that's what you have to talk about. Am I clear? So that is what we have to talk about. Is that clear? Is that clear? So phylloclades, right? So the lamina is reduced here, right? The lamina is reduced here. So see, the phylloclade term is not used in NCERT, but this is that particular thing, that what exactly the phylloclade, that is given in NCERT. So that's why I'm telling you, when I'm saying this, these words are important, then please pay attention here, okay? Right? Then kindly pay attention here. So phyllodes, the lamina is reduced. Instead, petioles or rachis, right? The lamina is reduced. Instead, petioles or rachis, right? Instead, petiole or even the rachis, it will become flat. It will become a kind of, sickle-shaped structure. For performing photosynthesis. So when you see this part, Australian acacia, right here, this is not the leaf. It is looking like a leaf, but it is actually the modified leaf stalk. So leaf stalk is petiole. So petiole, even the rachis, it will become the flat. It will become sickle-shaped, right, beta? It will perform the photosynthesis. So this is going to reduce the transpiration because it is vertically placed. It is having very less stomata. So example is Australian acacia. So as I said, it is given in the NCERT. So these three things are very important, that what exactly is phylloclade, what exactly is cladode, and what is the phyllodes. All that three things are very important. Am I clear? They are very important. So these two are the stem modification and this one is the leaf modification. The petiole or the rachis, it is becoming the flat here. So that's what you have to remember. Am I clear? Right? Then comes the leaf picture. Right, then comes what? Leaf picture. So lamina is modified here. It is forming a kind of picture. Okay, so lamina is modified here. Look at this. Look at this. Your leaf tip is forming a lid-like structure. So lamina is modified here. So this is the kind of structure that you all are going to see here. Am I clear? Am I clear? Right? Am I clear? So leaf or lamina is modified into a large picture or as a nepenthes. So this is the picture plant. So it will catch, it will digest the insect. So it is the tip here that will form a lid. So this is what it is. So this leaf-like structure here is actually the leaf base. Leaf base is forming this. This is the petiole and this part is the picture. So you really need to know that. So leaf base is winged and the petiole is tendrilar here. Am I clear? Sure? Sure? Any doubts? Done? So there is one more example, beta, that is Venus flytrap, right? Venus flytrap, that is Diania basically, right? This is basically Diania, that is the example. Diania species. So here basically leaf will form a kind of flap like structure, and it is also a carnivorous plant. It is what? It is also a carnivorous plant, right? So leaf is going to form, like leaf is having a structure. Just say if I have to show you something like that, right? And here it is going to trap the insect. So it is basically what? It is basically the carnivorous plant. Is that clear? Sure, beta? Sure. So basically here you will see that it is a carnivorous plant and it is having what? The leaves are basically modified, right? A kind of hinge will be there, lobes will form, so they'll trap it, right? Do read about it. Do read about it. So then comes the leaf bladder, this kind of structure you will see. So this is in an aquatic carnivorous plant. So from this, what can you learn? You can learn one thing, that not all the plants are autotrophic. They can be insectivorous also. They are heterotrophic basically, right? Thank you so much, beta Sabrina. Chalo. So now focus here. So these occur in aquatic carnivorous plants of utricularia, that is bladderwort. So some leaf segments here, they will get modified. They are forming, you know, some bladder-like structure. Some bladder-like structure they are going to form. So they have trap whelps, they have glands that are going to trap and digest small animals. So again, it is also the carnivorous plant. Is that clear? It is also the carnivorous plant. Now the next thing that we have to learn is inflorescence. Okay, the next thing that we have to learn is inflorescence. Basically, inflorescence, how can you define that? It's the group of flowers, or I would say arrangement of flower on the floral axis. So in the case of this inflorescence, you will talk about the floral axis, right? That how that flowers are arranged on that floral axis. So for that floral axis, students, you use the word peduncle. What is the term beta that we use there? We use the term peduncle there, and how the flowers are arranged. They are arranged in cluster. Yeah, what is the arrangement? That is what we see under this topic of inflorescence. So there are two types of inflorescence. One is racemose and the another one is cymose. These are the two types of inflorescence. One is racemose, another is the cymose. So in the case of racemose, how do I remember? I remember it like life is a race. So it's keep going, keep going, keep going, keep going. That's how I remember. This is my way to remember it. So it means here, racemose. So here the floral axis grows continuously. Like it will not terminate. It will grow continuously. Like life is a race. It just continue. So same way you will remember that here the floral axis will keep on growing. It will not terminate, right? So this is the one thing that you have to remember. Like this is my way to remember this topic. Secondly, let's suppose this floral axis is growing. Let's suppose if this is the finishing line. Near to the finishing line, we will see young flowers and we will see that old flowers at the base. So what type of arrangement it is? It is the acropetal arrangement. So in racemose, we will learn that the floral axis will grow indefinitely, that is continuously. The arrangement here is acropetal. For the cymose, obviously, you will go for the opposite thing. So here the floral axis, it will terminate here. At the end, you will see the flower will form. So you have to understand that this old flower will be there at the tip and young flower will be there at the base. So this is basically the cymose inflorescence. And here we will also discuss three, right, special type of inflorescence as well: hypanthodium, cythium or verticillista, right? We'll discuss it, so you have to remember. So what is inflorescence? It is the arrangement of flower on the floral axis, right? It is the arrangement of flower on the floral axis. This is what inflorescence is. So axis, this floral axis is known as peduncle. So students, the petiole, it is the stalk of leaf. Pedicel, it is basically the stalk of flower. And if I am writing peduncle, then what am I talking? It is the floral axis that I'm talking about. It is the floral axis that I'm talking about. Am I clear? So here when shoot tip transforms into a flower, obviously at the tip when there is a flower, that flower will be solitary, means the single flower, right? But depending on it, we have two types of inflorescence, as I told you already. So in the racemose, the main axis will continue to grow indefinitely, right? Important point. Youngest flower is at the apex or at the center. Like if you are looking at it in this way, so floral axis will keep on growing. So young flower will be near the tip. But if I'm arranging it in this way, right? So young flower will be there at the center, old flower at the periphery. The arrangement is like that. So acropetal succession, that's what you have to remember, the another important point. So in mustard, which belongs to family Brassicaceae, and the members of, right, not all, members of family Fabaceae, which is also known as Leguminosae, that shows that. So examples here, do remember it, because both these families are the part of your syllabus. Then comes the cymose. So just opposite to that. So cymose may the main axis will terminate in a flower. It is a definite axis. So the growth is limited. Youngest flower will be near the base or the outside. So flowers are born in the basipetal succession. Example is Solanum, belongs to Solanaceae family. But Solanaceae family is not the part of your syllabus, okay? It is not the part of your NEET syllabus. So this is how the diagram is. So young flower, they are near the tip, old flower at the base. What sort of arrangement is there? This is acropetal. And here, the floral axis will terminate. This is the terminal flower. So old flower is at the tip, young at the base. So this arrangement here is basipetal. So yes, these are important points, right? Even given in your new NCERT. So you have to remember it, right? You have to remember it. Beta, this class will continue till 11:30 or 12 today. Okay, because here after completing this chapter, we'll complete the rest part of the anatomy also, right? So if you are in hurry, then just sit home, right? Close the lecture, because today we'll finish the morphology first, then again you'll get the break. You'll get the breaks in between, there is no problem in that, and then we will finish the anatomy. Later on the thumbnail will be updated, so both morphology and anatomy will be covered today. Next is the flower. So see, you know that when it comes to the flower, like firstly we all know that there is something known as shoot apical meristem. I hope you all are well aware of it, right? What do we have, guys? We have shoot apical meristem. What do we have? Shoot apical meristem. So from that shoot apical meristem, it is the floral meristem that forms. From the shoot apical meristem, it is the floral meristem that form. And if it is the floral meristem, obviously it will form what? It is going to form the flower. So can I not say that ki flower is a modified shoot? Because ultimately this floral meristem is developed from the shoot apical meristem. So it is the modified shoot. So basically, when we see a flower on a plant, right, the signal for the synthesis of that particular flower, right, like why are we able to see that flower on a plant? Hormonal changes are there, structural changes are there. So the moment we see the plant, all the changes they have done before, right? And then later, what is the thing that we see, right? Yes, we will see the flower. So this is what you have to remember. So floral meristem, it forms the flower, which is a modified shoot, So structural changes, hormonal changes, they are basically, right, they are responsible for the formation of this flower as well. Okay, now when you are talking about this flower, when I'm saying that it is a modified shoot, right, when it is a modified shoot, so obviously in the flower also you will see the nodes and internodes. So here in the flower, internodes are highly condensed, right? Internodes are highly condensed. That's what you have to keep in your mind. What are these internodes? These internodes are highly condensed. Like let's say this is the structure, this is the stalk-like structure in the case of flower. So what do we call it? We call it as pedicel. So here it's this end, right? So it can be flat or dome shaped. It can be flat or dome shaped. It is known as thalamus or receptacle. We can call it as thalamus, or we can call it as receptacle. We can call it as thalamus, or we can call it as receptacle. Is that clear? Thalamus or the receptacle. Now when you see that part, as I said, that nodes will be present here, but internodes are highly condensed, right? Nodes will be present here, but internodes are highly condensed. So these nodes, they will show us what? They'll show us the modified leaves. They'll show us what? They will show us the modified leaves will be present here. They're going to form the whorls. What are they going to do? They are going to form the whorls. Is that clear? Is that clear? So basically the flower is having the flower is a modified shoot. So internodes are highly condensed here, right? So that flat or dome shaped structure is the thalamus or the receptacle. This is the pedicel here. Okay. So overall, we all have seen the structure of the flower. So right, so basically on these nodes, these modified leaves as whorls are present. Okay, so what do we discuss here? We start from the outermost to the innermost whorl. So outermost to innermost, what will you see? You will see calyx denoted by K. You will see corolla denoted by C. You will see androecium denoted by A. You will see gynoecium denoted by G. Yes, you have to remember the symbol. So calyx is actually the group of sepals. Corolla is actually the group of petals. Androecium is actually the group of stamen. Gynoecium is actually the group of pistil or the carpel. Am I clear? It is the group of pistil or the carpel. Is that clear? So sepals, petals, stamen or pistil slash carpel is there. Just allow me 1 minute. Done, beta? So that's what you have. So here, this part is actually the sepal. Their group will form calyx. This part is actually the petal. And this is basically the stamen having anther plus filament. And here, what do you see? You'll see the pistil slash carpel, right? So that is what we have. So overall in a flower, you have to understand this thing. It is a modified shoot with highly condensed. It's a modified shoot with highly condensed, right? With the highly condensed internodes. And here you will see that on that node, these whorls, these modified leaves are present. So we have the sepals. So sepals together, they are making the calyx. So how do we denote it? We denote it with K. We have the petals, right? That they are making the corolla. How are you going to denote it? With C, right? And then you have stamen. So stamens together. So stamen is this, anther plus filament. So it is forming androecium, denoted by A, and then you have pistil or the carpel, gynoecium, denoted by G, So that is what you have to remember. Okay, so these are the four whorls, and flower having all four whorls, you call that as complete flower. What do you call it? You call it as complete flower. So sepal, petal, stamen, pistil, all that things are there. And as I said, so this is basically what? So this is basically the non-essential slash non-reproductive, but yes, you call these two whorls as accessory whorls. What are you going to call them? You are going to call them as the accessory whorls. So this sepal, it will protect that flower in the bud condition. Just I think you all have seen the rose, right? In the case of rose, when you see when it is in bud form, that greenish structure that covers it, that are sepals, that colored petals, that colored things, right? That are the petals. And then comes this part. So stamen and pistil, they are basically making what? These are the reproductive whorls slash essential whorls, right? So they'll play a role in the reproduction. So androecium, it is the male part. Gynoecium, it is the female part. That is the symbol. Okay, that is the symbol. So yes, if a flower is having all the four whorls, then it will be a complete flower. If there is any single whorl which is missing, right, then it will be an incomplete flower. So let's have a look. A flower is a modified shoot. And here, what is happening? Your shoot apical meristem will changes to floral meristem. So internodes, they do not elongate. They are rather condensed, and the axis, overall axis will get condensed. So you can see shoot apical meristem, leaf primordia, then the floral primordia or floral meristem will form. So apex, that is tip, produces different kind of floral appendages laterally at the successive nodes instead of leaves. So these appendages are the modified leaves. So the lower internode will form the pedicel beta or the stalk of the flower. The region where internodes are condensed, they form the broad base. It can be flat, dome shaped. That base is actually the thalamus or the receptacle. This is important. So flower is a condensed shoot, so it is basically meant for sexual reproduction. So beta flowers, they are also having the brackets. So what are they? They are the reduced leaf found at the base of the pedicel, so they are known as bracteate. Flowers without brackets are ebracteate. Just see again, when you are talking about a leaf, at the base of leaf, this lateral appendage, this is stipule, right? So if stipule present, then the plant will be, या फिर leaf will be stipulated, है ना, if it is present. If they are absent, then you will call it as exstipulated. Then you will call it as exstipulated. Now, in the case of flower, right, here at the pedicel, base of pedicel, you will see the brackets. So if brackets present, then it is bracteate. Then it is what? Bracteate. That is BR. It is bracteate. If brackets absent, right? If brackets absent, E bracteate. Are you getting my point? E bracteate. EBR. Understood? EBR. So brackets, they are at the base of this pedicel, right? So they are basically at the base of this pedicel. And stipules, do not confuse it with the stipule. Okay, not all the flowers are bracteate. So bracteate, if brackets present, so it is bracteate denoted by BR. So BR here is not bromine. It is bracteate, EBR, ebracteate. Right? EBR, ebracteate. Am I clear? So already told you, if the flower is the complete flower, then flower having all the 4 floral organs. Incomplete may absence of any one or more floral parts. So let's suppose if sepals are not there, if pistil is not there, yeah, anything can be missing here. Okay. Now, we know it very well, right? We know it very well. Just a flower, it can be bisexual also. And if you remember, for bisexual, though I have given you the trick that you can also use in animal kingdom. Instead of BMV, you can make it BMH, right? Just a BMW is there, you can make it BMH. So bisexual, right? M stands for monoecious and H stands for hermaphrodite. Hermaphrodite. Okay. So when it is bisexual, also known as monoecious, that's the symbol, right? That's the symbol for being bisexual. If a flower is bisexual, if a flower is what? If a flower is bisexual, right? If a flower is bisexual, then what will you see? This is the symbol. This is the symbol, okay? Right? Bisexual flower. Now, the flower, bisexual means male and female both are present. If the flower is unisexual, so unisexual means dioecious. So if it is a male flower, so male flower is denoted by this symbol, right? It is also known as, because stamen will be there, it is staminate. You will call it as staminate. If it is female flower, means carpel is there, yeah, pistil is there, then you will call it as pistillate. 8 is at the end. But important as well as PYQ, but if stamen is sterile, for sterile stamen, the term used is staminode. So do not confuse staminate and staminode. Sterile pistil ya carpel is basically pistillode. Am I clear here? Please write down. Quick. Please write down. Done? Sure. So you can have a look. Bisexual, where both essential organs, reproductive valves are present. Unisexual may staminate if male, pistillate if female. So next is the floral symmetry that I have posted, I think yesterday, है ना, where even we discuss the tricks also. So next is the floral symmetry. Beta, same thing you discuss in the, right, same thing you also discuss in the case of what? This floral symmetry. The same thing you people also discuss in the case of what? In the case of animals as well. So I'll give you the example of animals. In the case of animals, when you talk about the poriferans, mostly, I'm not saying all, mostly poriferans, you say na they are asymmetrical. So what does it means when we are saying asymmetrical, right? Means any plane that is passing through the center, right? Either top to bottom, left to right, whatever it is, cannot divide that animal into equal halves, porifera, right? Or when we talk about the radial symmetry, if you remember, we discussed the example of starfish, that any plane which is passing through the center, any plane which is passing through the center, radial symmetry, when do we say radial symmetry? When any plane passing through the center can divide that organism into equal halves. Then we talk about the bilateral symmetry, like our case, even in the case of your arthropoda, etc., etc. So only one plane, that is from top to bottom, can divide the body into equal halves. Here, any plane that is passing through the center can divide the body into equal halves. Here, only one plane that is passing through the center can divide the body into equal halves. And in asymmetrical, no plane can divide the body into equal halves. So now same thing is in the case of flower. So in the case of flower, firstly we will talk about asymmetrical flower. So asymmetrical flower means no symmetry, no plane can divide it into equal halves. The example is canna. The example is canna. Again, canna is also a very commonly seen plant, right? Even if you will check the short section, you'll find it. Ye dekho, canna. This is canna. Okay, canna, right? So whenever next time you see this flower, talk to this flower and tell that flower, okay, bro, you are asymmetrical. No plane can divide you into equal halves, right? No plane can divide you into equal halves. Is that clear? Is that clear? Sure. Sure. So this is canna. Now the next is actinomorphic. Next is what? Next is actinomorphic, which is also known as radial symmetry. So obviously if it is radial symmetry, any plane that is passing through the center can divide it into equal halves. So here what is the trick? The trick here is mummy daddy chilling in America. Mummy daddy chilling in America. This is the trick for this. And for this actinomorphic, this is going to be the symbol. Right? If this is the symbol in the floral diagram, in the floral formula rather, so it shows us that the flower is actinomorphic. It is telling us, na, dekho, any plane that is passing through the center can divide the flower into equal halves. So what is the trick here? It is the mummy, daddy, chilling in America. So mummy, M-U, mustard here, dhatura here. So I'm just sticking to the NCERT examples. Chilling, chili, and America. A will remind you actinomorphic. The sound of this R will tell you the symmetry is radial. Am I clear? Will tell you the symmetry is radial. Now, next is zygomorphic. So if actinomorphic is radial, then it is obvious that it is going to be what? Bilateral. So here, just my trick is, you all play that game na, PUBG-CZ. I told you to remember that, hai na? That this is what you can remember. So one student in the comment section given us the better trick. PCB Gen Z. That's given by a student. That's not mine. Okay. So here you can remember the example. Just say P, bean. This gen can confuse you. That's why I didn't include there. Gulmohar, C for cassia, and Z stands for zygomorphic. Okay. So here that student said, ma'am, it can be PCB, Gen Z. Okay. P, cassia, bean, gulmohar, and zygomorphic they are. So you can denote it like this, or like this, or like this. That's how you can denote it. So this is about the symmetry. Write down the tricks first. Quick. Write down the tricks first. Quick. Okay, so I hope it's done. So look at the floral symmetry. So here you can see actinomorphic or radial, when flower can be divided into two equal halves in any radial plane that is passing through the center, it is actinomorphic, that is radial. So chili, mustard and dhatura, right? Look at the flowers also. Then zygomorphic bilateral, so pea, cassia, gulmohar and bean, when a flower can be divided into two similar halves only in one particular vertical plane, right? Only in one particular vertical plane. Then asymmetrical canna, right? A flower is asymmetrical, irregular, can't be divided into two similar halves. Understood? Now the next is the isomery, right? Next is what? Next is the isomery. Next is what, beta? Next is the isomery. Let's talk about that. Okay. One minute. So isomery is the presence of the same basic number of different floral organs of a flower, right? What is this? This is the presence of the same basic number of different floral organs of the flower. So if it is trimerous, tri means 3, so it will have the floral appendages, right, in the multiples of 3. So if I have to give you the example of trimary, I can give you the example of lily, I can give you the example of coconut as well. There also you will see the trimary. I can give you the example of tulip as well. So here you will see them as the multiple of 3. Is that clear? Is that clear? So in the grass family, this is going to be your basic, right? So in the grass family, this is basically the grass family. Again, it is also the part of our syllabus. We see the trimary. Then in the case of tetramerous, remember this, you'll take the example of mustard. So mustard belongs to which family? Again, the part of your syllabus, Brassicaceae, Brassicaceae, right? You have even the example of dhatura here. Okay, so pentamary में, tetramary में, हाँ ठीक है. This is the example here. And then in the case of pentamary, pentamerous. Anybody who can give me the example of pentamary? Right, mustard is there. Anybody who can give me the example of pentamary? Actually, dhatura is the example of pentamary there. Right, dhatura, even potato, right? They are the examples. So basically, Solanaceae family, potato family is showing it, right, some members. Now here even radish is also the example, again Brassicaceae family. So this is what you have to remember. So you need to understand the meaning here. So in morphology, leaf part, right, and the flower part is very important. So the next topic that we are going to discuss, right, so this next topic that we are going to discuss, it is very, very, very important, beta. It is very important, right? The next 3 or 4 topics that we are going to discuss, they are extremely important, right? You are literally not allowed to skip them. You should know everything about them. And do remind me, I didn't discuss the 3 special type of inflorescence, right? I just forgot to discuss the 3 special type of inflorescence. Do remind me. After the flower wala part, we will discuss it here in this lecture only. So now the next topic is the position of floral organs on the thalamus. Right, position of floral organs on the thalamus. That's what you have to discuss, right? Position of floral organs on the thalamus. So thalamus or receptacle. So floral whorls, right? All the whorls, right? Floral whorls are arranged successively on the swollen end. Floral whorls are arranged successively on the swollen end of the stalk or the pedicel. So this is the thalamus or the receptacle. What's that? It is the thalamus or the receptacle. So based on the position of calyx, corolla and the androecium, based on the position of what? Calyx, corolla and androecium. But what? With in respect to, right? Sorry, with in respect of ovary, like on the thalamus, we know that all these 4 things are present. So in respect to ovary, how are they placed? whether ovary is up, rest of the parts are down, yeah, ovary is covered, rest parts are above. So what sort of arrangement is that? On the basis of that, we describe flower. So when it is hypogynous, I'll tell you here itself, this is what you have to remember. The ovary is going to be superior, right? The ovary is going to be superior. This is the symbol for the superior ovary. Again, direct MCQ it is, and as I said, it is something very important that you have to remember, okay? Hypogynous flower, a very important MCQ. And as I said, the superior ovary, that's what you have to see here. So see, in the hypogynous flower, the gynoecium occupies the highest position. Why do we say it is the hypogynous flower? Why do we say that ovary here is superior? Because it is at the topmost position. So don't you think the degree of protection will be less? In the ovary, ovule is there. Ovule is present in the ovary. That will form the seed. So basically, you know that seed, product of fertilization it is. So hypogynous flower, superior ovary, that is the symbol. So the gynoecium occupies the highest position while other parts are situated below it. So this is the superior ovary, and do remember the example mustard, China rose and brinjal. Is that clear? Is that clear? Mustard, China rose and brinjal. Understood? Right, so here what can you remember in the hypogynous flower? What can be the trick? The trick is China bring, right? China bring mustard. Okay, that's what you can remember. Done. China bring mustard. That's what you can remember. So China brings a brinjal and then the mustard. So China means China rose will be there. Okay. So China bring mustard, right? China bring mustard, but how to remember, ma'am, hypogynous flower? So China bring mustard, either you can remember superiorly, bas. That's how I remember. But I just remember as China bring mustard, theek hai, hypogynous flower, because China rose, brinjal and the mustard, they are all three together. So China bring mustard superiorly. Theek hai? China bring mustard superiorly. So China rose, brinjal, mustard superiorly will tell me superior ovary. It means hypogynous flower. Is that clear? Is that clear? So China bring mustard superiorly. So remember that, okay? Okay. Done. So the next is what? Next is the perigynous. So if the flower is perigynous, the ovary is going to be half inferior. What's that? It is half inferior, right? So here gynoecium is in the center. Now here it is not at the top position. It is there in the center, and other parts of the flower are located on the rim of that thalamus, almost at the same level. That's why it is half inferior. Okay. So what is the trick here? Plum, rose, peach, perigynous. Okay, plum, rose, peach, perigynum. Is that clear? Is that clear? Done, beta? Sure, plum, rose, peach, they are perigynous. Am I clear? Done, beta? Sure? Any doubts? So here what will you remember? You will remember peri, peach. That's your example. Peri, peach. So this peri will tell me about the perigynous flower, that is half inferior ovary, and this P will tell me about the plum. Peri, R is also there, plum, rose and peach is written. So it is peri, peach. Okay, so one trick here is that China bring mustard superiorly. China bring mustard superiorly, and the another is peri, peach, right? So next example is peri, peach. So perigynous, from that peri you will remember plum, rose, right, and then peach. So then comes the epigynous, beta. So ovary is what? It is going to be the inferior ovary, right? But when ovary is inferior, it is having more degree of protection. So the flower is going to be what? It is going to be epigynous. So in epigynous flower, the margin will grow upward, right? They will enclose that ovary. They will completely cover that ovary, right? And will get fused with it. So more degree of protection will be there, right? So the examples are what? Guava, right? Cucumber and the ray florets of the sunflower. And what exactly is there? Ray florets of the sunflower. So in sunflower, ray floret, disc floret, both are there. So ray floret, females, ray of hope. So they can be female flowers or they can be neuter as well, like nothing, but they can be female flowers as well. If it is disc floret, D for, like in Hindi we call it dono, but matlab from D you will remember that here you will see male as well as the female part. Die, D say die. This is what you can remember, male as well as female, right? Don't confuse it with dioecious, but ray florets, either female or neutral. It is like that, right? So what are the examples? So here we have guava, cucumber, and ray florets of sunflower. Is that clear? Ray florets of sunflower. Am I clear here? Yes? Done? Done? Okay? So what is the trick here? That in Goa, right, sit in sun ray, sit under sun rays and eat cucumber. Okay? Go to Goa. So in Goa, sit in sun ray, eat cucumber. So basically Goa, from Goa you will pick guava, right? Sun rays. In say you will pick inferior ovary, guava, then sun ray, that is ray florets of sunflower and the cucumber. Is that clear? Is that clear? So that is what you have to remember. Am I clear here? Sure? Sure? Done? Next is the parts of the flower that we have to discuss. So what we see, again, these are some general terms that you have to remember. We'll start with the K for calyx. So calyx is what? Group of sepals. Already told you what are sepals, right? Already told you what are sepals. So let me show you the diagram. Jesse, this is China rose, right? Belongs to Malvaceae family. Okay? So this is China rose. Okay? China rose. You know about it, hai na? You all know about it. This is China rose. Now, in the case of this China rose, see this part. This part is actually the sepals. This one. I'm talking about the China rose first. But please see this part also. This is a stalk. This part here below the sepal, it is actually this is known as apicalyx. They say here, it is the calyx part. All the sepals together are making calyx. So this part here is apicalyx. Not all the flowers are having apicalyx, but here you can see that. And these are what? These are the petals. These are what? These are the petals here, right? So in China rose, this part is what? This is apicalyx. Now when you look at this particular part here, so see, so many stamens are there. This yellow color, they are anther, their stalk is filament, they are stamen, they are joined here, right? They are forming one bundle, and this is going to be the female part. Are you getting my point? This is what? This is the female part. So this is the example of China rose, where I'm just trying to show you, right? See, there is, let me look for some. So this is a very common flower. You all know, I think you all have seen it, isn't it? Isn't it? So now what you have to remember, see this. So parts of a flower. So this is the calyx that we have. So calyx, as I told you, K, right? So it is the outermost accessory wall. Why accessory? Because it doesn't take part in the reproduction, but it will protect that flower in the bud condition, right? Right. So generally they are green, leaf-like, protect the flower in the bud stage. Now, there are two words that we have to remember. There are two words that we have to remember. See, if sepals are free, like they are not fused with each other, the word will be polysepalous. Okay. If they are fused, if sepals If they are fused, then you will call it as gamosepalous, right? So how can we denote that? Like K is for calyx, right? Let's suppose K is for calyx. Now let's suppose if you have n number of sepals, it can be 2, 3, 5, whatever it is, n. This is also n. Here I'm showing the bracket. So this bracket is showing the fusion. So here I will call it as gamosepalous, right? If it is written like that, and if it is written like this, so I will call it as polysepalous. Now what does it mean? See, when you use the bracket, so bracket means you are talking about the cohesion. Cohesion means same kind of things are fusing with each other. Is that clear? Is that clear? Is that clear? Sure? Sure? So that's what you have to keep in your mind. So brackets, they show the cohesion. So let's suppose if there are 5 sepals. If I'm writing K5, it indicates 5 sepals and they are free. If I will write like this, 5 sepals, but they are fused. Got this? Isn't it easy? Now same for the corolla, right? Corolla denoted by C. So if it is Cn, then it is polypetalous. And if it is C, let's suppose N and the bracket, then it is gamopetalous, because here the unit is petals. Are you getting my point? Are you getting my point? Right, just say if let's suppose I'm writing C5, if I'm writing like this, so I'll say 5 petals and they are, matlab corolla contains 5 petals and they are free. 5 petals, but they are what? They are fused. So these terms are clear, yes or no? These terms are clear, yes or no? So just have a look. So generally sepal, so this part is done, huh? So gamosepalous sepals are fused. You know that. This is how we are going to show it. Polysepalous sepals are free. So sometimes some fruits, they have persistent calyx. Even after the fruit formation, the calyx, it persists. Like in the case of tomato, in the case of dhatura, you people will see that the calyx will remain there even after the fruit formation. Example is Solanaceae family. Understood? Example is Solanaceae family. Okay? Okay, so here also you people can see the corolla may be, yes, the corolla may also be united, जैसे gamopetalous, or it can be free, that is polypetalous. Am I clear? Am I clear? Chaliye. So now the corolla, it can have the different shapes, right? It can be tubular, it can be, bell-shaped, funnel-shaped, or wheel-shaped. Understood? Look at that diagram. Quick. Students, quick. Right? Look at this tube-like, bell-like, funnel-like, or wheel-like corolla. Okay? Done? So if I talk about this wheel-like, so I will see it in the case of brinjal, right? In the case of potato, okay? You'll see this bell-shaped, you'll see in the case of calotropis, right? Calotropis example we have studied, right? Right? So wheel-shaped is there. So if I talk about the tubular, so these examples are not given in the NCERT. So funnel-shaped will go for, yes, please. Just a minute. Actually, beta, this tubular will go for calotropis, right? It will go for the calotropis. This bell-shaped will actually there in solanum genus, that is potato here. Brinjal will be there, or funnel-shaped, which is also known as infundibulum. You can see that in the case of tobacco. So there are many examples. I'm just giving you one, right? You can just relate it and can see it there, okay? Done, beta? Quick. Okay. Please write down. So beta, potato mein, that's why I just got confused. Potato mein you will see this bell shaped ke liye na, it is not potato because it will go with that brinjal only. So for this bell shaped, you can remember the example of, this is the part, these examples na, sometimes it's tough to recall. So they are dhatura. ठीक है? Dhatura. Okay? Done? So calotropis, it is given in our NCERT, bell shaped mein dhatura. So this I was right earlier also, potato. So it will be there in brinjal, it will be there in this potato also, and tobacco. This is the example you can discuss here, but it is not given in the NCERT. Is that clear? Matlab, these examples are not given in the NCERT. Okay? Chali. So the next part is perianth. Perianth, this is what you have to discuss. Now I think you all have seen the lily flower. Right, lily, very beautiful they are. You know that lily, like different colors, white, even pink, red, yellow, right? Different colors they have. Now, when you look at this lily, right? Look at this. This is in a kind of bud stage. But can you differentiate the sepal or the petal here? No. Look at that open flower also. So sometimes what happen in some flowers, जैसे कि lily. Right, do check this lily. Lily will also show you the epitepalous condition. We will discuss about that. Right, please understand. Please mark this. So calyx and corolla, they are not distinct. Right, just say I remember it like this. What is a tepal? Tepal means sepal plus petal. Basically, they both are not distinct, they are not different. We cannot differentiate okay, this is sepal, this is there, they're like fused. So that is tepal. So group of tepals, right? Group of tepals, obviously you will call it as perianth, right? Which is denoted as P, right? It is perianth, which is denoted as P. So here also, this is the word that you can use. Right, you know that it will be polytepalous, that will be gamotepalous, right? Polytepalous and that will be gamotepalous. Got that? Right? Yes? Understood? So here you can see polytepalous or gamotepalous. Done. So now we will learn one more thing. Before starting estivation, we need to understand one more thing here. See, sometimes now we will talk about adhesion. See, in cohesion, I told you, right? In cohesion, I told you this thing very clearly that same kind of things they'll fuse. Isn't it? Isn't it? In the cohesion, I told you clearly that same kind of things they will be fused. Am I clear? Am I clear? But in adhesion, adhesive, they attach to different surfaces. So now sometimes what happen? Like this stamen, we will discuss it there also, but I thought we are completing the example, so we'll discuss it there. This is stamen, right? This part is anther. This is actually the male part, and this is filament. Even in class 10th, we learn this. So when this stamen, when it gets fused with petal, right? Stamen is getting fused with petal. Then this is known as epipetalous condition. What sort of condition it is? It is the epipetalous condition. So here, corolla, petals are denoted by C, androecium. This is the symbol. Okay, so if this is the symbol there, then you can see that it is the epipetalous condition. Am I clear? Am I clear? Right? And guys, like those who are not able to understand the Hindi, why are they obsessed about the Hindi like lectures? Right? You have this English lecture, you are able to understand everything. And if you are thinking that the content, better check the content. You can compare it with any of the Hindi session. Right? You are never going to regret it. And Rather, you'll say that, yeah, we are getting good content here, the better content here. So rather than focusing on such things, please focus here and try to remember these terms. So this chapter is easy, but here you have so many terms, right? So that is basically a kind of problem, right? That is basically a kind of problem. What kind of problem it is? Ki, yeah, you have to retain it. So it's easy, though morphology is not difficult, but too much to remember, right? Okay. Done. So where do you see this? You'll find this condition in some members of Solanaceae family. Do you know that? Solanaceae family. It is important. Now, if stamen get fused with tepals, then what will be the word? Now it is tepal. So obviously example is lily, right? Please check this lily flower, right? You'll very clearly see, right? Very clearly you people are going to see this, isn't it? Right? So then the word is epitepalous or epiphyllous. So that's the symbol, okay? So please remember the symbols, then the families part will be very easy for you. Understood? Done? So next is the estivation, another important topic. So for next 30 minutes, whatever topics I'm going to discuss, right? I told you that flower part overall is important, right? Hypogynous, epigynous, perigynous flowers, very, very, very important. So you have to keep it in your mind, you have to revise it. So just look at this. So even after that, estivation, placentation, they all are very important topics. So the next part is what? Next is the estivation. So it is the arrangement of floral organs, that is sepals or petals in relation to one another. What's that, people? It is the arrangement of floral organs, sepals or petals in relation to each other. Different types of arrangements are there, right? You'll see the twist, the velvet, twisted, right? Vexillary, estivations. So different types of estivations are there. Allow me 1 minute. So there will be different types of estivations. So velvet, when you are talking about what? When you are talking about the velvet, here what is happening? The sepals or petals, they are in a whorl. Look at this. 1, 2, 3, 4, 5. See, they are in the whorls. They are not overlapping each other. Their margins can touch each other, but they are not overlapping each other, right? So here, sepals or petals in a whorl, they just touch one another at the margin, but they are not overlapping. So example is calotropis. Example is calotropis. Am I clear? So what is the example that we have here? The example is calotropis. Understood? Next is twisted, right? As the name is saying, you see this is one petal. It is getting overlapped here. Here it is over. So now it will overlap next petal. Now because this petal is getting overlapped here, it will overlap next petal. So this is the twisting arrangement. Look at the China rose. Look at the China rose. Observe that China rose. You will see that. You'll see that, and then you are not going to hate the morphology part. You are going to love it. Okay, you are literally going to love it because this is going to be amazing for you. So look at that cotton, right? Actually that petals are arranged in such a way, right? They are actually arranged in such a way. Okay, you will find that. Done. So ye dekho, in the twisted manner everything will be arranged. So one margin of the appendage will overlap that of the next, and so on. So China rose, lady's finger, that is your okra, and the coconut. Is that clear? Is that clear? Sure. Next is imbricate. Here this overlapping is not proper. Like if you see this petal, it is getting overlapped from both the ends. This petal is overlapping from both. It is getting overlapped. It is overlapping these two petals. So when the margins of sepals or petals, right, when they overlap each other, but here there is not a proper direction, right? So this is basically the imbricate type of estivation. What sort of estivation is there? It is imbricate, cassia and gulmohar. What you have to remember here? You have to remember cassia as well as the gulmohar. Am I clear? Right? Sure? Any doubts? So this is imbricate. So next is this one, most important one. It is the vexillary estivation, and when the petals they show this type of estivation, then you people will see that there will be papilionaceous corolla. What sort of corolla will be there? There will be papilionaceous corolla. Are you getting my point? There will be papilionaceous corolla. So what does it mean? Here you will see the largest petal here. You will call it as standard or vexillum. And it is what? It is the largest petal. Then, see, this is going to overlap these lateral petals. So this is basically wings, or also known as LA. Remember, this is going to be the posterior side. All the points that I'm writing here, they are important, right? You literally need to remember this. Okay, then next petal, they are even fused with each other. See this. This is keel slash carina. This sign is showing the fusion here. And obviously it is towards the anterior side. Now, if I have to tell you, where do you see this? In the case of P, in the case of bean as well. So here, how many petals do we have? 5. How are you going to write it? 1 petal. It is showing that standard thing. Wings or LA, keel and carina, which are fused. So fusion, that's how you have to show it. 1 plus 2 plus 2. If, let's suppose, if in the exam, this is the MCQ that they give me, that what is the meaning of this? You should be very, very clear here that here we are talking about vexillary estivation. That is, we are talking about the papilionaceous corolla. Am I clear? Am I clear? So it is in the case of pea plant. So Leguminosae family is the part of our syllabus, so you should know about it. So in pea and bean flowers, 5 petals are there. The largest, the standard or the vexillum, it overlaps the 2 lateral petals, which are wings and LA, which in turn overlap the 2 smallest anterior petals. So if they are anterior, this is posterior. You have to remember on the basis of anterior, posterior also, question was there. So this type of estivation is known as vexillary or the papilionaceous. So pea and bean, that's how you will denote it. So this is the most important one. Look at the diagram, right? So this is your bean flower, this is your pea flower, right? Do check it, do observe it. Do check it, do observe it. Understood? So next is the endometrium, right? So we know that it is the essential male floral organ, right, composed of stamen. So this is the stamen. So in the case of stamen, this is filament. Are you getting my point? And this one is the anther. So that's what you have to remember here. See, this is flower. That's thalamus or receptacle. That's how you are going to attach it. So anther and filament, that's stamen. They will form what? Stamen. So this men is telling you it is the male part, hai na? So how are you going to denote it? Like this. So actually this filament, it is the proximal end of this filament, right? So proximal end of filament is attached to thalamus. And this one is what? This one here is the distal end. So that is having the anther. Yes, we need to know that detailing. Now already here you know it is attached to the, here you know it is attached to the thalamus. So let's suppose if it is attached to the petal, If this is petal and it is attached to the petal, so the condition will be epipetalous. Just now I discussed it and I'm recalling, right? I'm revising it again, right? So I'm helping you guys to revise. So epipetalous condition. That's the, right? So depend on the number of androecium, right? So you will write it here. So I'm showing you this. And let's suppose if it is fused with the tepal, as we are going to see in the case of lily. So then what is the word? Epitepalous or epiphyllous condition, or epiphyllous condition. Am I clear? Am I clear? So this is the proximal end of the filament. This is the distal end. So it is attached to the thalamus with the help of its proximal end. If it is not attached to the thalamus, might be attached to the petal or this sepal. But it is sometimes, you know, and male and female part, they are also fused, right? That type of gynandrous condition is also there. Okay, so look at this lily, which is showing the epiphyllous condition. So if you have, if you will ever get a chance to see the lily flower, right, it's very common. So you will try to open it. You will very clearly, you will see this part, right? You will very clearly, you guys will see this part. Okay, right? So now types of stamen. Now let's suppose, again, important information, of course. So in the case of stamen, if there are many stamens, let's say if the word is polyandrous, poly, you mean, you know that, free. Actually, there are two possibilities. Either in the stamen, right, sometimes what happens that all the filaments, they are fused, but this anther part is free. And that is also the condition that anthers are fused, but the filaments are free. Both the types of conditions are there. Both type of conditions are there. Just see, if I'm using the word polyandrous, I'm saying stamens remain free. Poly, we have used this word. Then it is synandrous when I'm saying that stamens, they remain united. Stamens, they are united. Now, how are they attached? Yeah, what are the conditions here? See, firstly, we'll talk about the we'll talk about this cohesion, right? Just let's suppose if the word is adelphous. This is important, very important. If you will understand the meaning of words, you will be able to remember that, right? If the word is adelphous, what's the word? If the word is adelphous, right? Yes, adelphous means, what is the meaning here? This is the part of your syllabus. Ki filaments are fused, but anther are free. All filaments are fused, but anther are free. This is the adelphous condition. Now, monoadelphous. Mono means single, like all the filaments fused into a single bundle, but anthers are free. The best example of it, you guys can see in the case of this China rose, right? Look at this China rose. These yellow colored, they are what? Anther. All that anthers are free, but their filaments are fused here. Their filaments are fused here. So China rose, it shows monoadelphous condition. So when you find this word adelphous, what you have to understand this thing, that filaments are free, are fused, but anthers are free. Filaments are fused, but anthers are free. Filaments are fused, but anthers are free. So monoadelphous, all the filaments fused into a single bundle, anthers free. Example is China rose. Diadelphous, very important. Diadelphous. Di means, I know there will be two bundles. So in pea plant, you'll see it is the condition. In pea plant, the androecium shows this particular condition. This particular condition, it means, what does it mean? What does it mean? So it means ki here we have two bundles, and in one bundle there is only one stamen, and in second bundle you'll see 9 fused stamens are there. So that is how we represent it. So pea plant mein, when it comes to corolla, that's the condition you have to remember, because this family is the part of our syllabus, and androecium. So this is very important. Very important, okay? Then polydelphous means stamens form more than 2 groups. Citrus or castor, they are the examples. So please write down. Done? Is it clear? Please write down. Quick. Quick. Guys, quick. Let's continue. So that's what you have to remember, okay? So now the next thing is stamens free, done. Now the next important information is didynamis and tetradynamis. So though what is happening here, ki polyandrous condition mane stamens are free. Now what sort of arrangement they have? Just let's suppose if I'm using the word didynamis, here you will say that 4 stamens are there. How many? 4 stamens are there, 2 long and there are 2 short, and they'll be arranged in 2 different whorls, right? Like here you can even write them like this, 2 plus 2. So 2 are long, 2 are short. Then tetradynamis, again very important, right? Mustard example you will discuss in the Brassicaceae family. So here you will see 4 are long, 4 are long, 2 are short. This is tetradynamis condition. So do not confuse this word. Ki tetra it is, so means 4 stamen. No, total stamens are 6. So 4 long, they'll be in center, and then here you will see short. So they are present in, they'll be present in 2 whorls. They say outer whorl will have 2 short and inner whorl will have 4 long. So you can write it like this. You can write it like this, tetradynamis condition, right? Tetradynamis condition. Understood? Understood? So told you already about epipetalous condition, Solanaceae family members, right? Just brinjal, even sunflower also shows that condition. So epiphyllous or epitapylous in asparagus, lily, or gynandrous means stamen fused with the gynoecium. They'll form gynostegium. Not given in NCERT, but still if you want you can remember. Example is calotropis. Example is calotropis. So here you people can see the example or the diagram. China rose. So you see this yellow part is what? This yellow part is your Anther. Filaments are attached together. They are forming one bundle here. Am I clear? They are forming one bundle here. Am I clear? Am I clear? Right? So here you guys can see, right, the one is the free stamen, 9 are fused together. So 1 plus 9, this condition, you see it in the pea plant. So pea plant is also having papilionaceous corolla. Understood? It is also having papilionaceous corolla. Then polydelphous stamen is there. Then look at this condition, 4 long, 2 short stamen in the mustard, salvia. There is the variation, right? So here you can see this example that they are showing. It is of the mustard, right? Tetradynamis. Okay. Understood? Done? Done, beta? So salvia will have didynamis condition and the mustard will have tetradynamis condition. Mustard will have tetradynamis condition. Am I clear? Sure. Now let's talk about the gynoecium. Okay. So should I continue or do you need a break? Should I continue or do you need a break? Chalo, let's continue then. So gynoecium, right? Here what you have to remember, see, stigma, style, ovary. We know that it is the, what is it? It is the lending platform. It is the lending platform for pollen grains. If there will be compatibility, you know that pollen grains will germinate. So this can be solid or hollow, tube-like structure that is, you know, connecting these two. Ovary is what? It is the basal, yes, swollen basal part. Understood? What is this ovary? It is the swollen basal part. So here in this ovary, you people will see, right, this cavity here, right, this ovarian cavity. What do you call it? This ovarian cavity, it is also known as locule. And it's not meant, and here in this ovarian cavity, we will see what, beta? We will see ovule that after fertilization will form seed. Am I clear? That after fertilization will form seeds. Is that clear? Right? So now if you will focus here, see, in the case of an ovary, ovary is not always unilocular. It can be unilocular. So unilocular means what will be there? There will be single locule, right? If bilocular, then there will be 2 locules. And if it is multilocular, we know there will be many, right? There will be many cavities. There will be many locules, right? That is what you have to remember, right? That is what you have to remember, okay? Right? So generally that is what we see, that if there is one ovarian cavity, generally we see that there is one ovule, and we know that that will form the seed later. Just say, let's suppose if I'm taking the section of a fruit, because fruit is ultimately ovary. If it is showing you something like that, it means it is having many locules. But here you can see each locule is showing one seed, means each locule is having, mostly we see that they will have one ovule only. Understood? So this is what the gynoecium is. So just have a look that it is the, gynoecium is what? It is the female reproductive part of the flower made up of one or more carpels. So carpel is having stigma, style and ovary. So ovary will have what? One or more ovules attached to the flat cushion-like placenta. So ovary bears what? It will have one or more ovules. To what they'll be attached? They'll be attached to a cushion-like structure that we call as placenta, through which the nourishment will be provided, right? Okay, so here you can see this thing very clearly, that when you are talking about the stigma, usually at the tip of style, and it is the receptive surface of pollen grain. Style will connect the ovary to stigma. Ovary is the enlarged swollen basal part, right, on which lies the elongated tube, the style. Is that clear? This is the diagram. These are the part. So give it a reading quickly. Give it a reading quickly. Okay. Done. So the next is what? Next is the types of carpel. See, in a flower, either you can see there is one carpel, so then the flower will be monocarpellary. Then the flower will be monocarpellary, means there is single carpel in it. There is single carpel in it. Am I clear? What is there? There is a single carpel in it, so it is the monocarpellary. But flower can also be multicarpellary, or you can also call it as polycarpellary. So it means there will be many carpels, है ना? Many carpels. Now what happen? If there are many carpels, there are two possibilities, right? There are two possibilities in the case of many carpels. Either you will see apocarpus condition, or you will see syncarpus condition. Apocarpus or syncarpus condition, right? Apocarpus, from A, you can remember alone. Like there are so many carpels, they are living alone, means they are not fused. So here you have to remember carpels are free. You have to remember carpels are free. So syn mane fused. So here you have to remember that carpels are fused, right? Carpels are fused. Am I clear? So here you have a trick, that is ma'am rose low. Here you have trick HPMS. So it is as per your class 11th and 12th examples. So ma'am rose low or HPMS. So M stands for, of course, multicarpellary here. A stands for apocarpus. M stands for mycelia. This example is given in your class 12th. Then rose will be for rose only. Low will be for lotus. And here, H stands for hibiscus. That is your China rose. P stands for papaver. And M stands for, of course, multicarpellary, syncarpus. Right, multicarpellary, syncarpus. Understood? Understood? And these are the examples which are given in 11th and 12th NCERT. But syncarpus, it is even there in the case of mustard, right? Even in the case of tulip, you can see that syncarpus thing. Is that clear? Okay, done. So please write down, quick. Please write down, quick. Okay. Noted? So types of carpel, even here also you can see the example. In syncarpus also I told you the examples. So next is the placentation. So before starting the placentation, I would like to discuss about the ovule thing, okay? Right, let's talk about the ovule and then see, let's suppose though you discuss it in class 12, sexual reproduction in flowering plants, I'll discuss it here only. Let's suppose this is the ovary, this part is showing the placenta, and here you have the ovule. So ovule is present in the ovarian cavity, which we are also calling as locule. Am I clear? Which we are also calling as locule. Am I clear? So what you need to remember here, right? That's the first thing that we need to remember here. Just see, this is the space that we have. Yes or no? So this is the ovarian cavity or the locule, है ना? Now to the wall of this ovary, like this ovarian wall, what will you see here? You people will see that the placenta is, like I'm just showing you this, right? So placenta is present at these ovarian walls, like different arrangement of placenta is there. You like see it in this way that okay, to this ovary wall the placenta is attached. Now a stalk-like structure will arise from this placenta, right? A stalk-like structure will arise from this placenta and it is going to touch this main body of the ovule, and this ovule here is covered by, right, these coverings. Now let's discuss it. So this stalk-like structure here, it is known as funicle, that is arising from this placenta. Now the part where it is touching the main body of the ovule, you are calling it as hilum, H-I-L-U-M. What do we call it? It is hilum, H-I-L-U-M. So hilum, we also define it in this way, that it's the junction between funicle and ovule, and you can also write it in this way, that it's the part, it shows the part where funicle is touching the ovule. Where funicle is doing what? Where funicle is touching the ovule. Now when you look at this main body of the ovule, okay, if you see this particular part here, right, if you people see this particular part here, this is the parenchymatous, right? This is what? This is the parenchymatous. This part, it is the parenchymatous. This one I'm talking about, tissue, right? This is the parenchymatous tissue and we call it as nucellus. It is the parenchymatous tissue, right? This is the nucellus, which is fully loaded with the, it is diploid, which is fully loaded with the reserve food material. Okay, so obviously megaspore will develop here, that will form the embryo sac later, so that will get the nourishment from this part, right? So here what will you see? You will see the nucellus. Now can you see these coverings here? These are integuments, right? These are the coverings. So we have outer integument here and inner integument here. So because in angiosperms mostly you will see the ovule is bitegmic, that is it is having two integuments. Now when you look at the area where this integument is not covering this nucellus part, it is micropyle or micropylar end, and opposite to it will be callosal end. Opposite to it will be callosal end. That's what you see in the ovule, and this is the ovule that will ultimately transform into what? This is the ovule that will ultimately transform into what? It will transform into the seed after fertilization. After fertilization. So if I have to show you that diagram, let's say, right, let's say I'm telling you this is funicle. Right, this is funicle. So just now I told you that the part where it will touch this will be hilum. junction between this funicle and ovule, and this tissue here this is what? This is nucellus, right? Having reserve food material. This one here is outer integument. This one here is inner integument. Okay, so this end is what? It is the micropylar end, and opposite to it will be callosal end. Got that? Right? That's a very simple type of ovule. Okay, very simple type of ovule, a tropous type of ovule. In sexual reproduction in flowering plant, you talk about anatropous ovule, but as of now, this is what you have to understand. And you know that it is in the ovary, ovarian cavity, right? So different types of arrangement of placenta as per that ovule is arranged. So this part we will discuss after a break. Okay, so let's have a break till 5:45 p.m. Okay, so half an hour ka break. Then we will finish this placentation, fruit, seed, families, and then we will also start the anatomy part today. Okay, the rest of the portion of the anatomy we are going to finish today only. Finally. Hi everyone, am I audible? Am I audible? Yes ma'am, but you look very scary. Now it's fine? Okay, so guys, again the power issue was there, but I got lucky. Right now it is resolved. So after the session, we'll cut these breaks so that you can get this one shot in continuity. You can see this lecture without the breaks. So let's continue this part. So tell me, up to this ovule thing, everything clear? Everything is clear up to this part? Today we will finish the anatomy as well. Yes, please. Am I audible? Am I audible? Guys, am I audible? Okay, chale. So this is the ovule thing, right? This is the ovule. This is where you have to pay the attention. So I hope this diagram is clear. Nucellus part, outer and inner integument, micropylar a part opposite to it, callosal end is there, funicle is there, and then here you have the placenta, you have the ovarian wall. Okay, so sorry for this thing, right? Due to the glitch itself, due to the power glitch, right, I was not able to start my class at 5:45, but okay, don't worry, but we'll finish this chapter, and after that we'll also start the anatomy. So let's talk about the placentation. Let's talk about the placentation. So when it comes to the placentation, it is the arrangement of ovules within the ovary. What is it? It is the arrangement of ovules within the ovary. Is that clear? This is what placentation is. I'll tell you why I taught you the structure of ovule here. We'll discuss that too. So in the marginal placentation, the very first placentation is marginal. See students, first of all, this is a very easy chapter. I know there are a lot of things which you have to remember here. I am aligned, but it doesn't mean that you should leave this chapter. Every day, please, 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 like I am this time, so I'm just begging, right? I'm not requesting, I'm begging. Please, every day, sit for 1.5 hour for the biology itself, in which revise diversity, revise morphology, revise diversity, revise morphology, and whatever part bothers you, just pick that up, number one. Number two, that I have to discuss here is ki in the morphology, if you are still planning, ki, ma'am, I'm not able to retain, do not forget the leaf part. The Phyllotaxy gave you the trick there, right? The simple and the compound leaf. Okay, venation thing, very easy. Then in the flower, the complete flower topic is important, right? So if you have to start the revision, start from the flower part, right? Start from the flower part. Just keep it in your mind, okay? So marginal placentation, this is the example. We all have seen this, right? You know this is the pea pod, okay? What's that? It is the pea pod. Is that clear? So these are actually what? These are the ovules which after fertilization are turned into the seed. Can you see that? This is the pea pod. So it is a fruit basically. It is what? It is the fruit basically. So in the marginal placentation, the placenta will form a ridge along the ventral suture of the ovary. The placenta will do what? The placenta will do what? Yes, please. Placenta will do what? Right? It will basically, right? It will form a ridge along the ventral suture of the ovary, and the ovules are born on this ridge, forming two rows as in pea plant. So here also you will see that the ovary is monolocular, or I would say the ovary is unilocular, right? Unilocular. The ovary is having the single ovule, right? What will you see here in the ovary? Ovary will have the single locule, and obviously here the flower itself will be monocarpillary, right? The flower itself will be monocarpillary. Am I clear? Sure? Sure. So in marginal placentation, the placenta will form a ridge along the ventral suture of the ovary, and the ovules are born on this ridge, forming two rows as in pea plant. So this is the pea pod, right? And do you see, like whenever you see these pea seeds, you will see there is a kind of scar there, right? That scar is actually hilum. What is that scar? That scar is actually hilum. Am I clear? Yes, am I clear here? So that scar is what? That scar is actually hilum. Understood? Sure? Done? Okay. So this is what we have. So next is the exile placentation, my favorite. I always say this line. This is my favorite placentation. Here you will see, beta, that placenta is exile. Look at the arrangement here. Right. And the ovules are attached to it in a multilocular ovary. The ovules are attached in it in a multilocular ovary. Is that clear? Is that clear? So here I'll give you one example and I'll demonstrate it rather. Right? I'll demonstrate it rather. Just a minute. Okay? Right? So when the placenta is exiled and the ovules are attached to it in a multilocular. So that the marginal one is what? It is the unilocular ovary. This is the multilocular ovary. So here you will see what? Right? Here, what will you see? So here you guys will see that this is what we have. Can you see that? Can you see that? This is what you are going to see here. Am I clear? Am I clear? So these are the locules, right? So these are the locules which are having these seeds. So this is basically showing what lemon. So lemon, China rose, tomato, this is given here, right? Even when we discuss about the families, we talk about the, right? We talk about the Rutaceae family, right? Citrus family is actually the Rutaceae family. So that there you will see the exile placentation. Got it? Done? Okay, so I'll give you one demonstration here. Allow me 1 minute. So guys, when you see this part, right, just say this is the tomato. Now when you cut it like this, okay, when you cut it like this, that's what we see. This is what we see. Yes or no? This is what we see. So in this tomato, this part is what? Obviously outer covering, right? Ultimately it is the ovary that will turn into a fruit. So this outer covering, this ovarian wall, this is the pericarp here. So when you look at this part, this is the placenta. And we know that, right? If you will see this, just have a look. Just have a look. Right, these are the locules here. So usually I said, yeah, generally we see that one locule will have one ovule, but it can be more than that also. So here if you will see, this is the placenta, and here see the exile. Why do we call it as the exile placentation? Look at the arrangement here. You can do the same with the lemon. Right, you are going to see the structure like this. So why I'm bringing this tomato? Because remember, in the tomato, what is the part that is the edible? It is the placenta that we eat. Right, in the tomato, what do we eat? We eat the placenta. So that is again a previous year question. Got it? That is a previous year question. So it is the placenta that we eat. So here you see, this is the placentation. So these seeds, what are they? They were ovule. After fertilization, they turned into seed. So that is how the arrangement is, right? That is how the arrangement is. So here you can see 2 locules are there, right? It can be 3. But in the lemon also, right? This is representing each locule. Here you have the seeds. Right, here you have the seeds. Can you see that? Done? Okay. So when the placenta is exiled and the ovules are attached to it, a multilocular ovary. So China rose, tomato and the lemon. China rose, tomato and the lemon. That is what we have. Okay. Now, this is the parietal placentation. Right. Diagrams need to be very clear, students. So this is the parietal placentation. Is that clear? What is it? It is the parietal placentation. So this one here, you will see at periphery, these are your ovules which are arranged here. So initially the ovary is unilocular, but later on there is the formation of a false septum which we call as rhapillum. Right, later on there is the formation of a false septum. It's not a true septum, right, because it will form later. So there is a false septum that will form here. We call it as rhapillum, right? We call it as rhapillum. Is that clear? Is that clear? So the ovules develop on the inner wall of the ovary or on the peripheral part. So ovary is one chambered, but it becomes two chambered due to the formation of the false septum repulum. Okay, so number of placental ridges correspond to the number of fusing carpels. Is that clear? Is that clear? Understood? So mustard, argemone, this is what you have to remember. Parietal placentation it is. Then comes the free central. So this is the free central. So in exile, actually the locules were there, but here it is the free central. So ovules are born on the central axis, septa are absent, dianthus and primrose. And then beta, here you will see gynoecium is polycarpellary and they all will fuse to make one locule. Okay, so multilocular we have discussed in the exile, and in the parietal later the ovary becomes bilocular because there is the false septum repulum there. Understood? Now the basal placentation, the most advanced one. So placenta develops at the base of unilocular ovary and the single ovule is attached to it. Is that clear? Right, so sunflower, marigold, wheat and maize, they are showing this placentation. Understood? Okay, so next is the fruit. Okay, but as I told you that I will discuss about the special type of inflorescence too, right? You should know about these three special type of inflorescence. You know, there is a PYQ on the basis of fig as well, right? So I'll come to that also. There are, so special type of inflorescence, that's what we are talking, right? See, this is what I'm trying to show you. Just I have already discussed that with you all. Euphobia, है ना? I told you about the euphobia, isn't it? Isn't it? I told you about the euphobia, remember? Remember? So when I was telling you about the euphobia, I told you that they look like flower, but it is not the flower, it is the inflorescence. So if you will check it, right, if you will observe it carefully, these are not the petals, these are the brackets, and here you have the cythium. So what exactly is the cythium? I'm not asking you to remember this complete paragraph, right? At least you should know these names as per me, right? It's my hunch that you should know about these three special types of inflorescence. So cythium inflorescence, example is euphorbia, told you already. We have discussed it before, right? In the case of phylloclade as well. So here it looks like flower, but it is a special type of inflorescence. When you talk about its structure, it's small conical receptacle surrounded by an involucre, which is a cup-shaped structure, right? Generally having the 5 brackets, right? Generally having the 5 brackets. Is that clear? Right? So basically that, in the surrounding, the part that you see, that are not the petals. They are basically what? What are they? They are the brackets. Now, male flower, right? They are many, naked, with one stamen and the long pedicel arranged centrifugally. Okay. Female flowers, beta, centrally placed, consist of gynoecium, of course. They are also pedicellate. Pedicel is there. So what is the unique feature? It looks like a single flower, but actually it is not a single flower. It is an inflorescence. So unisexual flowers you will see there are no petals, no sepals. So that are the brackets. That brightly colored are what? They are the brackets. Am I clear? Right? So that is how they are showing. Male flower is there, female flowers are there, right? Ovary style, everything is there. Then comes the hypenthodium. So hypenthodium you see in the case of fig, right? Fig, anjeer. Do you know anjeer? Right? That is the fruit that we thought that is a fruit, that is actually an inflorescence. This is hypenthodium, right? Even in the sexual reproduction in flowering plant, if you remember, we discuss about not pronuba mouth here. So fig and wasp. In NEET 2025, there was a question on the basis of that. So I'll discuss it here also. Okay. So actually, what is this hypenthodium? This hypenthodium, it's a ficus. It's a fleshy, flask-shaped receptacle with an opening called a pore or ostiole. Is that clear? It's a fleshy, flask-shaped receptacle. What is it? It is a fleshy, flask-shaped receptacle with an opening called pore or ostiole. So inside the receptacle, you will see male flowers are near the pore, female flowers at the base, gall flowers, which are the sterile flowers present in between. So here you will see, even in the case of people, that is Ficus religiosa, banyan tree, Ficus bengalensis, bengalensis, Ficus carica, that is Anjeer. So Anjeer, I thought you all know about it. So here I'll discuss one question on the basis of that, but that one I'll discuss. Like there was a question in NEET 2025. See, when we discuss about this fig, right, fig and the wasp, they this wasp help in its pollination. What happen is ki wasp, right, this wasp will enter through this pore, right, and that wasp is going to lay the eggs there. Now the flowers in which these larvae are developing, they are the gall flowers, right, because they have the larvae there. So here overall, if I talk about the fig, right, overall if I talk about the fig, so fig shows protogyny, right? What that fig shows? Fig show protogyny, right? What is the meaning of protogyny here? What is the meaning of protogyny here? So protogyny means ki in the fig, right, inside this fig, inside this hypenthodium, you will see that female flowers, they develop first, right, they develop first, they become mature first, then the male flowers. But that is not the case. The actual topic that I'm going to discuss here is that this wasp is going to lay the eggs there. So the flowers in which having these larvae are developing, they'll be the gall flowers. What will you call them? They'll be the gall flowers. What will you call it? They'll be the gall flowers. Is that clear? So when you talk about that gall flowers, na, they have these larvae. So what will happen from that larva? What is going to happen? Male will come out, right? And then female insect will also come out. Male is wingless, will not get a chance to go out. So male is going to fertilize the female wasp there. Now that female, right, somewhere we also write ki female also lay the eggs. Female get after this meeting, female will also start laying the egg there, or female, right, will create a pore, and through that female will leave. It will go to some other hypenthodium, will release the eggs there. That is also the case. Now the question that was there in the NEET 2025, they asked us ki fig and wasp, like wasp help in the pollination of the fig, and obviously when that wasp will move from one this fig fruit to another, fig hypenthodium to another, then obviously it will conduct the pollination. Now the point is that when the other statement was, it was a statement based question, two statements were there ki fig is the non-vegetarian fruit, right? So even when I solved the paper, I marked it correctly because this is what I believe. So as per the NTA has said ki fig is not a non-vegetarian fruit, right? But yes, vegan people, they still believe that it is a debatable thing, right? Fig is considered as the non-vegetarian fruit. Why? Because larvae are present there in that fruit. No doubt, right, the fruit is going to release the enzymes, it is going to, dissolve it, but still they are there, right? So many people, they don't prefer this fruit. They consider that insects are there in it, right? So they consider, vegan people especially, they consider it as a non-vegetarian fruit. But as per the NTA, fig is not a non-vegetarian fruit, right? Why? Because all that larvae, they'll be dissolved, right? They will be dissolved. Dissolved by what? They will be dissolved by the enzymes which fruit is going to release. So that was the question in NEET 2025. I want you people to, revise that question. Okay? Done. So now the next thing is, right? Now the next thing is this part, okay? Verticillester inflorescence. In Ocimum or in Salvia, you people will see this. So it is found in Lamiaceae family, right? It is found in what? It is found in Lamiaceae family. So in the axials of the opposite leaves, just say if you see, this is leaf, this is leaf. Here you will see the bunch of flowers will be there. So in the axials of the opposite leaf, flowers appear in two clusters, each with 3 to 9 flowers. So each cluster, this line you don't need. So that is the verticillester. So Tulsi, if you know Tulsi, there also you see the verticillester inflorescence. Okay, right? So this is all about the special inflorescence also. Now we will talk about the seed, right? Now we will talk about the seed, then fruit, and then the families. Okay, seed, then fruit, then the families. So that's what we are going to discuss. Understood? That is what we are going to discuss. Is that clear? Is that clear? Sure? Yeah, but I will also teach the families. I'll tell you the trick to revise the families as well. Okay, chaliye. So the seed. So ovules after fertilization, I told you that they will develop into seed. and the ovary, it will develop into fruit. So this part, before starting this part, let me discuss few things here. Just see, in the ovule, we have outer integument, we have inner integument. So we have outer integument as well as we have inner integument. Now when you talk about the seed, so in the case of seed, what will you see? You'll see, right, this outer integument. Obviously seed will form after fertilization indeed, right? The seed will form after fertilization, right? So outer integument will form outer seed coat, that is testa. It will form outer seed coat, that is testa. And here you will also see inner seed coat, that is tegmen. Are you getting my point? So outer seed coat is testa, inner seed coat is tegmen. So that's what you have to remember. Is that clear? Right? This is what you see in the seed. So seed, in the case of seed, and one second. Firstly, I'll complete this part then. And you know that ovary after fertilization will turn into what? It will turn into fruit. So both that processes are taking place simultaneously, right? Both that processes are taking place simultaneously. Is that clear? Is that clear? Done. So ovary wall will form fruit wall, that is pericarp. Ovary wall will form the fruit wall, that is pericarp. Is that clear? That is pericarp. Now remember one thing, that this pericarp, it can be, if there is a fleshy fruit like your mango, then this pericarp will have three parts. It will have epicarp, it will have mesocarp, and it is going to have endocarp. Okay, then it will have endocarp. And if it is dry, then it will be pericarp only. Okay, so overall in a seed, what do you see in a seed? What do you see in a seed? You will see seed coat, number one. Number two is the embryonal axis. That's what we see in a seed. You'll see seed coat, and you already know that outer one will be testa, right? Outer one will be testa, inner one will be tegmen, right? Inner one will be what? Tegmen. So embryonal axis, what do you call it? You call it as tigellum. You call it as tigellum. So in the embryonal axis, right? Just say, let's suppose here I'll discuss it in the case of two types of seeds, right? So firstly, tell me, this part is clear, that ovule, its outer integument after fertilization will form testa, inner integument after fertilization will form tegmen, ovary after fertilization will form fruit, ovary wall, right, it will turn into the fruit wall, that is pericarp. So if the fruit is fleshy, just like mango, right, so this pericarp is having three parts: epicarp, mesocarp, and endocarp. Am I clear here? Sure? Sure? Now that seed. So seed is having seed coat. Seed is having the embryonal axis, which is tigellum. So here you have the testa, here you have the tegmen. Now you know that in the angiosperms we have two types of two classes rather, right? What do we have there? We have two classes rather. We have the dicots and we have the monocots, right? We have the dicots and we have the monocots. So dicots means, di means two cotyledons, right? Di means two cotyledons, right? Monocots means, we know that here single cotyledon will be there. So this part in detail you will understand when you will have the clarity of this development. So that we study in the sexual reproduction in flowering plant class 12th chapter. So here I'll give you an idea. So actually in the ovule, within that nucellus, you will have that embryo sac, female gametophyte, in which female gamete forms. Then later for male gamete will come, fertilization will occur, zygote will form, that zygote will develop. So when there is a development of this now, so zygote, it has to take the nourishment. So within that ovule, within that embryo sac, the endosperms form. So it takes the nourishment from there, right? Right, just say angiosperms, we know that double fertilization is there in angiosperms. In angiosperms, triploid endosperm forms. So that endosperm is the nutritive tissue. In the case of dicots, right, when the dicot embryo is growing, right, when that dicot, just to teach you this diagram, right? So two cotyledons are there. Dicots, in the case of dicots, when that embryo is growing, what that embryo will do? What that embryo will do? That embryo is going to, right? That embryo is going to do what? That embryo is actually, when that embryo will grow, it will take all the cotyledon, like it will take the entire endosperm, right? It will utilize that endosperm when it is growing, and all that nutritive tissue it will save in its cotyledons, right? It will save that nutritive tissue in its cotyledons. Are you getting my point? So let's suppose this is the embryonal axis. So this is the cotyledonary node. This is the cotyledonary node, the point to which the cotyledons are attached. Now above it and below it. So imagine it in this way. Just say, let's suppose these two arms of mine, they are the cotyledons. They are attached to a point, cotyledonary node. Just above it, it is the apicotyle that will terminate into plumule. Just below that, that is the hypocotyle that will terminate into the radicle. So obviously the part above here will be what? It is apicotyle. Now it will terminate here into this part. This is plumule, and I told you in the starting of the lecture it will form the future shoot. Now below it you have what? You have hypocotyle, right? What do you have? Hypocotyle, and that will terminate into what? That will terminate into the radicle. So plumule will form future shoot, radicle will form future root, right? Radicle will form future root, right? And plumule, we know that it forms future shoot. So this is what we see in the case of the dicots. Now here, just to clarify each and everything in detail, here देखो, this is the diagram which is given in class 11th NCERT, and this is the diagram which is given in class 12th NCERT. Are you getting my point? So this is in 11th NCERT, this part. This diagram is there in the 12th NCERT. So here when you look at the dicot seed, this is the dicot seed as well. This is embryo, two cotyledon, this is plumule, this is apicotyle, terminate into plumule, hypocotyle, radicle, right? So api means above, hypo means below, right? So this is going to be the plumule. Now in 12th, right, how to remember this 12th wala diagram, how to relate these two here. So imagine if this, right, imagine if I am, these arms of mine, they are the cotyledons. So imagine if I'll bend like this, I'll bend like this, right? I'm putting my two arms like this. So obviously I'm going to mark this point as cotyledon. The tip here is the plumule. This is hypocotyle, then radicle, which will be covered by root cap, right? Then the radicle, which will be covered by root cap. Okay? Okay? Done? Okay? Sure? Done? Fine. So that's how this diagram is. So root cap, you know that root tip is covered by the root cap. So here, this is the dicot seed. You can see it in pea, in bean. So hilum will mark that scar to which we will see that it is attached to the fruit. Just even in the pea, you will see that dot wala structure. That what is that? That is hilum. It is that scar which is telling us, okay, at this point I was attached to my fruit. And this is the micropyle for the exchange of the gases during the seed germination, and the seed coat is there. So this is the dicot seed. So dicot seeds, right? Exceptions are always there. So dicot seeds are also referred to as non-endospermic. Why is it so? Why do we call them as non-endospermic seeds, beta? Why do we call them as non-endospermic seeds, या X-albuminous seeds? Why do we call them as the non-endospermic seeds or X-albuminous seeds? Why? Why? Because during the development of the embryo here, right, they are going to utilize the entire endosperm, right? Right. So here endosperm will not be there in the seed. But yeah, there is an exception. Exception is what? You will discuss the castor. Scientific name is ricinus. It's a dicot, but still have, it's a dicot, but still have endospermic seed. Am I clear? But still have endospermic seed. Am I clear? Sure? Are you sure? Are you sure? Done, beta? Please write down. Okay, right? So exceptions are important. Now, in the case of monocots, what will you see? Right? In the case of monocots, there is only one cotyledon. So that one cotyledon, it is what? It is shield shaped, right? Present laterally, of course. So it is basically known as scutellum. Is that clear? What do you call it? You call it as scutellum. So imagine, just say, let's suppose I am the monocot. Then in the case of monocot, one, like this is the diagram. So let's see, this is the cotyledon scutellum. The another cotyledon, right? The reduced cotyledon is what? The reduced cotyledon is what? It is epiblast, right? It is epiblast. Am I clear? So here you will see one more thing, that your plumule will have this, hollow foliar structure. That is your colutile. Are you getting my point? Colutile. Here radicle, then have root cap, then have coleorhiza, right? So coleorhiza is, right, radical, root cap, coleorhiza. So there it is, colutile. So P for plumule, right? P for plumule, and P for, from that P you can remember colutile as well. Is that clear? Right? So in monocots, because they have one cotyledon, so in monocots you will see this thing, that in monocots what you guys are going to see, that the seed will be endospermic. Right, the endosperm will be there in the seed. So it is going to be endospermic. It is albuminous seed, right? So here we have an exception. So exception may you will remember orchid. Okay, so orchid is a monocot. Though the family Orchidaceae, it lacks endosperm. So orchid is a monocot, but still do not have endospermic seeds. Understood? Still do not have endospermic seeds. Understood? Understood? Sure. So orchid is a monocot, but still do not have endospermic seeds. Okay, so that's what you have to remember. Okay, so this is epiblast. So epiblast will have what? Epiblast is basically the reduced cotyledon. Keep it in your mind. Okay, it is a reduced cotyledon, right? So even I have discussed the seed. So this is again the class 12th exam diagram. This is bean, right? But this is kind of common part. This is for the onion, and this is your castor, right? So this castor is having endosperm, right? This here also you can see that. And this maize grain, this is scutellum. Okay, beta. So this is the endosperm in the seed, and this layer here, it is alluron layer, right? It is proteinaceous alluron layer. It is beta triploid. Why it is triploid? Because it is also formed from endosperm. So endosperm here is triploid. So if alluron layer, if it develops from that, it is also what? It is also the triploid one. Am I clear? It is also the triploid one. Understood? Right? Understood? So this is the pericarp here. So this is basically a type of fruit which is dry. So in dry fruits, like imagine dry fruit, always doesn't mean you're talking about almond, this cashew and all. No, dry fruits means where that, fruit, like I told you, fleshy fruit may the pericarp is divided. Some fruits can be dry where the pericarp is not fleshy, the fruit wall is not fleshy. So this, the seed wall and fruit wall, it gets fused. So here also it is not just the fruit wall. Here the fruit wall as well as the seed wall, both are, that is seed coat, both are fused. Okay, so this is endosperm, this is the alluron layer, which is again triploid, right? So that is, this diagram is from class 12th. So here you guys can see, right beta, the outermost covering of the seed, right? Just have a look. Outermost covering of the seed is the seed coat, having outer testa, inner tegmen. The surface of the seed has the micropyle in the case of dicot seed. This is NCERT, bache. Beta, if you have the notes, you can just listen to me. So micropyle, how will it help? Please have a look. It will help in absorbing the water at the time of germination of the seed, right? It helps in absorbing the water at the time of germination of the seed. So there is also a scar called hilum on the seed coat, right? So this is a point where stalk or the funicle of seed is attached to it, right? Where it is attached to it. So within the seed coat is the embryo that is consisting of an embryonal axis and two cotyledons. So embryonal axis. For that, you also use the term tigellum. Am I clear? Tigellum. Understood? Understood? No, beta, colutile is having that foliar sheath. This is plumule. Sorry, this is plumule. That plumule is having that foliar sheath, colutile. Right? It is not equivalent. So the cotyledon, they are often fleshy, full of reserve food material. So at the two ends of embryonal axis, radicle and plumule are present. Understood? So endosperm is a tissue that is formed as a result of double fertilization, provide nourishment to the developing embryo, right? So food is stored in this endosperm, in endospermic seed. In non-endospermic seed, food is stored in cotyledon, right? So the two types of seed we will also discuss, but let's see this monocot seed first, okay? Let's see this monocot seed first. Understood? So in the case of monocot seed, you know that just cereals, your maize, the seeds have single cotyledon. What are we calling it, beta? We are calling it as scutellum. Am I clear? So seed coat is membranous and generally fused with the fruit wall. Right? From the 12th class diagram, I told you this thing. So below the grain covering, embryo and endosperm are present. Endosperm is bulky. It stored the food, provide nourishment to the embryo. So monocot seeds are endospermic. Right? So here you can see the outer covering of endosperm. You remember the part that I marked here. This is alluron layer. So in angiosperm, you know that it is also triploid. So scutellum, it's single cotyledon. It is shield shaped. Right? Why am I calling it as shield shaped again and again? Class 12th NCERT that I'm covering here itself. Embryo, you know that it is having radicle, plumule. So colutile is a protective sheath that is enclosing the plumule. It is the hollow foliar sheath that is covering that, right? So protective sheath enclosing the radicle is known as coluoriza. So plumule, P here, R here. Just keep it in your mind, okay? Right? So this is about the monocot as well as the dicot here. Now let's talk about the fruit and then we will talk about the families. Am I clear? So we know that, right, fruit is a characteristic feature of the flowering plants. It is a mature or the ripened ovary which will develop after fertilization. So fruit may you will see pericarp as well as seeds. Understood? So fruits are of three types, right? Let me write it here. So fruit, eucarpic, parthenocarpic, eucarpic, pseudocarpic, parthenocarpic. These are the three types of fruits. So U means true. Here actually it is the ovary, and to be more specific, the superior ovary is turning into the fruit here, right? When it is pseudo means false. So any part other than ovary, forms the fruit. Okay, right? So here we have the trick Ambika, Sharma, cute. So A for Ambika, A for apple, Sharma says strawberry, cute, cashew. So apple, you know na, it is the thalamus that we eat, right? That is a fleshy thalamus that we eat, pseudocarpic. And then parthenocarpic, which are also known as seedless fruits, What do you call them? You call them as seedless fruits. Understood? Sure? Seedless fruits they are. So they are not developed due to fertilization, right? So they are developed without fertilization. So seed is the result of fertilization only. So here you don't have, so it developed without fertilization. So that's why they are seedless. So you guys can see this part here, eucarpic, true fruits formed from superior ovary only. Important statement it is. Pseudocarpic में false fruit formed from any part other than ovary. Another important statement, apple, strawberry, cashew. Parthenocarpic, seedless fruit. So these fruits are formed without fertilization. So seeds are absent. Example is banana or grapes. Done? So fruit, you know that characteristic feature. I told you what is parthenocarpic fruit. So ये देखो, in the case of mango, जैसे mango and coconut, they are the different types of fruits are there. So mango and coconut, what are they? They are basically the example of drupe. Right? They are basically the example of drupe. Right? Here you see stony endocarp. So let me explain this to you. In the case of mango, right, because mango is a fleshy fruit. Even your coconut, I'll tell you what you have to remember. Just wait. There's a mango, that pericarp is having three parts. Pericarp is differentiated into. What is pericarp? Pericarp is the outer fruit wall, which is differentiated into epicarp, right, the mesocarp. So it is the mesocarp that is fleshy and the edible part here, and the endocarp, right, in the mango, when you see that stony part, that covering which is covering the seed, right, that is basically the endocarp. So in the mango it is the mesocarp that we eat, right, so stony endocarp. In the coconut, right, this brownish part, this is also mesocarp, that coir, right, and this here, if you have seen that coconut, that solid part, that is endocarp, right. So fruit consists of a wall, right, or pericarp and seeds. They may be dry and fleshy. When pericarp is thick and fleshy, it is differentiated into outer epicarp, middle mesocarp, inner endocarp. So in mango and coconut, right, the fruit is known as drupe, right? Again, a previous year question. So they develop from monocarpillary. This is important. Monocarpillary superior ovaries and are one-seeded. Very important. So mango mein pericarp is well differentiated. It is a middle fleshy edible mesocarp and inner stony hard endocarp. So in coconut also, the mesocarp is basically fibrous. What's that? It is fibrous. So please read it and do let me know whether it's clear to you or not. Please note it. Okay, done beta. So now see here, this is the groundnut, this is the dry pericarp here, right? This is the dry pericarp here. So look at this coconut part, right? This is your endosperm, and coconut maybe you know that it is the coconut water is free nuclear endosperm. This edible white part is cellular endosperm. So this is the inner shell, this is the endocarp here, this is the husk, mesocarp here, and that is the outer shell. Okay, okay. So there are few things that I would like to underline here, right? That in the edible parts of some fruits, edible, eatable, just I told you tomato and grapes. It is the outer pericarp and the placenta. So in the tomato, you know it very well, outer pericarp is also edible and it is the placenta that is edible. Grapes may be obviously same thing. Now, cashew, because it is the example of pseudo carpic fruit. So cotyledon and thalamus is the edible part. Wheat and maize, they have starchy endosperm. Pea and gram, it is the cotyledon. Mango, mesocarp. Coconut, endosperm. And even in the almond cotyledon, apple, pear, strawberry, it is the thalamus, which is the edible part. Am I clear? Am I clear? Done. So this is all about the morphology part, and now what we have to discuss, we will go for the families. Okay, we will go for the families. So please write down, these are the families which are the part of your syllabus. So Malvaceae, also known as cotton family. So cotton, hibiscus, like I'll write the example here, China rose, your cotton, it belongs to this. And in Malvaceae family, you will see that anther, usually anther is bilobed diathecus. Here you will see it is unilobed, monothecus, and tetrasporangiate. So if you are in, bisporangiate. If you are in class 11th, you will understand it in a better way in class 12th. Then cruciferae, also known as Brassicaceae. This is basically your mustard family, right? This is what? This is basically your mustard family. Am I clear? Sure. Then comes the Leguminosae, Fabaceae. Where not technically it is like that, I'll explain you. So your pea and all, it will belong to this. So here in the case of pea, I told you vexillary estivation, diadelphous condition, right? Important points. Compositae, it is also known as Asteraceae. This is the sunflower, or you can also call it as daisy family. And here Leguminae, which is also the Poaceae, A-C-E-A-E. So it is the grass family. What's that? It is the grass family. So this is your syllabus, right? This is your official NTA syllabus here for the families. This is what you have to remember, and I'll tell you how and what. Okay, so out of these 5 families, one type of question that can come is, right, one type of question that can come is, right, 1, 2, 3, 4, 5, basically 5, right? Right, basically 5. You have 5 families here. So out of that, right, out of this, this is the Graminae only, right? It is the Graminae only. That is the monocot family. Am I clear? That is the monocot family. Am I clear? Sure. So firstly, write down these important points. Then we'll teach you what you have to remember. Done, beta? So do you know why do we call it as cruciferae? Due to the cross-shaped corolla. Due to the cross-shaped corolla here. That's why you call it as cruciferae. Okay, and here you know that what are the examples? Your mustard, radish, etc. So radish, mustard, you remember? Tetradynamous condition. Hai na? So you have to remember these points, okay? You have to remember these points, okay? Clear? So firstly, write down this, then I'll explain you the next part. Then beta, so beta, Leguminosae is the, right, Leguminosum is believed to be the old name, right? It is believed to be the old traditional name. Fabaceae is the modern name, but further, in this also there are different subfamilies. Okay, there are different subfamilies as well. Okay, so Fabaceae, why do we call it as Fabaceae? It is on the basis of the genus Faba, right? Got that? So here you know that in the Fabaceae, just keep your P example in your mind. So here you will see the fruit is pod, right? So then comes the star, this Asteraceae family. So I'll explain you, right? I'll explain you the floral diagram and floral formula. Okay? I'll explain you the floral diagram and floral formula. Done? Just 1 minute. Beta, if you talk about the papaya, papaya is not considered as the parthenocarpic fruit. Matlab papaya, no doubt we also see that sometimes papaya is also seedless. But papaya is not truly the parthenocarpic fruit. In the case of papaya, we also call it as parthenogenetic. Why parthenogenetic? Because in the case of papaya, right, the seed formation is there, but that will be asexually. So it is like that. Right, it is like that. So let's not confuse it. And Komal, stop spamming here. Right, I have explained it multiple times that either I will take anatomy today or I will upload the third part. Right, I think you are irregularly attending the session. Done? So this is the general points. Now, see, when you see this part, first of all, semi-technical description of the typical flowering plant. So from this part, you really need to know the symbols first, which we have already discussed when I was teaching you this part. So there is only one correction that, so there is only one correction here. Let me do that. See this. If it is bracteate, brackets, flower is bracteate BR. If without bracteate, EBR. Actinomorphic, this is the symbol. You know that it is the radial symmetry. Zygomorphic, it is the bilateral symmetry. So that's the sign. This, or this. Now, bisexual, staminate, pistillate. If, like I told you in the case of China rose, if you remember. In the case of China rose, I told you apicalyx is present. In the case of China rose, I told you apicalyx is present. So you can denote apicalyx by EPI or EPIK. So now when calyx denoted by K. So if sepals are free, if they are not fused, told you already, polysepalous condition. If brackets are there, brackets are showing cohesion. Gamosepalous condition is there. For corolla, you will see CN. CN is fused here. So polypetalous, gamopetalous condition is there. Then students, this is the perianth, if you remember. Right? This is the perianth here. So in the case of perianth, you know that, right? Even, 1 second. So in the case of perianth, if the tepals are free, polyphylous condition or polytepalous condition is there. If they are fused, gamotepalous condition is there. Is that clear? Right? Is that clear? So that's what you have to remember. So andrecium, we know that denoted by A. So polyandrous, if it is simple N, and if bracket is there, cohesion condition are there. Right, beta? Gynoecium, also same thing. So superior ovary, hypogynous flower, inferior ovary, epigynous flower. Ovary is half inferior or half superior. That is the symbol. Epiphylous condition, epipetalous condition, and gynandrous. Right. So tell me any doubt from the symbols. Because when I was discussing the flower initially, I told you already about that. Yes, please. Yes, please. Yes, please. Clear, beta? Symbols are clear? Okay, so now this, in the NCERT, they didn't explain this Brassicaceae or the Cruciferae family, but the floral diagram is given. We'll discuss that. We will discuss that. Okay. Just 1 minute, beta. Just 1 minute, then we will discuss it. Just allow me 1 minute. Guys, just allow me 1 minute, please. Something urgent came up. Okay, so this is the Brassicaceae family. I told you it is also the ACEAE, Cruciferae. Now here what you have to see, whenever you draw the floral diagram, this is something very important. If you do not draw it, then it will be incorrect. This is the mother axis. If in any floral diagram, if this mother axis is not there, right? If this mother axis is not there, then the diagram will be incorrect. The diagram is not going to be correct, then it is going to be incorrect. This point is clear. This point is clear. Now in this case, what will you see? right? When you look at this floral diagram and floral formula, देखो, in the families, right, from last the year in which they introduced these families, they didn't ask any question from that. After that, some symbols were there which were given in NCERT. So what sort of questions you need to prepare from the families part? You will only start preparing the families once you are done with your complete syllabus or the complete morphology. Number one, you should know floral diagram and floral formula, right? Of all the families, at least know floral diagram or floral formula, right? Because question can come from symbols. They can give you the floral formula and then they can ask you which family it is, because they will show the that peculiar feature of the family is there, right? So question can come from floral diagram and floral formula. They can ask you the symbol. They can even ask you the economic importance, might be of some member or of that family, right? And obviously you will find that answer in the options also. And the other than that, the peculiar features of that family. So these are the kind of questions that they can ask you, okay, that they can ask you. Okay, this is the first thing. So if you remember these two things, many things will be clear. Just I'll give you one example. Let's suppose if they'll give you this condition, so you should know that here you are talking about what? You are talking about the vexillary estivation, which is an example of the pea plant, and here that belongs to Leguminosae family. So this is how you have to prepare the families. First thing. Secondly, do not forget these two things because they are directly given in the book. Now, we know it very well, right? Just say if this is the symbol, you know that what are we trying to say? This is the floral formula. Actinomorphic flower. That's what you can conclude from the floral formula. Actinomorphic flower. It is the bisexual flower. Look at the sepals there. K. 2 plus 2. Why? Because they are in two whorls. Two outer, two inner. That's why 2 plus 2 is given. But they are not fused. Now, obviously, if I will see this part, corolla. You know that Brassicaceae, mustard, cross-shaped corolla, cruciform corolla is there, C4. They are also not fused. And if somebody will ask me the estivation, I'll directly call it is velvet. Now I'll talk about the andrecium. So mustard is the example of Brassicaceae, and I know that here the condition is going to be tetradynamous. I have to remember this. I have to remember this. And then it is basically what? It is the, yes, please, bicarpillary syncarpous ovary. So what is it? It is the hypogynous flower. Yes or no? Hypogynous flower, superior ovary. So that is what I have to remember. Are you getting my point? So focus on the floral diagram and the floral formula, and I'll share 2 PDFs in the Telegram group. Now come to this part, this diagram. It is of hibiscus. It is of China rose. Do not get confused with these, markings. These are I have done it with the pencil. Now, Malvaceae family, that is your cotton family. These are some points that I have mentioned. Obviously, class is dicot, reticulate, venation, taproot. Flower here is pentamerous. Calyx, polysepalous, polypetalous means K will be N. C will be N. They are free. So, numerous stamen, but monoidelphous condition is there. That is also we have discussed. So, this is important. Right, gynoecium superior and hypogynous. Inflorescence cymose, brackets, bracteate it is. Placentation is exile. Again, that was also in the NCERT. So fruit type, they can ask you, it is capsule. So calyx, velvet estivation, twisted in corolla. Both examples are important here. Then example, hibiscus esculentus, hibiscus rosa sinensis. So your okra is your bhindi here, cotton here. So now, in the China rose, if you remember, I told you about the apicalyx, right? I was showing you with the help of that diagram. I've clearly mentioned that this is beta apicalyx, right? Now, if you see this part, here dekho. So this is your actinomorphic flower. This is your bisexual flower. That's why I'm telling you to focus on the diagram only. Then it is your apicalyx, right? EPK is showing that apicalyx is present. So because this is a general formula, not specifically in the case of your China rose. So apicalyx, it can be 3 to 9. They can be fused as well, or it can be absent as well. So apicalyx, where we discussed in Malvaceae, in the cotton family, we have discussed the apicalyx. So either it is absent beta, or it is 3 to 9, or it can be fused. So here they are showing what? Here in this case, they are showing that sepals are what? As per the formula, what are we saying? That sepals are 5 and they are fused, right? So here they are not giving us any option, right? But here we are saying that it is polysepalous, so we can also say it can be gamosepalous as well, gamosepalous as well. Okay, okay, right, right, this is fused basically, gamo will be the word. And here you will see, here you will see adhesion as well. So petals 5 free or fused, and rhecium they are many and they are fused, monoadelphous condition. Gynoecium, right, this can be, we know that bilocular to multilocular, fused, and the hypogynous. Now look at this floral diagram. So this is showing us apicalyx. So apicalyx ka number from this particular floral diagram only, and this is mother axis, 1 2 3 4 5 6 7, right? It is around 7, right? So you see this sign is showing the fusion, right? This sign is showing the fusion. Okay, so 1 2 3 4 5 6 7. So if I have to write the formula instead of 3 or 9, if they'll give me this floral diagram, that might be the question. They'll give you the floral diagram and then they can ask you that tell us the floral formula for it. So obviously I'll simply write 7, and this bracket is showing fusion. So if I have to write the floral formula here, so this is what I'm going to do. Then it is the, then it is what? Then it is what? This is the calyx. So this is apicalyx, this is the calyx, fuse. So 1 2 3 4 5. So K5, but it is also fused. Now when you look at it, forget, please ignore these lines. So ye dekho, one side it is overlapping this one, it is overlapping this one. So obviously estimation I can clearly see it is twisted. So 1 2 3 4 5. So calyx 5, right? So this is twisted, and if you will see this line here, where are you fusing? With the andrecium. So I will do it. And andrecium, they are many, or they are also fused. Monoadelphous condition is there. Gynoecium, here also beta, it is multilocularly, right? It is multilocular, right? So that's how I can write also. That's how I can write also. Are you getting my point? Are you getting my point? Right? So even if it is 1 2 3 4 5. So done, Malvaceae. Okay. So important examples, right? You have to remember Hibiscus osculentus, right? This Gossypium, right? So next is Cruciferae that we have already discussed, and Leguminosae. So Leguminosae mein, right, what you have to remember, brackets are present. BR is telling us that it is bracteate, it is zygomorphic. So from the floral diagrams, all the floral formula, remember which one is zygo, which one is actino. So this is bisexual. 5 sepals are there which are fused, and this means vexillary estivation. Yeah, papilionaceous corolla, this means diadelphous. So that's what you have to remember. So see, 1 2 3 4 5, this is showing the bracket, and see, look at this 1, then 2, these 2 are fused. So this one is single, these 9 they are shown as fused one, and gynoecium it is. Okay, so your gram, arhar, right, moong dal, soya bean. So this indigo fera, from this dye is obtained, right? In the old NCERT you will get this family. So dye, it will give us the dye. So sun hemp, again here from this the fiber is having economical importance, right? So sweet potato, millet tea, right? All that are the examples. So please pay attention on the examples. Now Astraceae, right? Again the dicot family, right? Here this is the next that we have to discuss. So here you might get confused. So let's pay the attention here, right? So let's discuss it, okay? Let's discuss this part quick. Done. So here you will see one thing in this family, that is pappus. This is what you are going to see. What is this pappus? This pappus is the modified calyx, right? This is the modified calyx into some hair-like or feather-like structures, into some hair-like, yeah, feather-like structures that help in seed dispersal. That help in seed dispersal. Am I clear? So here you will see pappus. Now in this case, I told you ray floret is there, है ना? So ray floret, right? We discussed that when we were talking about the flowers. So you know that either, just say ray floret, here it is showing G, that is gynoecium. So it can be having the female part, right? Or it can be neuter also. Disc floret mein androecium gynoecium. So it is bisexual, right? Sterile means sterile only. So ye dekho, K, here they are showing P, they are talking about the pappus, right? So bracteate, zygomorphic, bracteate, actinomorphic, bracteate, again zygomorphic. So disc floret is also showing epipetalous condition. So this is how you will be able to remember this. Is that clear? Sure, sure. Baki beta, here what will you see? So this is in the case of Graminae poaceae. Okay, I'll upload one more PDF here. So this is in the case of Poaceae gramina family, monocot family. Okay, so you know that in the monocot and the grasses, the stems are going to be like culm, right? Culm. We have discussed this word, isn't it? Right? So the leaf base is going to be sheathing leaf base. Whatever you have discussed so far, na, that is what you have to write. Sheathing leaf base is there, right? Sheathing leaf base is there. Or here you will see, if you will talk about the fruit. So fruit is basically a grain, caryopsis. This is the fruit type. So these are some, quick revision features. Okay, caryopsis. So perianths are also seen there. So tepals are also the lodicules here. So here, what is the term that you people use? You use the lodicules. But when do you need to remember this? You will only start this part. You will only start this part when you will finish the rest of the morphology. Am I clear? Right? So these lodicules here in this, just in the Asteraceae family, you talked about the pappus. Here you will talk about the lodicules. So they are nothing. They are some small fleshy scale-like structure present at the base of ovary. They are small fleshy scale-like structures present at the base of ovary, lodicules. So here you talk about what? You are going to talk about the lodicules, beta, right? So that is it. This is what you have to remember. So they are nothing but they are just the reduced perianth, right? This is the reduced perianth. Okay, so it help in opening the flower, right? So it is kind of reduced perianth that help in flower opening. That's it. So here just pick up the important points. So I'll share 2 PDFs in our official Telegram group where you'll find the direct points, and there is one PDF which is quite detailed, but there you will see some diagnostic, important, peculiar feature of this families. So this is all about the morphology, right? So tomorrow I'll ask, tomorrow to it's Sunday, so day after tomorrow I'll ask my team to cut the breaks in between, so you will get a complete one shot there, right? Then it will be very easy for you for your quick revision. So this chapter we have completed in, I think, around 4 hours 45 or 50 minutes, if I'm not wrong. Okay, so this is it. This is all about the morphology. Many students, they demanded it, that this is not on the channel, so put this chapter. So that is it. So the next part that we have to start, that we are going to continue here in this particular session, is anatomy, right? It is the anatomy of flowering plant. So now we will talk about the anatomy of flowering plant. We'll continue it here. So anatomy of flowering plants, right? Anatomy of flowering plants. So we are already done with the tissue part, right? I have already shared a recorded video on our channel. So now today we will start the tissue system. So you guys will get a break for 10 minutes only, not more than that. So it's 7:30. So let's meet at 7:40. Okay, then in one go. Guys, so after this, you will not get any break. Then in one go, we will finish it, okay? Then in one go, we will finish it. Ready? Chaliye guys, so let's continue. Ready? So what do you know about the tissue? So this is something that you have to tell me. This is something that you have to tell me. Quick. This is something that you have to tell me what is tissue. Then I'll continue this part. Now there will be no break. Okay. Now there will be no break. All the breaks are over. Now we have to finish the tissue system thing. What do you understand with the word tissue? If you will not answer, then I will not continue. Yes, please. What's tissue? Very good. Okay, so yes, you are right. It is the group of cells having, this part is already over, so I will just give you the overview. So it is the group of cell having similar structure and function. Structure and function can be dissimilar, but origin always need to be same, right? So after that, we discussed about the meristematic tissue. This is the flowchart that we covered in the last class, right? Plant tissue, mein we know that meristematic tissue is there, which is actually the group of actively dividing cells, and they are responsible for the continuous growth of the plant. We know that when we talk about the meristematic tissue, we know it very well, students. Yes, so we know it very well that the cells are undifferentiated here. They are just dividing. They are in the continuous state of division. Cells are immature. Cells are small size, metabolically very active. So because there is high metabolism, so obviously there is no reserve food here, right? So generally they are small. They are isodiametric with dense cytoplasm, and there will be a large prominent nucleus. So vacuoles are usually absent, and if they are present, my dear students, they are very small in size. So they have only primary cell wall, which is going to be thin, flexible, which will be made up of cellulose. Abundant plasmodesmatal connections are there, and because they are compact, no intercellular spaces will be there. So non-living substances, ergastic substances, they are absent. Is that clear? They are absent. Is that clear? Is that clear? Right? So the next part is what? It is the primary meristem. it is the secondary or lateral meristem. So primary meristem mein, this is the apical meristem, this is the intercalary meristem. So apical meristem, it is at the tips, right? Shoot apical meristem, it exactly at the tip, but root apical meristem is subterminal. Intercalary meristem, it is present in between the permanent tissue. Where is it present? It is present in between the permanent tissue. And in secondary meristem, we know that primary lateral meristem or secondary lateral meristem is there. So these are the examples. Now, today we will discuss it in the tissue system. Okay. So if anybody is having any doubt, guys, I have already discussed this chapter. I have already completed this chapter. You can see the video section. You will get the detailed explanation of this particular part there. Okay, so vascular cambium, cock cambium, intravesicular vascular cambium, intervesicular vascular cambium is there, right? And when it comes to the permanent tissue, it is the differentiated one. Okay, what's that? It is the differentiated one. It is differentiated, right? So when permanent, when the meristematic tissue, when it will get differentiated, so it is DM, that is the trick. So first there is differentiation, then there is maturation, right? And finally you will get the permanent tissue. So simple tissue, parenchyma, collenchyma, sclerenchyma. Complex tissue, xylem and phloem is there. Xylem and phloem is there. Am I clear? Xylem and phloem is there. Am I clear? So this is Unacademy NEET English, and see this. This is the anatomy of flowering plants, right? You guys can watch that. And this is a session which you have to like and which you have to attend, and that too tomorrow, right? Tomorrow you have to attend this session, because it will clear all your confusion. It will tell you that how to complete the syllabus in the rest 90 days, right? That too with revision, right? So hit the like button and make sure you attend this session. Done? Okay. So we are done with this meristem part. So parenchyma, collenchyma, sclerenchyma, xylem, phloem, it's all done. And now the next part is plant tissue system. The next part is plant tissue system. Is that clear? The next part is plant tissue system. Understood? Understood? Okay. The next part is plant tissue system. Understood? So let's start it, and it is even in the NCERT. Now when you talk about the plant tissue system, right, when you talk about the plant tissue systems, so they are widely spread across all the regions of plant based on type of cells. We have three types of plant tissue systems: epidermal tissue system, ground or fundamental tissue system, which is the largest tissue system here, and vascular or conducting tissue system. So how many types of tissue system do we have? We have three types of tissue system. We have the epidermal tissue system, we have the ground or the fundamental tissue system, we have the vascular or the conductive tissue system. Beta, leaving this epidermal tissue system or vascular tissue system, whatsoever is remaining, that is making the ground or the fundamental tissue system. Is that clear? Right, leaving this epidermal and the vascular tissue system, whatsoever is remaining, that will come under what? That will come under your vascular tissue system. Am I clear? So we will start with the epidermal tissue system, but before that I would like to show you a diagram of the sections. Jaise, let's say this is a diagram. Okay, I'll start with this part. I'll start with the diagram. In the case of plants, what exactly is happening? You have a plant, okay? You have to take a thin section of it, be it the stem, root or a leaf. You have to take a very, very thin section of it. Then you will follow the staining procedures, and following all that steps properly, you can see that under the microscope. So let's suppose if I'm going to take a transverse section. Now I'm not specifying, right? I'm not specifying that whether it is the tissue system or, right, whether it is the I'm not specifying it yet, right? I'm simply saying let's pick the stem, and when you take its transverse section, right? So look at this outermost part. So this outer covering wala part, what will you see? These cells here, right? You will talk about the epidermis, right? Now here inside it you will see that there will be different sort of arrangement of your vascular bundles, and then rest of the part, right, vascular bundles will include xylem and phloem. So rest of the part, what I just said, that will make the ground tissue, that will make the fundamental tissue. Is that clear? That will make the ground tissue, that will make the fundamental tissue. Is that clear? Is that clear? Sure, right? So that's what you have to keep in your mind. So when you will see these thin sections, so under the microscope you will see different types of cells and their function as well, right? So this is what firstly, right, we are going to discuss here, and when we will start the individual sections, then you will have the better clarity of it. So we will start with the epidermal tissue system. So how can you start? You can start with a simple diagram. So if it is the epidermal tissue system, right, if it is the epidermal tissue system, then don't you think, beta, then don't you think it is meant for the protection? Then don't you think it is meant for the protection? Yes or no? Yes or no? If it is the epidermal tissue system, if it is having the epidermis, what will you discuss in the epidermal tissue system? Right, you have, you'll get all the notes here, but still, epidermal tissue system, it is having the epidermis, and here you will see epidermis, you will see epidermal cell appendages, and then you are going to see the stomatal apparatus. What will you see? You will see the stomatal apparatus. Okay, so you have epidermis, you have epidermal cell appendages, yeah, epidermal appendages they are, right, epidermal appendages they are, and you will talk about the stomatal apparatus. Am I clear? You will talk about the stomatal apparatus. Yes or no? Yes or no? Yes or no? Now, when you talk about the epidermis, this is that outer covering, right? This is that outer covering of the epidermis. And students, this epidermis, right, I am not saying always, but mostly you will see it is uniserrated. What is the meaning of uniserrated? Uni mane single, serrated mane layered. It can be multi-layered also. It can be multi-layered also, but here it is what? It is uniserrated. Is that clear? So this epidermis, it is covered by cuticle. So this cuticle, right, it will prevent the water loss. It will prevent the water loss. Firstly, I am giving you the overview, right? This cuticle, it will prevent the water loss, right? And this is the epidermis, which is mostly uniserrated. So it is covered by cuticle. Cuticle will prevent the water loss. But here, remember, it is in the case of stem and the leaf that you will see. In the case of root, right? In the case of root, what will you see? In the case of root, you will see this thing that here the epidermis is not covered by the cuticle. Why? See, in the roots, epidermis is not covered by cuticle. So it is called, epiblemma. What it is? It is called epiblemma. Not just epiblemma, you can also call it as pyliferous layer. You will also call it as pyliferous layer. Are you getting my point? Are you getting my point? Why pyliferous layer? Because here you will see that these epidermal cells, they will emerge what? They will emerge, right, these root hairs. Is that clear? So root epidermis is not covered by cuticle, so it is called epiblemma. Am I clear? So it is the epidermis hairly, mostly uniserrated, but it can be multiserrated. Now, in the case of, now what are these epidermal appendages? Here you will talk about two things, right? You will talk about root here, you will talk about stem here, and that stem here are nothing. That stem here are trichomes. That stem here are nothing. That stem here are trichomes. Understood? Trichome. So जैसे if it is the epidermis, from this stem epidermis, this will be your trichome, right? And from the root, it will be your root here. right? And when I talk about the stomatal apparatus, here you know that we'll talk about it in the case of leaf and all. So we have to discuss it. So outer covering meant for protection. Let's discuss the points here. Any doubts? Any doubts? Any doubts? Done, beta? Done, beta? Sure? Done? Mm. So epidermis, it is the outermost layer of the primary plant body, right? Made up of elongated, right? Look at the cell shape there. It is elongated, and it is elongated. And the way I was making the diagram here, na, so you can name all, you can write all the important points here itself, right? You can make the short note. So mostly uniserrated. Here you will see, you can write it like this: ki elongated, compactly arranged cells are there. Are you getting my point? Elongated, compactly arranged cells are there, like this. Like this you people can continue. Is that clear? So here you can see it is made up of elongated, compactly arranged cells which will form a continuous layer outside the epidermis. So what I just said that it is covered with a thin cuticle. Done, beta? Done, beta? Right? So cuticle will prevent the loss of the water. So cuticle is absent in roots. So that is why the epidermis there is epiblemma or the pyliferous layer. So see, in the diagram format, it is so easy, so clear. Right? Done, beta? Okay. Okay, so next is the epidermis is usually single layered. I told you uniserrated, but it can be multi-layered. So where will you see it multi-layered? In the case of some leaves, beta, right? And aerial roots of orchids. So remember, today in the morphology, we discussed about the orchids. So in that orchids, right? In that orchids, we just talked about the velamen. So here you can see in the roots of the orchid, right? In the roots of the orchid, we know that epidermis is having, here it is multi-serrated, right? Multi-serrated epiblemma. What is there? Multi-serrated epiblemma. And here you will see that in the roots there, you will have the velamen. So what is that velamen? Velamen is the dead spongy tissue. It is the dead spongy tissue. Am I clear? What's that? It is the dead spongy tissue, which is what? Which is hygroscopic in nature, right? Which is hygroscopic in nature. Am I clear? Done. So epidermis, normally it is uniserrated. Normally it is uniserrated, but it can be multiserrated, right? So I have given you the example here. So epidermal cells, parenchymatous, right? With a small amount of cytoplasm, and here they are compactly arranged. So up to this part all clear. Up to this part all clear? So next is the epidermal appendages. So now when we talk about the epidermal appendages, we'll show you something here. One second. Okay, so epidermal appendages, see, in the case of shoot, in the case of stem, you will see, in the case of shoot, in the case of stem, you will see that these epidermal appendages, we call them as trichomes. What do we call them? We call them as trichomes. So these trichomes, what are they? They are, right, they are mostly, usually multicellular. Usually what is it? Usually it is multicellular, right? It is soft or stiff, branched or unbranched, branched or unbranched, glandular or non-glandular. So what is the job of these trichomes? So trichomes are the epidermal appendages of the shoot system. So they will prevent water loss due to transpiration, right? They just prevent the water loss due to transpiration. Prevent water loss due to transpiration. Am I clear? So see this, trichomes, cells of the dicot stem, they will produce the hair called trichomes in the shoot system, and they are usually multicellular. So in your short notes, please include this question, that trichomes are what? Trichomes are what? Trichomes are multicellular. Is that clear? Trichomes are multicellular. Done, beta? Okay, so as I said, they may be branched or unbranched, they may be soft or stiff, they can be secretory, right? It's not mandatory, they can be glandular, right? And cannot be, so they help in preventing water loss due to the transpiration. So this is again a previous year question. Am I clear? This is again a previous year question. Am I clear? So root hairs, they are what? They are beta unbranched, right? They are unbranched. Done, beta? I need water. No, just a minute. Okay, so root hairs, they are formed due to the elongation of epidermal hair, and I told you root hair will be what? Root hair will be unicellular. So these are the differences which we have to remember, beta. So trichomes multicellular, root hair unicellular. They will have thin wall, which is made up of cellulose and the pectic material. Root hair will absorb the water. So we talked about the epidermis. Epidermis is the outer covering, so it is meant for protection. In the case of shoot system, even in the case of leaves, the epidermis is covered with the cuticle. It is covered with what? Cuticle, right? So that it can prevent the water loss, because cuticle is thick, right? Thick covering which will prevent the water loss. Root hairs there is no cuticle. That's why in the root hairs, epidermis is not covered by cuticle. That's why we use the word epiblema or the pyliferous layer. In the case of shoot system, when you will see the epidermis, from the epidermal cell, that appendages will come out, right? That epidermal hairs will come out, right? In both the cases, you can call them as epidermal hair. But the point is, in the shoot system, you will use the term trichomes. What is the term that you people are going to use? You are going to use the term trichomes. So they can be branched, unbranched, usually multicellular, glandular, may not be glandular, right? So they'll prevent the water loss due to transpiration. Now come to the another point. What is that another point? That is the root hair. So root hair, they will be in the root. They will increase the area for the absorption as well. They do the water absorption and increase the surface area as well, and they are unicellular. So these are the direct points. Is that clear? These are the direct point which you have to remember here. Now we will move to the next part, that is the stomatal apparatus. Now in the case of stomatal apparatus, what you have to remember, just say I'll start it in this way here. So this is what we have. So in the case of stomatal apparatus, what do you see? You will see stomata. Stoma is singular, that is the opening. Plus you will see the guard cells here that are covering them. Plus you will see the surrounding accessory cells, which are also known as subsidiary cells. Am I clear? Sure? Sure. Any doubts? Done, beta? So in the case of dicots, because we have two classes, firstly what you have to see, this opening here, this is a stomata. Singular is stoma actually, so this is stomata. This is that opening. We know that, right, through which exchange of gases occur. And water loss in the form of vapor, that is transpiration, occur in the form of vapors, right, that is transpiration occurs. So this is that stomata, that opening. Okay, so we know that it will be there on the leaf epidermis. Now they are guarded by, they are having in their surrounding the guard cells. What exactly are these guard cells? They are also the modified epidermal cells only. So these guard cells, right, guard cells, in the case of these guard cells, what you have to remember, that in dicots, right beta, in dicots they are bean or the kidney shaped. That's a direct previous year question. It's the bean or the kidney shaped, right? It is the bean or the kidney shaped. But in the case of monocots, what will you see in the case of monocots? In the case of monocots, you will see that they are what? They are dumbbell shaped. I'm writing it here. Okay, so in monocots, what are they? They are dumbbell shaped. That is a direct previous year question. So in monocots, the guard cell dumbbell shaped guard cells are present, right? Dumbbell shaped guard cells are present. Is that clear? Is that clear, beta? So stomatal opening is there, through which the exchange of gases occur, through which the water loss due to transpiration occur, number one, right? Then it is the guard cell. So here in the guard cell, if you will see, right, in the guard cell, its inner membrane, inner wall is actually thick, but its outer wall is thin. So inner wall is thick. So inner wall, this inner wall near stomata. It is near stomata, it is towards stomata. It is beta thick. And the outer wall, what is it? It is thin. This part clear? Now in this guard cell itself, you will see these radially oriented, parallelly arranged. Firstly, understand the structure, then we will discuss everything, every function here. So these are radially oriented, parallelly arranged cellulose microfibrils. Done? Okay, so guard cells mein you will also see chloroplast is also present. Chloroplast present. Am I clear? Am I clear? So this is the complete stomatal apparatus. We have the opening. This is the stroma, stomata, through which the gaseous exchange and the transpiration will occur. So in its surrounding, you have the guard cells. Right here, radially oriented, parallelly arranged cellulose microfibrils are there. Inner wall is thick, outer wall is thin. Inner wall towards the opening is thick. Outer wall away from the opening is thin. Now in this surrounding, these are also what? These are the subsidiary cells or the accessory cells. They are also your modified, right? They are also your modified epidermal cells. Now what's going to happen? Because here you see the chloroplast, so photosynthesis will occur, the starch formation will occur. So here what is going to happen? Just say normally if you will see the guard cells, right? Normally if you will see the guard cells, just say let's suppose this is going to be closed, है ना? This opening is what? This opening is closed. Right, this opening is closed. Now in these guard cells, when there is an increase in the solute concentration, now right now in this guard cells, when there is the increase in the guard cells, when there is an increase in that solute concentration, then the water from the surrounding will enter here. है ना, the turgor pressure will increase. So the change in the turgor pressure, right? So the change in the turgor pressure of guard cell help in opening and closing of stomata. How will it help? It will help in opening and closing of stomata. Am I clear? So the change in the turgor pressure of guard cell will help in the opening as well as the closing of the stomata. So basically even the H positive and K positive ion movement is also there. So it is all related to the turgor pressure here. So basically where do we see the stomatal apparatus? We know that it is on the leaf epidermis, and in the case of plants which are growing in the dry condition, then the sunken stomata is there. It will not be at the epidermis, it will be little down so that water can be conserved. So this is about the stomatal apparatus. And let me tell you, monocots are better than the dicots. So monocots में these dumbbell shaped guard cells, they are better. Why? Because even in the less solute concentration, they will facilitate the opening. So here let's revise it. So what is stomata? Stomata are the structures present in the epidermis of leaf. Right? What is stomata? Stomata is the structure which is present in the epidermis of leaf. Yes, please. What is stomata? It is the structure present in the epidermis of leaves. So each stoma, right? Each stoma, what does it contains? Stoma mane this opening. Two bean-shaped cell, which are guard cell, right? That will enclose the stomatal pore. This part clear? Then the outer wall of the guard cell, the one which is away from the stomatal pore, the one which is away from the stomatal pore, it is thin. And the one which is, right? And the one which is towards the stomatal pore, it is highly thick. So guard cells, as I told you already, they are having the chloroplast. So imagine all that points, right? We have basically covered here in the form of diagram. So that is how you have to make the short notes. Am I clear? Now, sometimes a few epidermal cells, right? They are in the vicinity of the guard cells. They will become specialized in shape and size. So these are the subsidiary cells. So they are also known as accessory cells. Is that clear? They are also known as accessory cells. Got it? So few epidermal cells in the vicinity, right, in the proximity of the guard cell, they'll become specialized in their size and shape. So they are the subsidiary cell. Is that clear? So all that thing, as I said, stomatal aperture, guard cells and the surrounding subsidiary cells, they are together making the stomatal apparatus. Am I clear? They are together making the stomatal apparatus. So grasses means your monocot. So guard cells, they are dumbbell shaped, again a PYQ. So they are better. Why? Because they require very less solute concentration and less water, and they can open this stomata. Okay, beta? Right? So any doubts? Any doubts that radially oriented, parallelly arranged line is also important? Noted? Quick. Noted? Sure? Sure? Done. So this is the diagram. You can see this very clearly. Okay. So you have the epidermal cells, you have the subsidiary cells, you have the chloroplast, guard cells, and the stomatal pore is there. So inner lining is what? Inner lining is thick here, right? And this is also what? This is also thick. Am I clear? Done, beta? So next is the ground tissue. So told you already about it. We will discuss in the individual sections as well. So ground tissue system matlab from all the tissue, if you will minus epidermis plus vascular bundle. So whatsoever is remaining, that is the ground tissue. Is that clear? So it is the largest tissue system, students. It is the largest tissue system. So all the tissues except epidermis, vascular bundle, they constitute the ground tissue. All the tissues except epidermis and vascular bundle, they constitute the ground tissue. So it is basically having simple permanent tissue. It will contain parenchyma, collenchyma, sclerenchyma. So in individual sections, we will see where will parenchyma to definitely is there in all the cells. Where do you see the hypodermis made up of collenchyma or the sclerenchyma? Okay, so hypodermis, general cortex, endodermis, pericycle, medullary rays, pith, they are making the ground tissue, but it is not mandatory that it will always be differentiated like this. Okay, so one more thing that you have to keep in your mind, that in leaves, the ground tissue consists of thin-walled chloroplast containing cells, and it is known as mesophyll. What is it, beta? It is known as mesophyll. Understood? It is known as mesophyll. Understood? Now, very important topic, that is of vascular bundles. Right? It's a very, very, very important topic. If you are clear with the vascular bundles, then everything will be crystal clear for all of you. Trust me on that. Right? So it is the vascular bundles that you have to understand sincerely. Okay? Done? So should we continue? So vascular bundles, what do we have in the vascular bundles? In the vascular bundles, we know that xylem and phloem, they are present. They'll help in translocation, right? Translocation of the food, water and minerals. So water and minerals. So xylem, we know that it will carry what? It will carry water and minerals. I hope it's clear to everybody. And it shows the unidirectional transport. What will it show? It will show the unidirectional transport. Yes or no? And on the other hand, your phloem. So xylem is water and minerals. So it is unidirectional. So phloem will show. Phloem will do what? Food. So here as per NCERT, if I have to go, so there will be bidirectional, right? There will be bidirectional movement. Is that clear? Now, what is the next part that you have to remember here? Dekho. Though there are many other types of vascular bundles also, what you have to understand, even in the recorded part, I have explained it. There are two types of vascular bundles which we have to discuss. Radial vascular bundle, conjoint vascular bundle. Firstly, understand this terminology, right? And this is a very, very important part, beta. So please pay attention. So there is the radial vascular bundle, and what else do we have here? There is the conjoint vascular bundle. There is the radial vascular bundle, and what else do we have here? We have the conjoint vascular bundle. Radial vascular bundle, here you have to remember that xylem and phloem, beta, I'm just using X and P thing here, right? It is xylem and phloem. I'm using this X and P to denote xylem and phloem here. So xylem and phloem are present at different radii. Xylem and phloem are present at different radii in a vascular bundle. Now, what does it mean? Just say, for an example, let's say, so let's say this is the vascular bundle that you have. So this part is showing, this red colored part, xylem. This is showing phloem. Right now I'm not discussing anything other than that. So they are at different radius, right? Just let's suppose if here I'm making it, so I am showing you this is xylem, 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 xylem with this red color, and with this green color here I'm showing you phloem. So they are present at different radius, right? So this is the radial vascular bundle. What sort of vascular bundle it is? It is the radial vascular bundle. So R for radial, R for roots. So where do you see it? It is in the roots. Where are you going to see it? It is in the roots. And xylem arrangement, what is it? It is exact, right? It is exact. Now, though I have already explained it in last part, but still, I'm again explaining you this thing. What is a xylem? What is this exact and endark? Listen to me very carefully. Xylem mein beta, you know that when there is the primary growth of the plant, like initial growth, so there is the primary xylem will form, hai na? Initial growth is there when the length is increasing. We are not talking about the girth. So length is increasing, primary growth is there. So you talk about what? You talk about the protoxylem, and you even talk about the metaxylem. You talk about the protoxylem and you talk about the metaxylem. So protoxylem that will form first, right? It is the primary xylem that form first. And this one form later, right after this, hai na? Now in the case of xylem, right, so when do you say, just say let's suppose if your protoxylem, beta protoxylem will form first, right, comparatively smaller in size it is, then you have the metaxylem. Now in the case of protoxylem, if your protoxylem is at periphery, you remember it, you can check the recorded part also. If protoxylem is at periphery and metaxylem is there in the center, right, then what will you see? Protoxylem is at periphery, metaxylem is there in the center, right? When this is the arrangement, this type of arrangement is known as exact, right? So the newly formed, the xylem will start its formation because protoxylem is at periphery, means xylem formation has started from the periphery towards the center. So this is basically the centripetal arrangement of xylem. What sort of arrangement of xylem it is? It is the centripetal arrangement of xylem. It is the centripetal arrangement of xylem. Is that clear? Is that clear? Yes, please. Is that clear? Sure? 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 So, this is exact. Now, if the protoxylem is at the center, if protoxylem is at center, metaxylem is at periphery. This is called as endark arrangement. What sort of arrangement it is? It is the endark arrangement. What sort of arrangement it is? It is the endark arrangement. And right here you can see the xylem formation. Can I not say that? Like protoxylem is in the center. So from center it is moving towards the periphery. So here it is the centrifugal arrangement. Got that? So one thing that you have to remember, beta, roots will show you the exact arrangement, and dark arrangement will be there in the stem and the leaf, and the exact arrangement will always be there in the roots. That is what you have to remember. Firstly, tell me this part clear? Yes, please. This part is clear. Sure. Any doubts here? Any doubts here? Any doubts? Sure, beta. So that is what you have, right? So now when I was talking about this vascular bundle, I told you radial. So from R, I have to remember roots, and I have to remember that here the xylem arrangement is exact. Understood? Here the xylem arrangement is exact. Understood? So radial or this conjoint, obviously it is xylem and phloem are at same radius. They are not at different radius. Okay. So now we have to talk about this types of conjoint vascular bundle. Dekho beta, conjoint means xylem and phloem, they are at same radius. So now we will talk about conjoint collateral vascular bundle. Conjoint collateral vascular bundle. So here, first of all, when I'm saying that the xylem and phloem, they are at same radius, this type of vascular bundle, right? When, what am I saying? I'm saying that xylem and phloem, they are at same radius. Xylem and phloem, they are at same radius. Are you getting my point? Xylem and phloem are at same radius. This is conjoint. Conjoint collateral means xylem is towards inside and phloem is towards outer side. This is the another word that you have to understand here. Xylem is towards inside and phloem is towards outside. This is the meaning of conjoint collateral. Now after that, here you will discuss two more things. Here you will discuss two more things. What is it? So you will see open conjoint collateral, open vascular bundle and closed. Now what does it means? Just see this. If this is the vascular bundle, right, conjoint collateral vascular bundle. So let's suppose, students, here you have the xylem part and here you have the phloem part. See this. So I'm not making the ice cream cones here, but actually what is the story? Dekho, this is your xylem, this is your phloem, this is your xylem, this is your phloem, and it is known as cambium. What's that? Cambium. And to be more precise, intra mane within. So it is within the vascular bundle. So I will call it as intravesicular cambium. Intravesicular cambium it is. So when I'm saying that it is the conjoint and collateral vascular bundle, xylem is towards inside, phloem is towards outside, and they are basically sharing the same radius, right? They are basically having what? They are basically having the same radius. Now when you look at this part here, in the open vascular bundle in between xylem and phloem, cambium is there. So the presence of cambium means the secondary growth occurs. Right? Means, how do I remember it? I remember it like this. Open conjoint vascular bundle. Open means open for secondary growth. Means if the vascular bundle is open, the secondary growth will occur. That is, the girth formation will occur because this cambium, right? This cambium will help in the secondary growth. Now it is the closed, right? It is the closed conjoint collateral vascular bundle. So this will not help in secondary growth. So there is no secondary growth. So this is something that you will see in the case of monocot stem, right? Because monocots, that is your grasses, you don't see in the case of monocots that, you know, thick stem. But here, open it is. So here you will see what? Here you will see this will be there in the dicot stem. So xylem, phloem, it is open for secondary growth, and this one, this is closed, there will be no secondary growth. Is that clear? So vascular bundles are very important. I will also tell you what is bicollateral vascular bundle, which is a specific feature of Cucurbitaceae family. So that's what you need to know. Now, why again and again I am using red color for this xylem and green, this phloem for phloem, green color. So in double staining with fast green and with saffronin and fast green, so during double staining with saffronin and fast green, xylem become red, phloem become green. So that's why I'm using these colors to denote that. Is that clear? Right? So during double staining with saffronin and fast green, that's what you have to see. Okay? Okay, please write down. Please write down. So xylem will pick up your saffronin. So red, fast green will be picked up by your phloem. ठीक है? Okay. That's why I use this color. Okay. And saffronin, it stains the lignified cell walls. This one will stain that cellulosic, hemicellulosic cell wall. So this is important. If you think this is extra, so actually it is not. Wait, I'll show you. NCERT exemplar. Directly go to the Google, type NCERT exemplar, anatomy of flowering plants. So, ye dekho. Open the exemplars. So this is my advice. Beta, you should solve it. And today at the end of the class, we will also solve it. Okay. So ye dekho, transverse section of stem is stained first with saffronin and then with fast green. That's why I told you double staining. Following the usual schedule of double staining for preparation of permanent slide, what will be the color of the xylem and the phloem? So xylem and phloem is written, so red and green it will be. So baki dekho, next question. See, meristem, parenchyma, collenchyma, sclerenchyma, epidermal tissue. So match them, right? Then cuticle, bulliform cells, stomata, epidermis. We'll discuss it today, right? So, and after that beta, after that identify the simple tissue from the following. We know that it is your parenchyma. So do solve the exemplar. You just write the chapter name, NCERT exemplar. This will open and you should definitely solve it. At least try to solve the genetic question, physiology question. You'll be amazed to know that there will be some things which you will not be able to figure out, even the ecology questions. Okay, okay, done. Thank you, Beta Dipti. So this is about the vascular bundles, a very important topic. Okay, so let's revise it from the notes and then we will discuss the amphivasal, amphicribal and bicollateral vascular bundles also. So radial vascular bundle, right? Let me highlight it. That will be better. So radial vascular bundle, when xylem and phloem are present separately on different radii in alternate manner, in alternate patches, xylem, phloem, xylem, phloem, xylem, phloem, xylem, phloem. So this is the radial vascular bundle. Now, these vascular bundles, xylem is exarch. Now, do you know the meaning of exarch? Protoxylem is at periphery, metaxylem is at center. So, protoxylem form first, so it is the eldest. Metaxylem form later, it is the youngest. So, periphery to center. So, this is centripetal arrangement. Now, order of development, obviously told you, of xylem is, it is centripetal. And then the next is, so most of the roots, your dicot, your monocot, even gymnosperms or fern roots, they will show this. Okay. Now the next is, there is one exception also, that in radish, right, even in the carrot, turnip and sugar beet, they all are root modifications, right, conjoined. They are root modifications, so they have conjoined collateral vascular bundle. So this is just an exception here. Now, let's move to the next part. So next part is conjoint vascular bundle. So here in this vascular bundle, right, xylem and phloem are present on the same radius. Right, xylem and phloem are present on the same radius and combine into a bundle. Okay, okay. Then comes collateral bundles are the type of conjoint bundles in which phloem and xylem, it lie together on the same radius with phloem on the outer side of xylem, phloem on outer side and xylem on inner side. Right, so this one you can see cambium is there, so this is open, so it is closed because there is no cambium. So no secondary growth, dicot stem, monocot stem. Understood? So in dicot stem, vascular bundle is conjoint, collateral, xylem is endarch, centrifugal. Done, students? And open may, told you already, but still I am highlighting it here, that if the cambium is present between the xylem and phloem, then it is the open vascular bundle. So stem of dicots and gymnosperms, so closed may. So when cambium is absent between xylem and phloem of conjoint vascular bundle, it is the closed vascular bundle. So monocot stem and the leaves of angiosperms. Understood? And leaves of angiosperms. So look at the diagram. Now I'll tell you what is the bicollateral vascular bundle. Dekho, if you have xylem here, right? This is done. Chalo, I'll write it here. If you have xylem here and phloem on both sides, xylem here, phloem on both sides, and both the sides will have cambium in between. This This type of vascular bundle. Xylem in the center, phloem in the surrounding. Outer phloem, inner phloem. Inner cambium, outer cambium. In between there is a xylem. So this is the bicollateral vascular bundle. What sort of vascular bundle it is? It is the bicollateral vascular bundle, and it is the feature of Cucurbitaceae family. It's a unique feature of Cucurbitaceae family. What's that? It's a unique feature of Cucurbitaceae family. Am I clear? Am I clear? So then it is the concentric vascular bundle. So phloem inside, xylem outside, amphivasal. Xylem inside, phloem outside, amphicribral. Okay, but I will suggest you to remember this one. And here, it is not like that ki both the cambiums are functional. Not at all. Not at all. Not at all. So here you will see only outer cambium is functional. Only outer cambium is functional. It's not like that both the cambium is functional and it will help in the secondary growth. No, it is only the outer cambium that is functional. Am I clear? Sure? Sure. So xylem and the other sides are having phloem and phloem. So let's revise it. So conjoint bicollateral open vascular bundle, you know that. So it is bicollateral. So two patches of phloem, one on each side of xylem are found. So here you will see, here you will see there are two strips of cambium, outer and inner. So only outer cambium is functional. So order of development is centrifugal, means endark condition is found here. So it is in the case of Cucurbitaceae family and even in the some plants of family Solanaceae. Am I clear? Am I clear? Yes, please. Am I clear? Yes, please. So see this. So here you will see radial vascular bundle, right? Open, conjoint, collateral. Sorry, this upper one is closed, conjoint, collateral. This lower one is open, conjoint, collateral vascular bundle. Am I clear? Sure? Sure? Chaliye, so after this we have to start the root and the shoot system, and trust me, we'll start with the root system and you are going to find it amazing. Okay? Right? Now you are not going to find the sections difficult. So any doubt? Any further doubt? Any further doubt? Yes, please. So let's start the dicot root and the monocot root. So I'll give you a trick here, right? A very simple trick here. Dekho. Now we have to discuss the sections, right? So all the points will be mentioned there. So if you will see the dicot root, right? Dekho, dicot root and monocot root, what we know already, right? About the roots. If this root system is clear, then this part should be very clear to you. So dicot root or monocot root, that's the next part that we have to understand. Okay, so if it is a root, I am sure that you all know one thing, that if it is a root, the vascular bundle should be radial, the xylem arrangement should be exarch. Here also vascular bundle should be radial, xylem arrangement should be exarch. These 2 points are clear. These 2 points are clear, then what else do we need to see? And trust me, if these points are clear to you, you'll get to know many things there, right? And here we know that epidermis is not epidermis, it is epiblema. Now, how to differentiate that? I'll show you the structure, right? There are other things also. We will also see that how the ground tissue is differentiated or whether it is differentiated or not. But still, this is the dicot root. This is the diagram given in the NCERT. I know it very well and I know you know it. No doubt here it is epidermis is written, but we know that we will use the word epiblemma or pyliferous layer. So even in your NCERT, please make the correction. Done. So epiblemma plus pyliferous layer, because there is no cuticle. Root here, you know it, it is unicellular. Am I clear? Now, when you talk about the ground tissue, in the case of roots, you are not going to see that hypodermis, general cortex and all. Here you will see that this part is the cortex, right? Firstly, there is a sequence of layer that you have to understand. Just say, let's suppose this is epidermis. Okay? In general, I'm telling you, this is hypodermis, general cortex, endodermis, pericycle, xylem, phloem, and pith. And pith. I'll tell you what you have to see here. देखो, जैसे this is the general arrangement. With the help of this diagram, I'll explain it. जैसे मान लो epidermis. You know that it is making epidermal tissue system. If there is something below epidermis, what will we call it? We will call it as hypodermis. Below hypodermis, then you will say this is cortex. Or you can simply call this whole part is cortex, general cortex or cortex. Then this is the endodermis. What is the name of this layer? Endodermis. Then inside it, you will see pericycle, which can be uniserrated or multiserrated. Then we know that vascular bundles will be there. Vascular bundles will be there having different arrangement. And the central portion is the pith. First of all, remember this sequence. And here in this sequence, keep one thing in your mind, that innermost layer of cortex is endodermis. Innermost layer of cortex is endodermis. This is what you have to write. Do it quickly. Innermost layer of cortex. So just focus on these layers first. Then I'll tell you what is there in the roots, what is there in the shoots. But firstly, focus on this part. Is it done, beta? Done. So epidermis, below epidermis, hypodermis, general cortex, endodermis, pericycle, vascular bundles and pith. Now in roots, right? In roots, we will see the cortex part, the endodermis part, and obviously the pericycle part. Here you are not, and obviously vascular bundle and the pith. We know that except this epidermis and vascular bundle, rest everything will make the ground tissue. That is why it is the largest tissue. So this is something, if you see proper hypodermis and all, you are not going to find it in the roots. And this endodermis, it is not something separate. It is actually the innermost layer of the cortex. A very important PYQ, right? Now, I was telling you that if you look at this diagram, this is the anatomy of the dicot root. This is the anatomy of the monocot root. I just forgot to, place them together for better comparison. So now you see again, this is the anatomy of the dicot root. Observe the structure well, right? Everything, please pay attention. Quick. Observe this diagram. Now this is the another diagram that you have. This is the monocot root. This is the monocot root. Okay, that was dicot, monocot. Now if somebody will ask you to notice the differences, right? If you are smart enough, you will see that in the dicot, right? Ultimately, because they are root, so the vascular bundle is going to be what? Radial type. So in dicot root, if you have seen, xylem was less. In monocot root, not just the xylem, I'll talk about xylem and phloem both, xylem are more. So this is the very important difference that you have to see here. That is a very important difference that you have to see here. Understood? Understood? So if you will understand one section, the other section will be easy for you. Okay? Now, this is one thing. Now, second thing is, just a root. We know that if this is the root epidermis, you know that it is going to be your root here, right? It is going to be your root here. So you know that the root here will absorb water. What that root here will do? It will absorb water. And here we will see that here we will have the cortex cell, the parenchymatous cells having intercellular spaces, right? And finally, the innermost layer, here it is what? It is the endodermis. Now I will explain you some things here. So root here will absorb the water, that water will pass here. Now this water, this will also pass from the cell, it will also pass from these empty spaces and all. So when you talk about this water transport here, this water movement or water transport here, when you will discuss this part, people, you will see, right, what you guys are going to see here, you people will see here that there is, one is apoplastic pathway and one is symplastic. One is symplastic. I'll tell you why am I telling you. Now imagine when root here, they are absorbing this. You know that in a mature plant cell, even the cell wall, just say, let's say, you know that if it is immature, then there will be primary cell wall and inside there is the formation of the secondary cell wall. But when the plant cell, it is differentiated, it is mature, we know that it will have secondary cell wall that will be in the layers. And in a mature plant cell, we say the cell wall is believed to be dead. You remember? Dead. Like it is also kind of dead. Empty spaces are there. So when water will be absorbed, so when the water, when it passes from the empty spaces, when it passes from the empty spaces, right? Obviously, if it will pass from the empty spaces, it will move quickly. What, like, let's suppose if you have to run, if I will give you an empty ground, it will be easy. If that ground will be filled, then obviously there will be a problem. So in apoplastic pathway, that water which was absorbed, right? The water which is absorbed, what will happen to that water? That water will pass from the empty spaces, from the intercellular spaces, from these dead cell walls. Now, when it is the symplastic pathway, then what will happen? Just say plant cells, you know that they have the continuous cytoplasmic connection. Plant cells, what do they have? They have the continuous cytoplasmic connection. They have the continuous cytoplasmic connection in the plant cell. So when you look at these plant cell, they are having continuous cytoplasmic connection. So you will see, right, when the water movement is through the cytoplasm, which will obviously take time, which will obviously comparatively slow, then it is the symplastic pathway. But ma'am, why are we discussing it? Because we have to discuss the structure, but we have to understand one more thing, that in the case of roots, epiblemma is there, root here are there, then cortex is there, intercellular spaces are there. So innermost layer of endodermis, this is having something very important, that are Casparian strips which are water impermeable. Because we have to reach this part, that's why I am teaching you this. Okay, that's why I am teaching you this. Just say, let's open this part. So anatomy of the dicot root. So very first point, we have covered it, we have discussed it, that the outermost layer is epiblemma, because it is made up of compactly arranged thin-walled flat parenchymatous cell. So here it is what? It is devoid of, like it is without cuticles and stomata. Right, root it is, right? So it doesn't have stomata, but today we discuss the photosynthetic roots, that is Trepa and Ctenospora. Right? Right? So many of the cells of epiblemma, they protrude in the form of unicellular root hairs. So these cells are also known as trichoblast. So that's not mandatory to remember trichoblast, but yeah, you should know that the cells of the epiblemma, they'll protrude out. They are forming this unicellular root here. So due to the presence of root hairs, right, due to the presence of root hair, epiblemma is also called pyliferous layer. Am I clear? So that is something that you need to highlight, and that is something that I started also, that hypodermis is absent in the roots. These are the quick revision points, QRPs. Okay, hypodermis is absent in the roots. Am I clear? Hypodermis is absent in the roots. Am I clear? Now let's talk about the cortex, ground tissue, where you will see cortex, which will have several layers, right? And what sort of layer? Thin-walled parenchyma cells. We know that parenchyma living, they are having thin wall. So here you will see they will also have the intercellular spaces. Any doubts? Any doubts? So again, a repeated question, repeated PYQ, and even I have discussed it multiple times, that the innermost layer of the cortex is called endodermis. So in the endodermis, you will see barrel-shaped cells are there, right? Barrel-shaped cells are there, and their radial and tangential walls, they will have suberin thickenings, beta, and that will make it water impermeable. Right, that will make it water impermeable, a very important point. The cell shape is also very important. So single layer of barrel shaped cells without any intercellular spaces. So endodermis, it is the innermost layer of cell, right? Endodermis, it is the innermost layer of cell. Yes or no? Endodermis, it is the innermost layer of the cortex. But in general, we have seen that cortex is not having the cortex is having parenchymatous cells and having intercellular spaces. But endodermis, which is the innermost layer of cortex, it doesn't have, right? It doesn't have intercellular spaces. Rather, it has the barrel-shaped cells. So the next point that I told you, that the tangential as well as the radial walls of these cells, they have the deposition of water impermeable waxy material, that is suberin. And this forms the Casparian strip. So where these Casparian strips are present in the endodermal cells, and they are present at the radial and the tangential wall. So endodermis is the biological checkpost. So now I believe here you will understand this part in a better way. So I was telling you about the water absorption. I told you water will be absorbed. So when it is moving through the intercellular spaces or the dead spaces or the dead cell wall, then it is apoplastic movement. The moment it will reach the endodermis, what will happen? The Casparian strips are there. They will make it water impermeable. They will not allow the water movement to go further, right? That will not allow the water to go further, right? So here, how the water will pass further, because next to it is pericycle, and then there are the vascular bundles. So then what will happen? So at this, it is the biological check post. But why is it so? Because here, then now instead of apoplastic pathway, the symplastic pathway will be followed. The water will enter in the cytoplasm here, right? Now you must be thinking, ma'am, how is it possible? I'll tell you. Just say, let's suppose, just giving you a random, just making a random diagram, not the accurate one. I hope you know the radial vascular bundle. Now I hope the radial vascular bundle thing is clear to you. So this one is what? Endodermis, right? Next to it is pericycle. I told you this endodermis is having what? This endodermis is having the Casparian strips. Now what am I trying to tell you? I'm saying that, see, pericycle, and this is the protoxylem. We know it now. This is the protoxylem. Now the cells of the endodermis, which are opposite to this protoxylem thing, here you will see that Casparian strips are absent, right? Casparian strips are absent here, and these cells are known as passage cells. Okay, just say, let's suppose this here, Casparian strips will be absent in this cell. So this will be the passage cell. So that is how, this is the biological check post. The water will not be apoplastically, but symplastically it can enter. And vascular bundles, again, xylem and phloem. Xylem is going to conduct the water, which is again having the dead spaces. So again, it will move apoplastically. So that is what I am trying to specify here. Okay, students, that is what I'm trying to specify here. So here you can see endodermis function as the biological check post. So next to endodermis lies a few layer of thick walled parenchyma cells, and it is few layered, right? Mane multi-layered, yeah, multi-serrated. So here I told you it is what? It is pericycle. What's that? It is pericycle. Is that clear? So the lateral roots, right? What? So the lateral roots, right? Lateral roots will arise from this pericycle. In the morphology part, I told you, right? So lateral roots, they arise from this pericycle. So that is why they are endogenous in origin. So initiation of lateral root, and even this pericycle, not just forming the lateral roots, because dicots, they show the secondary growth. Even in the stem, we will see the secondary growth, even in the root. So when there is the secondary growth, even this pericycle will also form the cambium. Now that secondary growth topic is deleted from our syllabus, but we used to see that this pericycle, it used to, you know, divide, it used to form the vascular cambium. Initially it is wavy, because in between vascular bundle, in between xylem and phloem, there is the conjunctive tissue as well. We'll discuss it one by one. So dekho, initiation of lateral roots and vascular cambium during secondary growth take place in these cells. So the pith here is actually what? Pith here is smaller, inconspicuous. So cells of endodermis, right? Just made it. Cells of endodermis, which are situated in front of protoxylem, they don't have these Casparian strip. So you call them as path cell or the passage cells. Important it is. Okay, important it is. So just pick up the important points. I'll show you the comparison tables as well. Okay, so it is the passage cells beta that will provide a path to absorbed water from cortex to pericycle. So intercellular spaces are absent between the cells of endodermis as well. Told you. So endodermis is the watertight jacket. Understood? So ground tissue clear. Ground tissue clear. And vascular tissue system you already know, right? You already know about the vascular tissue system, that there are here, usually you will see 2 to 4 xylem and phloem patches, right? Xylem or phloem patches. So we call it as diarch to tetrarch. Diarch to tetrarch. This is the xylem. Can be hexarch in some cases, but not more than that. Right? Right. So these are vascular, these are radial vascular bundle, and here you guys can see that here you will have the exarch. So parenchymatous cells which lie between the xylem and phloem, this is the conjunctive tissue. So when beta there is the secondary growth, the pericycle, even the conjunctive tissue will also become meristematic. Its de-differentiation will also occur, and a cambium ring will form right between xylem and phloem. So this is the another important PYQ, that all tissues on the inner side of the endodermis, such as pericycle, vascular bundle, and pith, they constitute the stele. Very important part it is. Quick. Very important part it is. Quick. Quick, beta. Done, beta? Sure. So this is a very important point. So look at this part. Yes, please. Look at this part. Yes, please. So look at this diagram. Endodermis, next to it is the pericycle. So protoxylem, the cells here of endodermis will not having the Casparian strip passes cells. So ye dekho xylem, phloem. So in between this tissue which is present, it is the conjunctive tissue. So beta, when there is the secondary growth, even your pericycle, even your conjunctive tissue. So during secondary growth, these permanent tissues, they get de-differentiated. So it is the de-differentiation that will make convert a permanent tissue into the secondary meristem. In the last lecture, right, we have completed it. So what will happen, this pericycle will also start making the cambial ring, this conjunctive tissue will also start making the cambial ring, and here you will see that a wavy cambial ring will form. It is not going to be circular initially. Later on, it will become circular. But initially, it is going to be what? It is going to be wavy. It is going to be what? Wavy vascular cambium. Okay, so that's how it works. So it will start making the xylem and the phloem, and that's how further the growth is. Now, if you literally understand the dicot root structure, then the monocot structure is going to be very easy. That's why I told you, if one structure is crystal clear to you, the another structure will automatically be very easy. So anatomy of the monocot root. So anatomy of the monocot root is similar to the dicot root. It has epidermis, it has cortex, it has endodermis, it has pericycle, vascular bundle, even the pith. Now, here you know that, right, dicot root is having few xylem bundle, but here more than 6. So it will be what? It will be polyarch, है ना? Xylem bundle, exarch again. Now here there will be no secondary growth. So pith here is large and well developed, and they do not undergo any secondary growth. So see, the diagram is also very easy. So here you can see the secondary wall as well. So I'll show you a comparison table that will make it very easy for you all. ये देखो, dicot root, monocot root. Dicot root give rise to the lateral roots, phallogen. Phallogen is basically your coccambium and a part of vascular cambium. What? Pericycle. Give rise to lateral roots only. Why? Because in the monocot roots, there is no secondary growth, so there is no requirement of the secondary meristem. So vascular tissue, it is usually limited number of xylem and phloem strips. There are more number. Here the conjunctive tissue, this is important. The conjunctive tissue, the one which is present between xylem and phloem, it is parenchymatous, and later on it will be differentiated into vascular cambium. But here you will see that conjunctive tissue will be mostly sclerenchymatous, sometimes parenchymatous, but it will not form vascular cambium because there is no secondary growth. So cambium here appear as the secondary meristem at the time of secondary growth. Altogether absent. Xylem usually tetrarch, usually polyarch. So that is it. This is all about it. So let's meet at 9:30 for next topic. So I will need your another 1 hour, or this anatomy part will also be over, but today we will also practice the question. We will practice this morphology anatomy all together, right? So your this portion will be over by today itself. Okay, so we will have that revision too. Sorry, not it's 9:16 now, so we will meet then. hope I'm audible. So now we will start the dicot and the monocot stem. So you tell me, up to this part all clear? Is it clear? Okay, so let's talk about the next part, that is dicot stem and the monocot stem. Is that clear? Right, let's talk about the next part, that is dicot stem and the monocot stem. Is that clear? Okay, so now in the case of dicot stem and the monocot stem, firstly, if root part is clear, then this is going to be very easy for you. And I believe roots, they are not difficult even. Okay, see, this is the diagram which is given in the NCERT. Can you see that? This is the diagram which is given in the NCERT. Are you getting my point? What is it? It is the diagram which is given in the NCERT. Now, please pay attention. Okay, please pay attention. So, in the case of dicot stem, this is the outline. First of all, if I have to observe the thing, I can notice that pith is quite large. In the dicot root, the pith was very small. Pith was very small, but it is quite large here. Number one. Number two, if you will see this part, what is it? What is it? Can I not say that ki vascular bundles, they are in a kind of ring? They are making a ring-like structure. And actually, this is a characteristic feature of the dicot stem. Right? This is the characteristic feature of dicot stem. Am I clear? Right? Now, what else you need to see here? What else you need to see here? Then you will see, other than this, right, we know that the vascular bundles that we have already discussed, the xylem arrangement is going to be in dark, and we know that it is going to be open conjoint collateral vascular bundle. I hope this part is also clear to all of you. Open conjoint collateral vascular bundle. I hope this part is also clear to all of you. Right? Right? So pith is large, right? You have the circular ring of the vascular bundles here. You will see the xylem arrangement is in dark. Open conjoint collateral type of vascular bundle is there. So in the dicot stem, we will see what will we see? We will see that there will be the secondary growth, right? Secondary growth will be there. The girth will increase. Now, observe it. So, epidermis, right? These are what? Epidermal hairs. We call it as trichomes, right? We call it as trichomes. Epidermal hairs are there. Trichomes are there. Then here you will see that proper, division of that cortical, ground tissue. That here you will see the collenchyma. Right? What will you see? Collenchyma. But what exactly it is? It is the hypodermis. Right? Firstly, I'll clarify everything from the diagram, then we will revise it in detail. So this is the dicot stem. When you see the outline, you will focus that the pith is quite large. Then you will see that vascular bundles are in the form of a ring. We know that if it is the shoot system, right, the epidermal hairs, these epidermal hairs, what are they? They are multicellular and we call them as trichomes. So they will prevent the water loss due to transpiration. Now here below the epidermis, what is it? It is hypodermis. So hypodermis is present here and it is collenchymatous. Below hypodermis, you will have the cortex that will made up of parenchyma. So here also endodermis is there, and this endodermis is known as starch sheath. Here endodermis, here there will be no Casparian strip. In this case of stem, you are not going to see the Casparian strips. So in the roots, we talked about the Casparian strips, but here you will not see the Casparian strips. Understood? You are not going to see the Casparian strips here. You will see that endodermis is without Casparian strips, and here it is also known as the starch sheath because the cells will have the starch grain. So below it, pericycle will be there. This pericycle, it can be continuous, the homogeneous layer, or it can be in the patches also. Below it, phloem, cambium. Then we know that xylem, so metaxylem, protoxylem, and this is the pith part. So tell me the diagram clear? Tell me the diagram clear? So pericycle, in some cases you can see it is in the form of patches. It is in the form of patches, or it can be the homogeneous layer. It is in the form of patches, or it can be the homogeneous layer. We will talk about it. Firstly, tell me this part is clear. Sure. So before discussing other things here in detail, ye dekho. Now, in the form of ring, that's what I said now. So I'll explain this part and then we will revise it in the form from the notes. So this is how the vascular bundles are in the form of rings. Now, in between these vascular bundles, let's say this is the vascular bundle. So in between these vascular bundles, this is the cambium. Okay, obviously it is helping in secondary growth. So it is intra mane within, intrafascicular cambium. It is what? It is the intrafascicular cambium. Guys, what is it? It is the intrafascicular cambium. Please repeat again. Intrafascicular cambium. Now, this intrafascicular cambium, it forms during the primary growth of the plant. It forms during the primary growth of the plant. So it is also referred as primary lateral meristem. It is not the result of, it is literally not the result of the secondary growth or the dedifferentiation. It is formed during the early stages. So this is the lateral meristem, but it is primary in origin. Okay. Now, in between these two, you have what? You have the conjunctive tissue that we are aware of, है ना? We know about it. Now, what's going to happen? So, during the secondary growth, जैसे conjunctive tissue in the case of roots we marked, right here in the stem, in between this, you will call it as the medullary rays. They are having the parenchymatous cells only, but these are the medullary rays. Now, I told you, let's suppose this is endodermis, and this endodermis is not having the Casparian strip, rather it is having the starch grains. So it is the starch sheath. Told you already. Now this pericycle, it is multi-layered. In some cases, you will see the pericycle, beta, it is a continuous layer, like homogeneously, it is a complete layer. But sometimes what happens, it is in the form of semi-lunar patches. It is in the form of what? It is in the form of semi-lunar patches. Like this. Like it is in the patches. Let's suppose this is the pericycle. This patch, है ना, it is semi-lunar patch. It is semi-lunar patch, and it will be beta sclerenchymatous. Sclerenchymatous. So whenever in the NCERT you will see the cells like this, right, double boundary. So you are talking about the sclerenchyma. So yes, in the pericycle, you will see that it will be either it is a complete homogeneous layer, like having the same composition. Sometimes it is in the form of patches. So when it is in the form of patches, you will see that here you will see the semi-lunar patch, right, in between the parenchymatous tissue will be there. This is sclerenchymatous. It is like that. So this pericycle here, which is just above this phloem, you can also call it as bundle cap. Right, you can also call it as bundle cap. Okay, now when there is the secondary growth, this tissue here, it will also become meristematic, and between these vascular bundles also, you will see that this is the cambium formation will occur. So this is because it is in between two vascular bundles, so it is interfascicular cambium. What do you call it? You call it as interfascicular cambium. Am I clear? What do you call it? You call it as interfascicular cambium. Am I clear? Am I clear? So it is the interfascicular cambium that we have here. Pith is quite large, but right now this is what I'm showing you. So now, if you will observe it carefully, so this is a complete cambial ring. Right, this is the complete cambial ring, if you will observe it carefully. So, but here in this cambial ring, what you have to observe that if you look at this part which is present in between two vascular bundles, right, if you look at this part which is present in between two vascular bundle, so this part is secondary in origin. It is the result of dedifferentiation here, but this is primary in origin. So ultimately during secondary growth, this will act like a complete ring. Are you getting my point? It will act like a complete ring. This will act like a complete ring, complete cambial ring, right? It is going to act like a complete cambial ring and it will do the secondary growth. Inside it will form xylem, secondary xylem, outside it will form secondary phloem. First of all, the arrangement clear? The arrangement clear? Sure, beta? Okay. See this. So epidermis below that hypodermis in the dicot stem, that is collenchymatous. This is cortex having parenchyma. In some plants you will also see the raisin ducts. So this is endodermis, this is pericycle. So if in this diagram you will observe carefully, right? So you will see that pericycle, it forms the semilunar patches. That pericycle, it forms the semilunar patches. And here this is made up of sclerenchymatous tissue. Here it is made up of what? Here it is made up of sclerenchymatous tissue. Here it is made up of what? It is made up of sclerenchymatous tissue. Am I clear? Am I clear? Sure, it is made up of sclerenchymatous tissue. Then phloem, then cambium, then metaxylem and the metaxylem and the protoxylem is there. So let's discuss it one by one. So ready? Yes, please. Ready? Sure. So just look at it very carefully. Look at it very carefully. So epidermis is the outermost protective layer of the stem, right? It is the epidermis is the outermost protective layer of the stem. It is the outermost protective layer of the stem, number one, having compactly arranged parenchymatous cells, very tightly arranged parenchymatous cells, and these cells are devoid of chloroplast. Obviously in the epidermis here you will not see the chloroplast, rather here you will see that they are covered with the cuticle, and that cuticle will have that cuticle will have the trichomes. So told you already, these trichomes, what are they? They are usually multicellular, usually multicellular they are. What are they? Usually multicellular, soft or stiff, glandular or non-glandular, branched or unbranched. Okay. Now, ground tissue is having ground tissue system. So it is having the outer hypodermis. That outer hypodermis is made up of collenchymatous cells. Do we need to remember it? Obviously, we have to. We have to remember it, okay? We have to remember it. So it is just below the epidermis. So it will provide mechanical strength to the young stem. Understood? It will provide mechanical strength to the young stem. Now below it you have the cortical layers, right? So it is having rounded, thick-walled parenchymatous cells with conspicuous intercellular spaces. Is that clear? So cortical layer, which is below the hypodermis, it will have the parenchymatous cell and they will have the intercellular spaces. Now the innermost layer of the cortex. Innermost layer of the cortex. Now what? Now this. See this. This is the innermost layer of the cortex. So innermost layer of the cortex is called endodermis. Innermost layer of the cortex is called endodermis. So cells of the endodermis here, they are rich in starch grains, and the layer is also referred to as starch sheath. Understood? And the layer is also referred to as starch sheath. Understood? Understood? Pakka? Done? So, pericycle, told you it is present on the inner side of the endodermis, right beta? It is present on the inner side of the endodermis. It is above the phloem in the form of semilunar patches of sclerenchyma. So, this is sclerenchyma here. Is that clear? So, this is the sclerenchyma here. Is that clear? Sure. So, these are going to be the sclerenchyma. So this is sclerenchyma, right? So whenever you will see the double walls here, they are showing the sclerenchyma, okay? Okay. So this pericycle is multi-layered in stem, right? This pericycle is what? This pericycle is multi-layered in stem. Am I clear? What is this pericycle? This pericycle is multi-layered in stem. So in sunflower, it is made up of alternates band of sclerenchyma and parenchyma. So as I said, let's suppose this is sclerenchyma band, this is parenchyma band, sclerenchyma band, parenchyma. So this is a kind of heterogeneous pericycle. So pericycle is multi-layered. Important point it is, beta. So the part of pericycle, right? That's why in the starting itself I have explained it. So the part of pericycle which is in front of vascular bundle, that will be sclerenchymatous. Am I clear? So this is bundle cap. So this sclerenchyma part of pericycle is also called the hard bast, and pericycle is present in front of phloem as semilunar patch. Matlab same point got repeated here. Understood? Understood? So this is what your dicot stem is. Now vascular tissue, obviously it is very easy, right? There is no problem in the vascular tissue part, right? There is literally no problem in the vascular tissue part. We know that there are large number of vascular bundles, right? There are large number of vascular bundles which are arranged in the form of a ring. Okay, so this arrangement of vascular bundle is a characteristic feature of the dicot stem. Now each vascular bundle is conjoined, open and with endark protoxylem. Now in between vascular bundle, radially placed parenchymatous cell, they are the medullary rays. So conjunctive tissue is in the roots, right? So I rectified that part there. So it is what? It is actually the medullary rays. Now these medullary rays, they will help in the radial conduction of water, minerals and other organic substances. So a large number of rounded parenchymatous cells with large intercellular spaces will occupy the central portion of the stem. So it constitutes the pith. Is that clear? Any doubts here? Yes. Any doubts here? Sure. Any doubts here? So this is about the dicot stem. Now monocot stem, in front of it, it is very easy. Why? Because see, this is the basic plan, right? This is the basic plan here. Just look at this part. Now if you will see this, can we not say that just here, firstly let me tell you, the hypodermis is there, that is going to be sclerenchymatous. In the dicot stem it is collenchymatous, but here it will be sclerenchymatous. It will be sclerenchymatous. And what else you have to see? You will see that vascular bundles, they are scattered. Vascular bundles, they are scattered. Is that clear? They are what? They are scattered. Is that clear? So these are the small vascular bundles which are at periphery. These are the small vascular bundle which are at periphery, and there are large vascular bundle, right, which are there in the at the center. Sorry. So vascular bundles are scattered here. So here you will not see that there will be proper, hypodermis, proper this and that. You are not going to see that. Is that clear? Is that clear? Sure. Sure. So you will see hypodermis. That hypodermis is your sclerenchymatous. Then you will see vascular bundle. So this vascular bundle, we know that it is going to be closed type, and this vascular bundle is also covered with the sclerenchymatous sheath. Okay, so these are the points that you have to see here in the case of what? In the case of monocot stem. Okay. Okay, so that slide is deleted, I guess. So this arranged in a ring form, done. So you can just read this part from the NCERT. This is what you are going to find. The scattered vascular bundles are there, right? Hypodermis is this. So this is the comparison. This is a very good comparison table. So hypodermis, it is collenchymatous here. It is sclerenchymatous here. Ground tissue. Here it is properly differentiated into cortex, endodermis, pericycle and pith. Here it is not differentiated, but it is a continuous mass of parenchyma. So star sheath present here, star sheath absent here. Medullary rays are present here. Medullary rays are absent here. So dicot stem में collateral and open, arranged in a ring, secondary growth occur, collateral and closed, scattered in ground tissue, and secondary growth is usually absent. Understood? Yes, please tell me. Understood? Sure? Sure? Quickly tell me, sure? Done, beta? So this is just about the monocot stem. So the next thing is the dicot and the monocot leaf. So now you are already know that what exactly is the venation, है ना? You people know about the venation. So if you know about the venation, yes beta. So if you know about the venation, then this internal structure is going to be very easy. Just see, in the case of dicot leaf, right? So what sort of leaf is there? Dorsiventral leaf is there. First of all, right here you will see that there is going to be the midrib, which will be comparatively thick. Okay, which will be comparatively thick. Is that clear? Then you have what? You have this vein, and then you have what? You have that veinlets. You have that veinlets. Am I clear? Now here in the case of this dorsiventral leaf, you will see that there is what? Upper epidermis, right? And the lower epidermis. So here you will see that upper epidermis will have less stomata, or even they can be absent here, and the lower epidermis will have more stomata, right? Why? Why is it so? Because upper surface, the upper epidermis, which is also known as adexal, and this one is also known as abaxial, right, bache? So here it is in the direct contact of sunlight, so that is why. So if I have to make the diagram and to explain you this, so firstly we know that it is the leaf. So stomatal apparatus will definitely be there in the epidermis. This is upper, this is lower. Now, why do we say it is dorsiventral? Because here, this is the upper epidermis. Here I'll show you the cells. Wait, let me draw this diagram and then done. And obviously leaves will not show the secondary growth. So here there will be closed conjoint collateral vascular bundles are going to be there. Okay, and there will be the surrounding bundle sheath also. So overall, if I have to tell you that what exactly is going on, dekho, in simple words, if I have to explain, so this is going to be your upper epidermis. What is it? It is going to be your upper epidermis. So upper epidermis obviously covered with cuticle here, and what this part is basically the stomata, right? And it is the adaxial epidermis, adaxial surface it is. Are you getting my point? Now, this is the lower epidermis. This is the abaxial one. Here also you will see the cuticle. And in between these two epidermis, this is your mesophyll, right? So you know that the ground tissue of the leaf, we call it as mesophyll. So it is divided into two parts. One is palisade parenchyma and another is spongy parenchyma. One is palisade parenchyma. The another one is what? Another one is spongy parenchyma. So palisade one, it is near to the upper epidermis adaxial. Spongy is near to the abaxial epidermis. Now, if you will see this upper epidermis, you can see here elongated, vertically placed, parallelly arranged, cells are present. And obviously they are fully loaded with what? They are fully loaded with the chloroplast, है ना? Because leaf, what is their job? Their job is to do the photosynthesis, right? Their job is to do the photosynthesis. Am I clear? Yes? So their job is to do the photosynthesis, okay? Okay. Now, when you look at this, they are rounded. And here so they are rounded with large intercellular spaces. See, these are the air cavities also, yeah, air spaces also. So this is a spongy parenchyma. So lower epidermis, more stomata. Upper epidermis, less stomata, or sometimes they can even be absent. Okay? So here in the surrounding, you'll see the bundle sheath is present. So these are the main cells, main things that you have to discuss here in the case of dicot leaf. So let me discuss the points with all of you. Ye dekho, look at the diagram here, right? Upper epidermis, both the sides are having cuticle. Bundle sheath cells are also there, right? They are having the stoma, right? This is that opening stoma. And here you know that this is the epidermal here, lower epidermis, upper epidermis, okay? Lower epidermis, upper epidermis. One more wonderful diagram I would like to show you. Ye dekho. So these are vertically placed, right? Parallelly arranged. So here you won't see the intercellular spaces, but here you will see these air cavities. So this is the abaxial epidermis. So the diagram clear? Yes, everyone. So the diagram clear? Quick. The diagram clear? So let's read the points. Right, let's read the points about it. So see, this is your dicot leaf. It is as per NCERT. So look at that. So the ventral section of the dorsiventral leaf through the lamina shows three main parts. So epidermis is there. So this epidermis, it covers both the upper surface adaxial and the lower surface abaxial epidermis. Okay? And it is having the conspicuous cuticle. What sort of cuticle is there? It is having the conspicuous cuticle. Now, when it comes to the abaxial epidermis, so it generally bears more stomata, right? Abaxial, lower, adaxial, upper. How to remember? DU, Delhi University. So then the adaxial epidermis, right? So this adaxial epidermis, it may even lack stomata. Now, the tissue between the upper and the lower epidermis, what do you call it? You call it as the mesophyll. And this mesophyll, that will contain chloroplast, that will carry out photosynthesis. So you know that there will be palisade parenchyma and the spongy parenchyma. So adaxially placed palisade parenchyma, beta elongated, vertically placed, parallelly arranged cells are there. So lower side will have oval or round and loosely arranged parenchyma. So it is having the, right, so it is from the lower epidermis. Am I clear? So it is having the lower epidermis. Is that clear? Is that clear? So there are numerous, means large air spaces, yeah, air cavities are there between the cells. So vascular system will include vascular bundle, which can be seen in the veins and the midrib. Understood? Right, beta? So the size of vascular bundle depend on the size of vein. So here you know that the veins vary in thickening, right? Because the reticulate venation is there and the vascular bundles are surrounded by a thick walled, right? Thick walled bundle sheath cells. Am I clear? They are surrounded by a thick walled bundle sheath cells. Am I clear here? Right? So the diagram, so in the diagram itself, you can write down all the points, okay? Now, next is the, so dicots, they are dorsally ventral, right? Dicots. So dicots, they are what? They are dorsiventral, है ना? And they are what? Bifacial leaf. Both, two different faces are there, two different sides are there. Now the next is isobilateral. Okay, this is in the case of monocot. So it is isobilateral. It is also known as equifacial. It is isobilateral and it is also known as equifacial, beta, right? Isobilateral and equifacial. So जैसे let's suppose this is the epidermis. Again cuticle, both the sides are same, and here the vascular bundles, right? They will also not show, they will also, they will not show that like thickening is almost same. Done, beta? So see, here no doubt we write it like this, ki this is the adaxial epidermis or upper epidermis. Even in the NCERT it is written like that. But truly, you know that ki here both the epidermis, they are going to be same, right? Because here it is not like that ki upper will have less stomata, lower will have more. No, it's going to be same. But what you have to remember, like few differences are there here. Okay, few differences are there. So here in this case, you will not see the mesophyll is undifferentiated into dicot and into spongy and parenchyma. Right, here you are not going to see this thing. Yes, please. Here you are not going to see this thing ki there will be, you know, the spongy or the palisade parenchyma is there. No, like this is not the case here, right? So I'm making it. So, so roughly I'm making it. Will tell you. And here we will talk about जैसे let's suppose if this is epidermis. So here I will specify only one thing. जैसे देखो, this is the stomata part. So if this is upper epidermis and lower, I told you already. So mesophyll is undifferentiated here, right? Mesophyll is undifferentiated. It is undifferentiated into what? Undifferentiated into spongy and palisade parenchyma, right? It is undifferentiated into spongy and palisade parenchyma. Am I clear? Am I clear here? Yes, please. So here you will see the stomata is there, but in the upper epidermis, you are going to see something very important, that is motor cell or bulliform cells. So that's a recent previous year question as well. So it's an important PYQ that you have to understand. So overall the things are same. Vascular bundles are of same size, right? The stomata, they are equally present on both the epidermis. But here the upper epidermis, this is something that you have to specify, that upper epidermis will show the motor or the bulliform cells. So it is in the monocots, in the grasses you are going to see. I don't know whether you have noticed or not. Do you know about the curling of the grass leaves? Just say, let's suppose if there is the water scarcity, if water is to that grasses, if the water availability is less, right? If there is the water scarcity, if the water availability is less, so in that case what is going to happen, my dear students, that these guard cells, they will lose the water, they will become flaccid, they'll lose the water. So leaf lamina, instead of staying exposed, it will just curl. When the water is available properly, right, these motor cells, in these motor cells, the water will enter, they'll become turgid, and the leaf lamina will be exposed. Right? Is that clear? Is that clear? Done, beta? So isobilateral or monocotyledonous leaf, this is what we have. So this is the anatomy of isobilateral leaf. It is Hanji. So it is similar to that of dorsiventral leaf, right? So here you will see that stomata are present on both the surfaces of epidermis. Mesophyll is not differentiated into spongy and palisade parenchyma. So in grasses, you will see that certain adaxial epidermal cells, right, along the veins, they'll modify themselves into empty colorless cell. So this part here is very important, beta. You will call it as bulliform cells. What do you call it? You will call it as bulliform cells. Am I clear? Right? So when these bulliform cells in the leaves, they have absorbed water, they will become turgid, right? And the leaf surface is exposed. But when they are flaccid due to the water stress, they will make the leaves curl inward, and it will minimize the water loss. Am I clear? It will minimize the water loss. Okay. So the parallel venation in monocot leaf is reflected in near similar sizes of vascular bundle, except in main veins. Understood? So this is about the monocot leaf. This is about their sections, right? And that's it. This is all about this chapter. Even the morphology and the anatomy, both are covered. So here you can see, right, the stomata usually more on lower epidermis, kidney-shaped guard cell in dicots, dumbbell-shaped guard cell in monocots, palisade, spongy, mesophyll is undifferentiated, bulliform cells absent, there they are present. Okay. Okay, so this is the complete snapshot, right? This is the complete snapshot. Just read it, and then let me check the questions. We will have the MCQ practice, and then I will end the class. Okay, let's have some MCQ practice. One minute, please. Just 1 minute, beta. So we will have the MCQ practice, right? Okay. So let's have the MCQ practice. Done? Chaliye, so let's start this MCQ practice, and here these are basically the NCERT fill-ups, okay? They are basically the NCERT fill-ups, the best way, right? The best way to analyze what you have retained so far. So just look at these lines and then let's start. Okay, so what should be the answer? Chalo, try to fill that. Try to fill that. Ready? Just try to fill that. So here you can see that in majority of, right, in majority of dicot plant, the direct elongation of the radicle will lead to the formation of obviously primary root. Yes or no? It will lead to the formation of primary root. So in dash plants, the primary root is short-lived and is replaced by large number of. First of all, you are talking about the monocots, and if I have to be more specific, so we will talk about the grasses. Is that clear? Right? Then in some plants like grass, monstera, and banyan tree. So these are NCERT fill-ups. So pay attention. Okay. So roots arise from the part of plant other than radicle. Is that clear? Right, so it is what? It is other than radicle. So and are called adventitious roots. So root here are present in the region of maturation. Region of maturation. Okay. Then the zone proximal to region of elongation is called again maturation, isn't it? So dash roots of. Chali, now you tell me what is in 6th, 7th or 8th. Quick. Quick. Just tell me what is there in 6th, 7th and 8th. Quick. It's very simple, beta. So carrot and turnip, they are the modification of tap roots. So tap roots of carrot, turnip and adventitious roots of sweet potato. Today itself I told you, get swollen and the stored food. So hanging structure that supports a banyan tree, they are the pillar roots or the prop roots. The stem of maize and sugarcane have supporting roots, and we know that they are the stilt roots or the brace roots. Any doubts? Any doubts? So this is from the morphology. Now I'm giving you some time to see these lines and then fill that up. Quick. And then fill that up. Quick. So just look at it, right? Just look at it and fill it. Quick. Quick. I want everybody to answer the questions. They are easy. Yes. Done, beta? Chaliye. So what should be the answer in some plants such as rhizophora growing in swampy areas? Roots that come out vertically upward. So here they are talking about nematophores. What are they talking? They are talking about nematophores. So the region of stem where leaves are born, right? What do you call them? They are called as nodes. So underground dash of zamican. So it is the underground stem of and it is, we know that it is the corm that we are talking, है ना? So this is the underground stem. So stem tendrils which develops from axillary buds are cylinder and spirally coiled. See, in this way you can revise your entire NCERT. A lateral branch with short internodes and each node. So here you are talking about the pista or matlab lamina is there, pistia and acornia. So there are many examples. My God. Done? Yes, please. Done, beta? I have done something here. So it is Terror of Bengal. Done. So leaves. Sometimes it happens now. We just forgot the spellings. है ना? We just forgot the spelling, that what exactly spelling. Sometimes we just get stuck. So this is happening to me right now. So this is a cornea. So leaves originate from shoot dash. Right, yes, leaves originate from shoot. That is offset, but they are asking about the examples. Right, they are asking about what? They are asking about the examples. So ye dekho, today itself we discussed this. So leaves originate from the shoot, apical meristem, and are arranged in an acropetal order. So a typical leaf consists of three main parts: leaf base, leaf stalk, that is your petiole, and leaf lamina or the leaf blade. Now in dash, the leaf base expand into a sheath covering the stem wholly or partially. You are talking about the monocots here, right? The grasses here. Am I clear? You're talking about the monocots here. You're talking about the grasses here. Am I clear? Right? So particularly you're talking about the monocots here. Now the next part, just have a look. Quick. Just have a look. Quick. Yes. Quick. So in some leguminous plants, the leaf base may become swollen, which is called pulvinous. The recent PYQ it is. Okay, so dash provide rigidity to the leaf blade. Venation provide rigidity to the leaf blade and act as channel like midrib and veins and veinlets. So that's why I have just written the venation here, right? Yeah, chalo, venation is that complete arrangement. I'll say veins. So arrangement of veins and venation is in the leaf lamina of leaf. In the leaf lamina of leaf is termed as venation. Is that clear? Is termed as venation. So leaves of dash plant generally possess. So what should be the answer here, everybody? Statement number 20. Leaves of dash plant, dicot plants, right? Exceptions are there. And parallel venation is the characteristic of most monocots. Now when the incision of lamina reach up to the midrib, breaking it into number of leaflet, it is called, leaf is called compound leaf. What do you call it? You call it as compound leaf. So in pinnately compound leaf, a number of leaflets are present. On a common axis, the rachis, which represents the midrib. Now in dash compound leaf, the leaflets are attached to a common point. So these are palmately compound leaf, right? Palmately compound leaf. Bombyx is the example. Now the next part, dash is the pattern of arrangement of leaves. It is Phyllotaxy. What's that, beta? It is Phyllotaxy. Now in dash type of inflorescence, the main axis terminates in a flower. Quick, it's easy. The main axis terminates in the flower. Guys, quick. Quick. Yes, please. So it is the cymose. It is the cymose. So flowers, if it is cymose, the flowers, then they are in basipetal order. Okay. So in lily, the calyx and corolla are not distinct and they are termed as tepals. Tepals together, what are they forming? They are forming perianth. They are forming perianth, okay? Now, when a flower can be divided into two equal halves in any radial plane, then it is said to be actinomorphic, right? Or we can also call it as radial. So, in based on symmetry, flowers of Cassia gulmohar, it is beta zygomorphic. So see, now it is in a fill-up type thing, right? They can ask you in the MCQ format very easily. So racemose fluorescence, inflorescence may, you know that there will be the acropetal succession. In hypogynous flower, the ovary occupies the highest position. Epigynous flower may the margin of thalamus grows upward and will enclose the ovary completely. Right? So quickly read these statements. Please read these statements. Read these statements and do it quickly. Yes, please, quick. Any doubts here? Any doubts here? Just check it out, read it. Okay, so let's go to the next part. So in hypogynous flower, ovary is said to be, hypogynous mein ovary is said to be superior. What's the symbol? This is the symbol. Plum, rose and peach. You remember? Peri-peri. So it is, flower is perigynous and the ovary is said to be half inferior. The mode of arrangement of sepal and petal in floral bud with respect to each other is known as estivation. When sepal or petals in a whorl just touch one another at the margin without overlapping, as in Calotropis, without overlapping, they are just touching each other. Velvet, hai na? They are touching each other. They are velvet. So if the margin of sepal or petals overlap but not in a proper direction, then it is imbricate. So sterile stamen, staminode. Do check the PYQs. You'll find all these statements there. Right? You are literally going to find all these statements there. So when stamen are arranged, when the stamens are, attached to the petals, they are apipetalous, as in brinjal, or dash when attached to the, and epitapilous, epiphyllous when attached to the perianth, as in the flower of lily. Understood? Done? Yes, everyone, done? Sure? So if the stamen must be joined in one bundle, we know that it is the adelphous condition and it is one bundle, so it is monoadelphous. Right? It is monoadelphous. Chaliye move to the next part. Now see, read these lines and answer these questions. At least solve 100. We'll solve 100 statement and then I'll end the class. Quick. Right, I'll solve, let's solve 100 statement and then let's end the class. Quick. Okay. So a carpel consists of three parts, namely, a carpel consists of three parts. Okay, it is easy. Stigma, style or ovary. Stigma, style and ovary. So stigma is usually at the tip of the style and is receptive surface for the pollen grain. Each ovary bears one or more ovules attached to the flat cushion like placenta. Am I clear? So when more than one carpel is present, they may be free and are called apocarpus. So a seed is made up of seed coat and an embryonal axis. Am I clear? And an embryonal axis. So seed coat has two layers, the outer testa, inner tegumen. Dekha? This is the best way to revise it quickly. Then beta, the dash is a scar. The dash is a scar. The hilum is a scar on the seed coat through which the developing seeds were attached to the fruit. So at the two ends of the embryonal axis are present plumule and the radicle. है ना? What do you have here? You have the plumule as well as the radicle. So in the so plumule as well as what? Radicle. Plumule and the radicle. So in some seeds like castor, the dash formed as a result of double fertilization. Endosperm. The outer covering of endosperm separates the embryo by a proteinaceous layer. Told you about this proteinaceous layer also. It is alluron layer, right? And that is also triploid. Now, epidermal cells are dash with a small amount of dash and large vacuole. So, epidermal cells are compactly packed, yeah, elongated with a small amount of. Hanji, so this is what epidermal spaces are there. Hai na? This is about the epidermal spaces, yes or no? So according to you, what should be the answer here? So according to you, what should be the answer here? Epidermal cells are what? They are parenchymatous with a small amount of cytoplasm and large vacuole. Okay. So now spermatophytes in which phloem have albuminous cells and sieve cells. So here you are talking about the gymnosperms. Chaliye, next part, permanent tissue. Now we have entered in the anatomy part, right? We have entered in the anatomy part. So guys, quickly. Right? So please pay attention here. So this diagram is again from the NCERT. Please observe it carefully. Please observe the diagrams carefully. Right? It's a humble request. Please do not miss the diagrams at all. Okay? Please do not miss the diagrams at all. Understood? So, permanent tissue which are made up of more than one type of cell and these cells work together as a unit. So, you are talking about the complex permanent tissue, xylem and phloem. So, which simple permanent tissue is having long, narrow cells, thick and lignified with a few innumerous pit? You're talking about the, Hanji, sclerenchyma, And sclerenchyma mein what beta? Sclerenchyma mein what? Fibers या sclerids? Fibers. Isn't it? Fibers. Isn't it? है ना? Sclerenchyma fibers they are. So which simple tissue perform functions like photosynthesis, secretion and storage? You are talking about the parenchyma. And which simple permanent tissue forms the major component? Again parenchyma. Meristem which occur in grasses between mature tissue. It is basically intercalary meristem. So here you know that they are marking what beta? Right, the diagram, it is again from the NCERT. This is something that you have to remember. So here B is obviously axillary bud. This one is your, yes, leaf primordia, isn't it? So this one is your leaf primordium because it is singular, and this one is axillary bud. है ना? So next, study of internal structure anatomy, easy it is. So next statement, the meristem which occurs at the tip of roots and shoots and produce primary tissue. Primary shoot or in the root apical meristem. So dash consists of cells which are much thickened at the corners due to the deposition. So here you are talking about the collenchyma. है ना? Collenchyma, this is what you are talking. So cellulose, hemicellulose, pectin, they are deposited. Yes or no? Yes or no? So it is the collenchyma. Here it is what? Here it is the pectin. So this diagram is of, right? We have the fibers as well as what do we have? We have the fibers, and here what do you have? Right? This is the sclerid na? But this A part marked here is lumen. So a mature sieve tube element possesses a peripheral cytoplasm and a large dash. Right? The peripheral cytoplasm is there, and other than that, there is a large vacuole, but it doesn't have the nucleus. So that is why beta it is compared to the mammalian RBC. Now dash is a non-technical term. It is bark. Bark is a non-technical term that will represent all the tissue. This is from the deleted part though, but I believe this is something you should remember. So it is a bark, that is a non-technical term that will include all tissue exterior to vascular cambium. Cambium. So dash is impervious to water due to dash in the Casparian strips. They are impermeable to water due to the suberin deposition. So in dicot stem, bas theek hai, this is it. So this is all about the morphology, right? Right. This is all about the morphology as well as the anatomy. Both the chapters are done and dusted. You guys were, asking for it. So this is all about today. And for tomorrow, what is the important thing? What are the important instructions here? So, I closed it. So go to our YouTube channel, that is Unacademy NEET English, and what you have to do there, you will see that there is a session scheduled for 7 p.m. Okay. So you have to watch this because that will solve all your problems. You'll get to know that how to crack the NEET examination in these, how to utilize these 90 days in a better way. That's what you will learn. Okay. So take care. Bye-bye. See you all soon. Keep studying. If there is any doubt, do let me know in the comment section. So thank you so much, everyone.